A very good morning, everyone. We'll start our session in few minutes. Thank you for your patience waiting. A mesmerizing morning, everyone. I welcome you all to the third International Conference on Clinical and Applied Microbiology organized by BioLeaks. BioLeaks welcomes you to join third International Conference on Clinical and Microbiology to be held on 26th July and Indonesia Hybrid Conference. BioLeaks is a non-profitable professional association which prominently promotes research and development. We at BioLeaks have brought a revolution in the field of worldwide conferences. This conferences brings together the professional wizards and leaders who have explored all the avenues to reinforce the field of life sciences and medicine technology. We conduct events worldwide, which helps in enhancing the skill sets of the people from diverse industries and also forms a common platform for eminent personalities, physicians, researchers, doctors, academicians, professional business figures, and much more. BioLeaks conferences encourages better comprehension about improvements and progressions over the world through global conference with the speed of science and technology. We work with our motto of creating a different tomorrow by organizing conferences and creating network which will help to grow with the help of advanced technology and achievement. Now I would like to welcome our CEO, Mr. Rudrabanu Satpathy, founder and CEO of BioLeaks India. And as keynote speakers of the day, Mr. Neelkant Banerjee, Head of Microbiology, Empire Pharmacy, USA. Dr. I.R. Zoni Kusnadi, Director of Halal Inspection Institute, MS Technical Manager of Central Laboratory of Life Sciences, Lecturer of Food Science and Technology, Universitas Barvijaya, Indonesia. Associate Professor Dr. Suraina Sabari, Associate Professor and Deputy Director, Department of Microbiology, Faculty of Biotechnology and Biomolecular Sciences and Center of Academic Development and Leadership Excellence, University Putra, Malaysia. Associate Professor Dr. Ram Prasad, Associate Professor, School of Life Sciences, Mahatma Gandhi, Central University, India. Professor Dr. Pranabis Chakraborty, UGC Professor and former Director, Department of Pharmaceutical Technology, Maulana Abdul Kalam Azad University of Technology, India. Dr. Raja Chakraborty, ICMR Scientist C, Department of Critical Care Medicine, IPGMER SSKM Hospital, India. Professor Dr. Bharati S, Vice Principal and Dean of Academics, Professor and Head, Department of Microbiology, the Oxford College of Science, India. Professor M.B. Eriba, Professor, Department of Microbiology, Faculty of Science, Kotos State University, Nigeria. I'm so pleased to introduce all our keynote speakers of the day, and I would like to welcome our session chairs of the day, Dr. Mukta Sharma, Professor and Deputy Dean, Department of Microbiology, School of Life Sciences and Technology, IIMT University, Meerut, India. Dr. Mukta Sharma, Professor and Deputy Dean, Department of Microbiology, School of Life Sciences Technology, IIT University, Meerut, India. Dr. Uma S., Assistant Professor, Department of Forensic Science, Bangalore University, India. Dr. Shivavira Kumar S., Assistant Professor, Department of Microbiology, Devagiri University, India. Thank you. Now, I would like to welcome Mr. Rudrabanu Satpathy, founder and CEO of Technorate Group BioLeaks, to address the gathering. Sir, over to you, please.
Thank you, Roshni, for the invitation and introduction. It was really wonderful, but unfortunately, I cannot switch on my cameras because of some technical glitch I faced today. A very good morning, one and all, esteemed dignitaries, my revered thought-provoking speakers, my respected organizing committee members, my loved team of BioLeaks, and my dear presenters and delegates. It's a wonderful morning here at the headquarters of BioLeaks to have you all for third ICCAMS International Conference on Clinical and Applied Microbiology. I express my gratitude to all our faculties, speakers, and session chairs for their generous considerations to spare their valuable time to educate our community through BioLeaks. We ensure the presentation opportunities to our platform may bring a paradigm shift in their research journey, and we are dedicatedly committed to be your partner in promulgate your research and innovations. We are empowering organizations with higher potential for research and innovation, and we foster global collaboration towards research and professional education. We create and empower professional communities and drive them towards advancement of innovation for welfare of the community. We create content in partnership with speakers, faculties, and healthcare leaders that would help in educating the learners and beginners to empower the healthcare community of the globe. We ensure this conference will be a great opportunity for all of us to learn, to share ideas and innovation, and network with each other. I feel this conference will be a valuable experience for all of us, even though organized virtually, but we'll try our level best to connect with each other and plan and organize a conference in physical mode where we can meet in person in upcoming months. I wish everyone a wonderful conferencing ahead. Thank you. Good luck.
Thank you, sir. Thank you so much for your words. Now, I would like to welcome our first keynote speaker of the day, Dr. Ayad Zoni Kusnadi, Director of Halal Inspection Institute, MS Technical Manager of Central Laboratory of Life Sciences, Universitas Barvisia, Indonesia. Sir, over to you, please.
Thank you very much, Mr. Rosini, for the time. And first of all, I would like to thanks to the committee, which is give me an opportunity to share about my experiences, especially in halal. Maybe you are little bit because my presentation actually related to the microbiology or bio-based product, but I'm talking about the halal, related to halal. So my topic this morning is microbial and bio-based halal product. Thing to consider. I am Johnny Kusnadi from Halal Inspection Agency, Universitas Brawijaya, and Department of Food Science and Biotechnology, Universitas Brawijaya, Malang, East Java, Indonesia. Here's my outline for morning presentation. The first one, we are talking about the halal and haram. And the next, we are talking about the microbial and bio-based product. And then why microbial and bio-based product should be halal. And then related to microbial and bio-based product, we have to consider related to the halalness of the product coming from microorganism and many kind of bio-based product. Next, urgency of halal in Indonesia and also in the world. The last one is conclusion. Okay, let's talk about the microbial and bio-based product. When we are talking about the bio-based product, yeah, actually this is wholly or partly derived from material or biological origin, such as plant, animals, enzyme, and microorganisms, including bacteria, fungi, and yeast. And they do not include material that are embedded in geological formation and or fossilized. Okay, before I continue my topic, actually, we have to know about what halal is and what haram is. According to Islam, halal means allowed or lawful. In the case of diet and food, most of them are considered to be halal unless they are specified or mentioned in the Quran and Al-Hadith. How about the haram? Haram means prohibited or unlawful. In Islam, haram food means unlawful to be consumed. So when we are talking about the halal, this is many kind of product which is allowed or lawful to be consumed. But when we are talking about the haram, actually this kind of product prohibited or unlawful to be consumed. In Islam, according to Al-Quran, Al-Baqarah, Surah Al-Baqarah 168, Ya ayuhan nas, kullu mimma fil ardi halalan thayyibah. Wala tattabi'u khutuwati syaiton innahu lakum aduwwum mubin. Mean that all mankind eat from whatever is on earth that is lawful, lawful means halal, and good. In Islamic terminology, good means tayyib, halal and tayyib, and do not follow the footstep of Satan. Indeed, the Satan is a clear enemy for you. So for Muslim, actually, not only for Muslim, Allah, the God Almighty, call to the all mankind to eat what is lawful and good. Yeah, lawful and good. So how about the bio-based product and microbial product? Yeah, according to Al-Baqarah, that we are recommended to have lawful, halal, and good product. Yeah, because Islamic teaching aim to maintain religious safety, soul, mind, offspring, and wealth, and therefore everything which provide benefit for achieving this goal ordered, recommended, or permitted to be done. Being which this is detrimental to the achievement of this objective is prohibited or recommended to be avoided. Yeah. So Islam always asking the people, asking the Muslim to eat which is lawful and also good, yeah, in order to save our religion, our soul, our mind, our offspring, and our way. Yeah. When we are talking about the microbial or bio-based halal product, we are talking there are so many products produced are coming from microorganism and based on many kind of organism. So it is important for us to make sure that the food coming from microbial and bio-based halal, bio-based product should be halal. Yeah. Now, in the modern era, there are so many sources of food coming from microbial and bio-based, yeah, bio-based products. So we have to be careful. As a Muslim, we have to be careful in consuming many kind of product coming from microorganism.
I guess there is a technical glitch. Kindly let us wait for a couple of minutes. In a meanwhile, I'll request all the participants to turn on the camera so that we can have a gallery image. I repeat myself. Meanwhile, I request all the participants to kindly turn on the cameras to have a gallery image. I repeat myself. I request all the participants kindly turn on the cameras to have the gallery image. Thank you. I request all the participants kindly turn on their camera to have a gallery image. Thank you. Thank you all. Just let us wait for a couple of moments so that Sir can sort his technical glitch and join back. Sir has joined. Just give us a moment.
Sorry for interruption of the link. Yeah, I'll continue. Is it clear right now? Okay.
Yes, sir.
I'll continue. Okay. This is the first one. Maybe I want to repeat again. The source of organism. That this is natural organism or genetically modified organism. Yeah. And then the second one is a growth media for inoculum media for plant seeding or media for plant seeding. The content of the growth media, yeah, should be also halal. Yeah. In addition to the organism. Yeah. So when we are talking about the halal product coming from the microorganism or any organism, bio-based organism, bio-based product, the growth media for inoculum should be halal. We have to make sure that halal. And the second one, and the third one is fermentation media. The content of the growth media should also be approved as halal in addition to the microorganism. Yeah. If the fermentation media is not halal or haram, the final product will also haram. And then the next is fermentation process, fermentation equipment such as bioreactor. Yeah. Which fermentation atau bicarator should be appropriate. An assurance is needed that no impurities that violate the halal status of the fermentation remain due to the previous uses of equipment. So when we want to produce the product which is halal, this is the facility, the instrument cannot be used for production, yeah, for non-halal product. So the halal product should be produced in the halal facility. How about the downstream processing? Sometime after the product is already produced, we use of some filters, flocculant, and many kind of substances for downstream processing. All material being used, yeah, we have to make sure that all halal, yeah. Some chemical, flocculant, and many kind of filters should be halal. And also, the last step for production is packaging and labeling. Since the material that contact food directly may migrate to the product. So we have to make sure also that the packaging and the labeling material should be halal. Okay. Next, we are talking about the urgency of halal in the world and in Indonesia. As we know that number of Muslim population, yeah, in Indonesia around 18% to 20% of the world population. Yeah, estimated Muslim population in the year 2030, 2.2 billion, and 26% of world population are Muslim. That's why Muslim need halal product assurance for the product they consume or they use. That's why Muslim, global Muslim, need halal certified product. Whether this is coming from microorganism also coming from other bio-based product. In Indonesia, number of Indonesian Muslim, around 87.2% of the world population, of the population in Indonesia. Estimated Indonesian Muslim in the year 2030 around 239 million people. And Indonesian Muslim also need the product assurance of the product they consume or they use. In Indonesia, now there is a law number 33 year 2014 about the halal product assurance. Article number 4 Indonesian law number 33 2014 that product that enter, circulate, and traded in the territory of Indonesia must be halal certified. So based on law 33 2014, the nature of certification is mandatory in Indonesia right now. Law number 33 2014 had been implemented since October 2019. And then up to now, we also implement our law number 33 2014. That's all about my presentation. And then we come to the conclusion. Halal means low or lawful. Most of the food are considered halal unless it's specifically stated in the Quran or Hadith. To guarantee the halalness of microbial and bio-based product, there are several things that need to be considered, starting from the microorganism, source of microorganism, till the final process. Since the population of Muslim in Indonesia and in the world continue to increase, it is urgent to implement halal guarantee for various products, including microbial and bio-based products. I hope some of the information, is have a benefit for us, and that's all of my presentation. And back to Josia Rossi. Thank you very much.
Thank you, sir. Thank you so much for your valuable presentation. If any of the participants is having any queries, kindly text in the chat box.
If you any question or any comment related to my presentation, please address directly.
Hi, Prof. Can I ask a question?
Okay, please.
I'm Shakira from Malaysia. Assalamualaikum. I have one question. Thank you for the good presentation just now. What is your comment on the impact of advancement in biotechnology on the certification process and assurance of halal product in Indonesia? I think in terms of halal. Thank you, Prof.
Okay, thank you very much for your question. Actually, right now in modern era, there are many kind of organism which is already engineered. We call it genetically modified, genetically engineered or modified. Actually, the modified or GMO or any kind of organism which is already engineered or already modified can be halal certified, as long as the gene being transferred to GMO, or genetically modified microorganism, it's already coming from the halal organism. But if coming from non-halal organism, it will be prohibited. It will be grouped into non-halal or haram product. Actually, in Indonesia, for the halal certification, there are 3 institutions involved in the halal certification. The first one, in national institution, we call it BPJPH, Badan Penyelenggara Jaminan Produk Halal. This is a body, national body for registering, verification, verifying, and of the product want to be certified. The second one, we call it Lembaga Pemeriksa Halal. Halal Inspection Agency, like in my institution also, we call it LPH, Lembaga Pemeriksa Halal. Halal Inspection Body, Halal Inspection Agency. In this institution, the product submitted to get the halal certification should be audited, should be examined, examined from, and we are talking about the halal product, actually many kind on the whole product involved for certification should be halal. Starting from the raw material, and also not only the whole material, raw material, but also any kind of material being used for the production should be halal. It can be so by the certificate, halal certificate, but also it can be checked through the laboratory. That's why in Lembaga Pemeriksa Halal, Halal Inspection Agency, not only conducting audit, related to the document, but also to the facilities, for the production of the product being inspected, to make sure that the whole component, the whole facility is in accordance with the halal, And then the last one is ulama, fatwa ulama. This is the ulama is the expert, in terms of the religion. They know more about the Al-Quran and Al-Hadith. They know about the rule. And the last institution is Komisi Fatwa, Fatwa Commission. This is the institution to judge whether a product is halal or not, halal or haram. If the product agree to be classified into the haram, and then the BPJPH will issue the halal certificate. That is all about the process for halal certification in Indonesia. Actually, any kind of product, can be submitted to get the halal certificate. But the certificate being issued or not, it's depend on all product process and all facilities. Okay, that's all about my question, my answer.
Thank you, Prof.
Okay, thank you very much. Any more?
I guess there are no more questions, sir. Thank you. Thank you so much for your valuable presentation and time.
Okay, thank you very much for giving me opportunity to give presentation and share my experience related to HAL. Thank you very much.
Thank you, sir. Happy to have you here. Now, I would like to welcome our next keynote speaker of the day, Mr. Neelakanta Banerjee, Head of Microbiology, Empire Pharmacy, USA. Sir, over to you, please.
Hello, everyone present in this conference. I'd like to congratulate everyone and I'm sorry for a little delay because I am in EST time zone. So I thought it is a 26 tomorrow. So actual, yes. By the way, I appreciate everyone be a microbiologist, but I believe by reading a book, not become a microbiologist. You have to be practice. So that practice made you become a microbiologist. And the moment it is come in applied microbiology, idea until it is not implemented, there is no use. So implementation of idea is very important. But I can say microbiology, about microbiology is the ocean, is the ocean of knowledge. And we are just identify point fraction of knowledge here. But I like to share one my skin. So I think everyone can see here. So this is the presentation today I can give. That is the contamination control strategy and how application can help you guys to understand where especially microbiology can involve and how your day-to-day life can, utilize for that. First of all, I'm a pharmaceutical personnel. Last 20 years, I'm practicing microbiology in many pharmaceutical companies, especially injectable is my core competent area. But the one thing I learned during this process, contamination control strategy building is very, very important to make a successful sterile injectable. So there's a many regulation is involved in this, such type of operation, especially, we are very, very importantly follow United States of Pharmacopoeia, US FDA, and especially, Annex 1, WHO, and other various regulatory guidelines.
Hello.
And nowadays, microbiology is given very, very importance because it's invisible. And invisible means you have to be more cautious. So in this case, your act, your behavior, your technique, your process, and all those control give the successful sterile medicine. So I can give you these are the thing is given based on your strong foundation, contamination control strategy, classification, qualification, monitoring, and especially your behaviors. If you see this pyramid, top of that pyramid, there is a sterility assurance. We are talking about that. But you cannot achieve that sterility assurance without having the strong pillar or strong foundation. To make that strong foundation, pharmaceutical quality management system is very, very important. And after that, quality thought. That quality thought give you the quality design, facility design, equipment design, personal design, training, gowning, aseptic technique. So these are the things is, give all the control, prevention, how we will create a sterile medicine. But that creation, we supposed to be create a classification and qualification of this area. So our area must be qualified and it should be ISO 5 grade A condition. You know that ISO 5 and grade A is not easy. There is a microbial recovery less than 10. To achieve this type of requirement is not easy. This cleaning is matter, disinfectant is matter, right disinfectant, right practices. So these are the things is matter. So we need to think about that and we need to design about that. And then only you can achieve your, sterility. But before that sterility, we have to be ensure the media fill aseptic process simulation and then, sterilization and all those things, equipment validation. I like to tell you guys one very, very essential and important thing. We see more than 11 years data of FDA. There is a 5 very important thing to make, more 483. The one of the thing, the laboratory design. If you not have proper laboratory design, proper mindset, proper equipment, or proper test method, you cannot detect the right thing. So application and applied microbiology is very, very important in this case. You have to do it. If people are not doing that thing properly, then they are always having the 483 or regulatory citation. So you have to be designed properly. You have to be design your lab. You have to be work your method on that. You have to validate your method, and then you have to be release any pharmaceutical sterile drug. To do such type of sterile drug finished product, not the first priority. We are doing analysis. We will start from the starting material, API, excipient, primary packaging material, and then in process bulk, then finished product, and then followed by stability. So every angle microbiology play very, very essential and important role in sterile pharmaceutical industry. So you understand this thing, how important, because it's directly patient life is matter. So knowledge also matter, experience also matter, but you have to be act on time. That is very, very important in pharmaceutical management, and especially your dedication, your honesty, integrity, transparency. So it's the combination of all subject microbiology, I believe, is a very good in biology. You have to know biophysics, you have to know chemistry, you have to know engineering. It is a combination of all subject. Then only I can say it is a microbiology. I move in next slide. The cost of contamination. If any failures happen, the cost is very high. That cost not only the monetarial, that cost can be the organization brand value. And finally, the patient safety, patient life. So you have to understand, how your method, your act is impacting someone, and directly from the business and patient life. So you have to validate everything. So again, I'm saying what I'm telling earlier, the design also matter, your classification matter, your personal qualification matter, utility qualification matter. In this case, we are using the final utility is the WFI. We are using water for injection, pure steam, compressed air. This must be qualified. Raw material, all must be qualified. All container closure integrity, especially your media film must be successfully done. Then only you can take any sterile batches. So your PPQ, process control, must be happen, and all personnel should be qualified and trained. Personnel play major role in microbiology because that is the source of contamination. Especially microorganism cannot go one area to another area. It's require vehicle. So personnel, particle, movement, these are the vehicle. So we have to be ensure, we have to be protect, we have to be do proper gowning, we have to be do proper training. Now there is a many requirement for the ISO 5. There is a less than 0 requirement for the microorganism, as well as particle. Also we have 3600 particle nearby, but this non-viable particle count is as per ISO requirement. And you can see that is the 3520 is the address in operation, also same requirement, and followed by grade B, grade C, grade D requirement. And X1 specially is saying new guideline implemented, which is talk about contamination control strategy, but I believe there is a lot of scope microbiology. Environment play very important role, but how you ensure your environment is, clean, your environment is meet the acceptance criteria. You do air sampling, you do settle plate, you do contact plate, you do swab, you do, particle, then only you can ensure your environment. So environment play very important role. So you can see the source of contamination. Contamination come from where? Contamination come from your facility. Contamination can come from your equipment. Contamination can come from your process, your material, your utility, your personnel. If you control everything, then only microbial control can possible. So these all are strategy, these all are method required, these all are process required, procedural control required. So you can see this is the schematic diagram I presented here. It's helpful where microbiology play important role in a pharmaceutical industry, especially in sterile manufacturing industry. It's any sterile manufacturing industry. This is the important area where microbiology play important role and microbiological control needed. Obviously, single-use, bag single-use system right now is very demanding and using one-time use, pre-sterilized container, pre-sterilized bag. And during filtration, we are using, a sterile grade filter, 0.22 micron filter to filter the product having many type of filtration, as well as product having terminal sterilization also. So these all are, sterilization process, sometimes gamma irradiation, sterilization, depyrogenation. So many way microbial control is needed. So I'm saying barrier system, there is a restricted access barrier system to where manufacturing happen, as well as the latest system right now is the isolator, which is completely, protected from the environment, where we are not allow any organism or any basically organism not allow because of VHP system, vaporized hydrogen peroxide. But the thing, we are totally cut the human intervention. So two type of isolator to use over there in the industry. One is open isolator and closed isolator. Open isolator, especially where there is a mouse hole is there, that is called open isolator. Closed isolator, there is a no mouse hole. Especially sterility test we are doing in closed isolator. So over there in the testing environment and manufacturing environment, always in the higher classification or equal classification of the manufacturing environment. So we have to be maintain this thing. Then only we can protect the sterility. So I already tell about the single-use bag, single-use system, container closure seal integrity. But again, I am saying is important the aseptic, observe and program. Make sure it's very important, your training, your procedure, and how it is implemented. Replacement training also important. Validation, method development, all are very important to make sure what you are tested, that is validated. So again, I am saying the final touch, sterilization play important role in pharmaceutical, disinfectant, barrier system, filter, cleaning, all are play important role to have a aseptic product. Your periodic train, so finally you get from the failure through the investigation. And I believe this is the short and fast I deliver. If I discuss one topic, it take one day at least. Do you have any question? Anyone have any question?
I request if any of the participant is having any questions, kindly unmute yourself and ask the questions, or else you can text in the chat box. Oh, I guess there are no questions from the participants, sir. Thank you.
Thank you. Thank you,
Thank you.
everyone.
Thank you, sir. Have a good night.
Thank you. Bye-bye. Take care.
Thank you, sir. Now I would like to invite our next keynote speaker of the day, Associate Professor Dr. Ram Prasad, Associate Professor, School of Life Sciences, Mahatma Gandhi Central University, India. Sir, over to you, please.
Okay, thank you. Yeah. Good morning to all of you. Yes, I am audible. Hello.
Yes, sir, you're pretty audible. Kindly go ahead, sir.
Okay. My like topic is recent trends in. First of all, thanks to invitation, and now first I would like to tell our recent advancement in the engineering tailor nanoparticles, what we did and what we doing like that. So the story of that about the fungus, because the Serendipita indica is previously called Perifermospora indica. So this was discovered from the Thar desert of India, and that was my supervisor, Professor Dr. Ajit Verma in JNU. And then we isolated from the root section. Then after we grow in the liquid solid media, then after we work on the like different time interval, what is the sporulation. So this was already published, and why we call Periforma spora indica, because the spore is pear shaped there. So that why called Periforma spora indica, and because Periforma spora indica because they discovered in the India, and this publication is already published and cover page of the journal. So we go for the taxonomic position. They already have discovered by the different like Finley, Severcinals, order, and Finley, Severcinaceae. So where is the in the dendrograph? Here is the position of the Periferma spora indica. That is this is the SSN number and IND India. So the culture deposited in the in the Germany, USA, and they also they have filed the patent in the Germany in 1998. And so this fungus is the multiple importance, and they are the grow on the different culture media, and they use as a large scale multiplication and also the commercial agrobiological products. So the story started from 1998. They have the plant promoter, biofertilizer, bioregulators, bioherbicides, immunomodulators, phytoremediators, biological control against insect and pest pathogens, stress tolerance, and then a stimulator with PGPRs, antioxidant and drug enhancer, enhances anti-aging agent, promote anti-cancer drugs, and many more. So this fungus was plant host promoter, like different types of plant. We also tested on the Arabidopsis like model plant. Then also this xerophytic pinus, they grow in the cypress and they have the control with P. indica. And when the P. indica treated plant is a robust type of and highly like branches and rootings. So also they found they have the plant which has 60 days and 120 days comparatively. They have the more biomass, and also they have the flowering and fruiting takes place yearly. We have by the work like P. indica because they also experiment with the root system. So he promote the root growth. And also under the see the fluorescent microscope here the colonization takes place. So they have these plants also tested with the Bacopa monera, that is called Brahmi. They have the tested in the greenhouse and this plant tissue culture also. And we found that they have the in the root colonized, they have the make the cushion layer of the root and also protect from the plant pathogens. We also tested the antioxidant activity and bacoside. So we found 20-fold enhances the antioxidant and 3-fold enhances the bacoside. We also tested with the Artemisia annua, like they have the used as a anti-malarial drugs. So antibacterial plants, so also lower fever and check the bleeding. So we have also tested and found the 2.5-fold increase the artemisinin content. Then also tested the Osimum basilicum, that is the tulsi. Then also we did experiment with the lab to field condition. So in the chlorophytum, that is called musli, they have the 90% of the survival rate. So we have also tested different plants. They also tested the Curcuma longa, that is the haldi. So not only increase the biomass, but also increase the volatile oil and curcumin content. So also tested like Brus Victoria, Sarati, Tridex plants. So they are the different plants we have the tested. Also, I got the MPCST project. Then we work on the field condition, that is the Artemisia in the central part of the India, and that is called Panchmari. So they tested and found the front row. They have the control, and the treated row is there the in back side of the. So we found that control they have the yearly like yellowish, but the fungus inoculated plant have the robust type and green leaves, means they have the more artemisinin content. And that fungus also not only for go for the biomass, but also go for the culture and then use the culture filtrate of the fungi. So growing the fungi, then filtrate, then we use the culture filtrate of the, then use for the seed germination. So we found the early seed germination also in different plants, also in the plant growth. So, and we also tested in Helianthus sinus, the sunflower. So not only increase the plant biomass, but also increase the seed oil content. And we found they have the fungus because they are beneficial to the root system because they secrete different types of hormones like auxin-like compounds, some enzyme, oligosaccharide, and flavonoids. So already have the published several paper on this. Apart from that, we also work on the nanotechnology. So these are the naturally occurring nanomaterials. And this is the man-made nanomaterials every day we are using. So and they are the cutting edge research area like two things. So our work is maximizing productivity of agriculture because I am working in the plant. So we also tested the different types of nanomaterials with the fungi. Then we found that the biomass of the fresh biomass increased like TiO2, titanium dioxide is 38% increased, CNT 29%. In fact, silver also increased the 16%. We also co-inoculated with bacteria and found they have the nanomaterials have the more colony and bigger in size. So that also did the experiment with the nanomaterials with P. indica inoculated culture filtrate in broccoli plant tunnel and increase the percentage of the plant growth. The reason behind that because nanoparticles provide more surface area. Maybe nanoparticles cross the barrier of the cell membrane and penetrate deep inside the fungal spores. And these may be acts as the direct carrier of the nutrient from media to the help to better absorption and utilization of the nutrient compared to the control. And higher biomass observed in TiO2 may be due to the because TiO2 is well known photocatalytic activity which break down the water molecule and the hydrogen and oxygen. So maybe further growth of the fungus. This is only proposed model how the nanomaterials put into media and how they have the enhanced the growth. This was already patent granted in 2015. So apart from that, I also synthesize the biological synthesis of the different materials, nanomaterials from the different biological, yeah, like plant, bacteria, and fungi and algae. So our work is mainly focused on the microbial system of the nanomaterials. So microbial nanomaterials, because they are microbial nanoparticles develop clean, biocompatible and non-toxic, eco-friendly and cost-effective. This is the green and sustainable route, and our focus is mainly fungi. So why we use fungi? Because easy to handle and process, a large surface area mediated by suitable growth of mycelia leading to higher production rate and also some simple downstream processing. But they have also some disadvantage sometimes because they are the low repetitive rate. Also the pure nanoparticles have the sometimes they are the devoid and capping agent and other biomolecules. So little bit difficult in also the genetic manipulation of the fungal platform. So this is the some disadvantage. So we synthesize from the algae chlorella pyrenoids. They we use the different first we check the growth curve, which is the maximum growth activity. So Thereafter, we use the different time scale, which is the nanoparticles which time has the maximum synthesize of 0 hour, 1 hour, 2 hour, 3 hour, 24 hour, 72 hours. So we can go for the different time observation like wavelength, and here we found the 420 around 450. They have the maximum time for the like synthesis of nanomaterials peak. And we can also go for the same and same EDX, then same and EDX we found they are the silver as the 81% have the silver element have based. And same we also found the silver nanoparticle are spherical in shape and the particle size from 2 to
Torino,
3
questo
nanometers.
adesso non è.
Hello? We also identify the biomolecules responsible for the silver ion reduction. So because they have the biomolecules, they have the cover the actages, the cutting agent. Apart from that, we have also used the different gram-negative and gram-positive bacteria with the different nanoparticles, and we also go for the chemical and biological synthesis of nanomaterials. So we found the biological synthesis nanomaterial is more prominent because they have the biological molecule, they have the protein, they may be act as a more cutting agent. We and then also for the proposed antimicrobial activity because the bacteria that have put in the silver nanoparticles they release and of the silver nanoparticles they inside go and disturb the cell membrane and ROCA and DNA damage and protein degradation. So this is the proposed model we have already published. Then other also have the same fungi we use like plant promoter. We use for the different time interval, and then we found the growth of the fungi. So we have same type. We did experiment like same EDX XRD, and we found the XRD the face centered cubic nature and the and biology. So different type of aspect of use cell extract, biological nanoparticles, and the synthesized silver nanoparticles. So then we have also used the found that had the like a sponging agent like biological and chemical, and we are comparative to the also the L-ascorbic acid. So we found the biological nanoparticles are the more prominent. We also tested the cytotoxicity activities of the chemical synthesized nanomaterials and biological synthesized nanomaterials with a different cancer cell lining like MCF, HeLa, HEPA. And we also propose the model maybe due to the silver ion they have the NADPH dependent nitrate reductase to produce silver, then reductase, then aggregation to peptide. So peptide is kept with the silver molecules. So silver nanoparticles and outside they have the protein cutting agent. So maybe the protein cutting agent this particle has the use in the first stabilizing the silver nanoparticles and also help in the reduction process. We also use the cytotoxicity activity with biological and chemical nanomaterials. So biological nanomaterials treated with the disturbed, this is the control, and this is the biological nanomaterials, this is the chemical nanomaterials. So they also have disturbed the morphology and low density number of compared to control. So we propose the mechanism also the silver nanoparticles, they produce ROS, and they have the oxidative stress to fragment the DNA and chromosome. Maybe some nanomaterials they interact with the macromolecule like protein and lipid and produce apathetic body, and they may be apathetic, they show cancer there. Maybe they have the ROS, so dysfunctional, the mitochondria. So they have the different, maybe some nanomaterials leak across the cell membrane, and maybe they have the loss here of the membrane integrated and breakage the membrane. So they propose the nanomaterial. So also they nanomaterials. They have the tested the gram negative and gram positive bacteria. So they have the and biological nanomaterials compared with. So they have the different like biological nanomaterials have the we found this is the more common. And also we have the tested with the fungal pathogens like Candida albicans, Fusarium, and Aspergillus. So they have the also so the activity like biological nanomaterials is more prominent compared to the chemical nanomaterials. And also they propose the possible mechanism like by they have this stuff the nanomaterials are so already protein damage and some mitochondria denatured and also the cell membrane and DNA damage. So some enzyme also disturbed. So this is the mechanism. So we have acknowledged the like thanks to Burman Laboratory. Here I work on John Hopkins University and also my collaborator in the Kanika Children Innovation Center and also the agriculture part. We are one of the my collaborators in the United States Department of Agriculture and our previous before that I work in the MIT University Noida and also a special thank to convener and organizing secretary for the international conference on the clinical and medical microbiology, biology team members and general audience. So thank you so much. If any question, I am happy to answer. This is my team and there are two PhD students working with me.
If any of the participant is having any queries, kindly unmute yourself and ask the queries, or else you may text in the chat box. I repeat myself, if any of the participants is having any queries regarding the presentation, kindly unmute yourself and ask the queries, or you may text in the chat box. I guess there are no queries from the participants, sir. Thank you. Thank you so much for your valuable session.
Okay, thank you, ma'am.
Thank you, sir. Have a good day.
Okay, thank you.
Before we move on to the next presentation, I request all the participants kindly turn on the cameras for the gallery image. I repeat myself, before we move on to the next presentation, I request all the participants kindly turn on the video for the gallery image. I repeat myself before we move on to the next presentation. I request every participant to turn on the cameras for the gallery images. Thank you. Thank you all. We are so sorry for the delay. Just give us a moment. There is a technical glitch from the keynote speaker side. Okay. Now I would like to welcome our next keynote speaker of the day, Dr. Raja Chakrabarti, ICMR Scientist C, Department of Critical Care Medicine, IPG MER SSKM Hospital, India. Sir, over to you, please.
Yes, am I audible to everyone?
Yes, sir, you're pretty
Okay.
audible, sir.
At the outset, I would like to extend my gratitude to the organizers of today's international event. And I would also like to make a disclaimer that my views are represented as my own and does not represent ICMR, which I work for. I'll be presenting a snapshot of the work that we have been doing at the Department of Critical Care Medicine at IPG MER Kolkata. And hopefully, this should be quite a two-way lecture, which would add some values to some of you, distinguished dignitaries present here. So basically, I'd like, with a background, before starting to share my slides, I'm basically a pharmacologist who has done some work on antimicrobial resistance. So this was one of the major reasons why I chose for this particular program and this conference, where I thought I would be making myself useful. So this is going to be a small but learning experience for you and me. And I hope my slides are visible now.
Yes, sir. But yeah, now it's pretty good, sir. Thank you.
Right. So as I was saying that I'm basically a biomedical scientist who has had his training in pharmaceutical technology and pharmacology. So it's been a decade-long experience of working with ICMR, and we will focus on antimicrobial resistance and antimicrobial stewardship, which are the buzzword in today's time. Now, one of the main essence of what we are trying to do here is that we are trying to gather evidence, because in clinical medicine, in microbiology, and all other biomedical disciplines, one first thing or the first hurdle that faces researchers, scientists, doctors from the Indian subcontinent is that there is a paucity or lack of data, credible Indian data. Now, these data has to be there, like in most of the Western studies, you have a lot of data which backs it up. In our case, in the Indian scenario, that data is, however, very much missing and found wanting. So this is kind of a work which has been envisaged by the Indian Council of Medical Research with some fellows under our supervision and some JRFs, SRFs, and with nurses and data entry operators at the government level. These are all public funded programs, and hence we are accountable to the people and the Union of India for our work. I will be presenting just a bit of what we did over the last couple of years. So quickly moving on. This would be the outline of my presentation. Now, the picture on the right of your screen actually represents a very important work and a milestone in medicine. The very same study that actually promulgated or said that antimicrobial resistance, seen here as AMR, would be one of the largest killers on the planet by 2050. One should not even doubt these predictions from O'Neill and his seminal work in 2016. But it will actually be one of the biggest public health crises. It is a big public health crisis today, and it will have more implications and ramifications in time to come. The background of the study would be one of our previous works, which was published in the Journal of Antimicrobial Chemotherapy. This features authors from 5 studies. This is a multicentric study, which was published in the year 2021, where we found that doctors or clinicians have a very big role in preventing antimicrobial resistance. And this is actually a very important part of the work. So we found that surgical prophylaxis, antibiotics were being given post-surgery for 7 to 10 days without valid rationale. So this is one of the major takeaways. So we needed to start stewardship activities. This formed the groundwork of our present study. This was, in a nutshell, the methodology which was adopted by all the 19 centers that were part of the study. And it was broken down into 3 major activities: the infection prevention control, the antimicrobial stewardship practices, and the surgical antimicrobial prophylaxis activities. And we had to give, we still do, give all the results to the Government of India, the Department of Health Research, as a part of a portal which was created for antimicrobial resistance surveillance program. So the Government of India has allocated grants worth $75 million for one particular year to all the centers and the staff for carrying out this kind of work. So this is going to be all this foreseeable future. Some findings. This is obviously a part of the findings. We have covered around 10,000 beds. This was this presentation dates back to the times when the total beds covered were around 1,800, but that will give you a crux of the matter. This will give you a bird's eye view of the total amount of proportions of the beds that were surveyed, the type of infections that we are talking about, and the patient outcomes. We followed up the patient clinically through a dossier, through a CRF, from the admission till the outcome. The outcome could have been death, a discharge, a leave against medical advice, or a referral. So any of these outcomes were also noted very carefully by the nursing personnel involved in the study. So this brought us to the main question, the crux of the matter. Every hospital, every medical college, for example, the one that I am in, actually must have an antibiogram, and therapy should be on the basis of an antibiogram. The antibiogram directed therapy or the culture-based therapy should be one of the most essential things that a hospital does. A very stable and robust microbiology and clinical microbiology department comes here in this scheme of things. And the antibiogram over at the Institute of Postgraduate Medical Education and Research, which I represent for the year 2022 and 23, is there for you on your screens for your display. This represents the various strains of gram-negative and gram-positive bacteria, as you can clearly see. And these represents the pathogenic strains of bacteria and their resistance profiles. So this is something that we are also trying to sensitize and inculcate amongst the doctors, the surgeons, and the other clinical staff of the institutes to have a look and prescribe according to the antimicrobial antibiogram of the institute. It is imperative to use a culture-based therapy rather than an empirical-based therapy. The empirical-based therapy would look at clinical signs and symptoms of the patient, whereas the culture-based therapy would take into consideration the results and the findings of the culture, the urine culture, the wound culture, the blood swab culture, the CSF culture, the ascitic fluid culture of the patient. So we are trying to do this paradigm shift in this country through our network of 19 institutes, of which we are a very small part. We find that the escape pathogens, the main predominant hospital-acquired infections in the country, occur as a result of these escape pathogens. Something like the Escherichia coli, Klebsiella, Acinetobacter baumannii, Pseudomonas. All of these pathogenic strains have created havoc in this country, have caused deaths, increasing costs, increasing hospitalization stay, and increase the risk of nosocomial infections or hospital-acquired infections. We are trying to find solutions to the problem as directed by the Director General of ICMR in his latest meeting on AMR, on the Lancet series, which we were a participant to. And it is not that ICMR is not taking steps. CDSCO and ICMR are forming a part of a conglomerate along with pharmaceutical companies to have more and more meaningful dialogues, discussions, and way forward for this burning problem. Doctors should come up more to assist or to augment this process, because ultimately they are the persons writing the prescriptions. The PharmD and the M-Pharms are also eligible to write some prescriptions in some states, but not pan-India. That's why the onus is on the medical fraternity of India to stand up and be counted, which they are in bits and pieces, but this is also leading to the problem and the global menace of antimicrobial resistance. This was the antifungal antibiogram. The surgical antimicrobial prophylaxis is also one of the crux of this matter, the essence of it. If I leave today with some key takeaways from this talk, which hopefully you will kind of remember, these are the basic 3 points. the right drug, the right dose, and the right duration, and for the right indication. This is the principle which needs to be followed for every patient being admitted in the ICU or non or the general beds of any hospital. If we do not start acting today, and if we do not find credible research for it today, our parents, our families, siblings could be the next victim of antimicrobial resistance. With this thought, with this very scary thought, we started with the surgical antimicrobial prophylaxis study, this sub-part of our project and program, where we found that the doctors, the nurses, the pharmacists complied submaximally or very less to the guidelines of the country, even in prestigious NIRF ranked institutes of the country. And institutes such as AIIMS has reported to have a lot of occurrence of hospital-acquired infections. Now, if this trend were to continue at a place like AIIMS Delhi, what would be the condition of the public health facilities throughout the country? So we are trying to find out the proportion of such cases of medication error and antibiotic indiscriminate usage, so that we can factor in the determinants and possibly find a solution. Surgical prophylaxis, the surgeons have a very busy schedule, but despite that, they give antimicrobials as prophylactic coverage for gram-positive and gram-negative cocci and rods bacilli for 7 to 10 days, which should not be the case. This should not be a rampant use of antimicrobial at our disposal, which leaves the chance for specific selection and antimicrobial resistance. I was happy to see that microbial evolution was also part of the title of this esteemed congress or conference. That led me to realization that actually microbial evolution is actually greater than human evolution. I say so because microbes have been there for greater, longer periods of history than human beings have inhabited this earth. So, surgical antimicrobial prophylaxis is a very big problem in leading hospitals. The hospital I represent or work in is a 3,000 bedded hospital. So ours is a 3,000 bedded hospital in Kolkata. This is completely government funded. Yet, we don't have guidelines for surgical antimicrobial prophylaxis. What antibiotics is to be given to a lab chole patient, a patient undergoing cholecystectomy for gallbladder stones, is not yet finalized. And this could mean the difference between life and death of the patient. Yes, it is free, but the treatment quality and the treatment outcomes actually determine or get determined from such lackadaisical approach, if I may use that word within quotes, of the authorities in concern. As basic medical scientists, as applied scientists, we have tried our best to convince, persuade the people at high positions in the hospital and in government, but still,
Yes,
research is one thing, and
yes, conference.
the resistance profile to antimicrobials. This is perhaps a busy slide, and this is the resistance profile to antimicrobials at our institute. I also had the data for all India institutes, but the 19 centers, but I don't have permission to, because it is under publication in Springer Nature, and some part in infection and resistance. So this is the resistance profile. Some of it is really scary. Most of the carbapenem resistance, the beta-lactam resistance, the ESBLs, the MRSA are actually cutting down our patients. We are losing patients every day because of antimicrobial resistance. So what can we do? We can look at the data, we can make solutions. One big solution is the wash. Hand washing saves lives. Even the World Health Organization, in one of the recent meetings of the AMR stakeholders from various countries, and the Director General, no less than the Director General of the World Health Organization, Dr. Tedros, said this to us, that please escalate efforts for hand washing in ICU and other units. This should always be the case. Even this should not have any exceptions. The doctors also must wash hands. The nurses also must wash hands. And in meaningful ways, the COVID pandemic taught us this. There is no reason why we should be forgetting this, because AMR could be a bigger problem than COVID combined, because it is projected to kill 50 million of our fellow citizens by 2050. We should not also have to wait till 2050. By 2030, we will have lost many near and dear ones due to antimicrobial resistance and the menace of antimicrobial resistance. These figures are really alarming. And if we do not take notice, if the policymakers don't take notice, then public health, unfortunately, will be in a very big doldrum. Now, some of the solutions, I spoke extensively of the problems. The solutions are also there. We have started antimicrobial prescribing behavior changes, where the pharmacist or the pharmacologist also changes the prescription after consultation and feedback with the clinician or the doctor. In our healthcare system, the doctor obviously assumes the greatest significance, and understandably so. But here, the pharmacist and the nursing staff are also equally in this as an equitable stakeholder. So we have done this WASH criteria. We have done the AWARE classification as proposed by the WHO. We periodically monitor our antimicrobial prescribing behavior of the doctors at our hospital for all 3,000 patients. And we also de-escalate or escalate the prescriptions and the drugs that are being given. Some drugs are unwantedly being given. So this is rampantly misused and irrationally used, abused also in some form of the word. So we have had some changes. When I joined this institute at 2018, this was the scenario of the access antimicrobials, the watch-listed antimicrobials according to the WHO, and this is the current state. The more the greens, the better it is. The less the red, the better it is. So you can quite hopefully see the difference between 2018 and 2023, where there is more amount of access antimicrobials, some amount of the watch list antimicrobials, and a depletion of reserve antimicrobials. This can be taken up as a template, and I'm hopeful that in a country as vast as India, this can be done everywhere. And this could be also replicated, and perhaps is being replicated in other countries with similar problems of AMR. Some multidisciplinary discussions, and from the takeaways, we have created our own hospital antibiotic policy. This gets revamped every June. This year also, we had an issue coming out, and we circulate this amongst doctors so that they are aware of the antibiogram of the institute and the antibiotic to be prescribed at the institute. And we have also in the process of setting up of an antimicrobial stewardship committee of the hospital, a formulation restriction committee, as well as prescription audits. This is also very important in the hospital scenario. Now, quickly winding up, I would like to thank the Indian Council of Medical Research, the Director General, the director of the institute, the medical superintendent and vice principal, the dean of student affairs of the institute where I am based, and all the heads of departments and stakeholders for their unflinching support and guidance. This is my team. These are my two nurses and the data entry operator and the lab technician, who are all public funded by sponsorship from in the Indian Council of Medical Research. In essence, I just end with this, saying that be antibiotic guardian and let's have a safer tomorrow for our family, and let's save everyone, because each and every life is very, very precious. Thank you so much for your kind attention. I would be open for any questions. Gladly. Thank you. Thank you all.
I request all the participants kindly text your queries in the chat box or unmute yourself and ask the queries. If any of the participants is having any queries, kindly text in the chat box or unmute yourself for asking the queries. I guess there are no questions from the, yes, sir, there is a question from the participant. So the question is, sir, may I know the role of a pharmacist in controlling antimicrobial resistance?
Yes, thank you for that question. That very important question. I would like to just say that this is a very important question that you have asked. I would like to just congratulate you for thinking like this. Every part of the healthcare profession, a pharmacist, a nursing staff, and a doctor, they work in a team. In the Indian context, more so because we still fall under the lower middle income country, unfortunately, or the global south. We have this problem of health and economics intruding into each other's space. A pharmacist is supposed to be a drug expert, and a pharmacist should have distinct knowledge of antimicrobials like any other drugs. The dose, the drug, the selection of the drug, the dosage. So in critical care or general words, any pharmacist or pharmaceutical technologist or a PharmD person can also have a very big role in curtailing antimicrobial resistance. They can perform quality audits. They can do prescription audits, prescription surveys, and also give the feedback to the clinicians. It is a team effort, largely a team effort, what we have seen. The enemy is ahead of us. Like in the COVID times, the whole world gathered for the vaccination, the drug discovery process. A large fragment of that was the healthcare workers. But another large part of the world were the patients or the laypersons. Here, the pharmacists are an integral part of the healthcare system. We have ICU pharmacists, which is a concept which is also coming up in India and taking shape. We have a lot of institutes in India, a lot of universities. We have thousands of colleges and hundreds of thousands of students coming up. Why don't you follow clinical pharmacy more? This is my humble suggestion. Try to engage in patient care. I know the scenario is not exactly rosy right now, but we can change the system collectively. The ICMR and the stakeholders are perfectly aware, and more opportunities will be coming up in pharmacovigilance and in the post of new recruits for hospital infection control staff, which will also include pharmacists and hospital infection control nurses. So yes, coming back to that very nice question that you asked, a pharmacist is integrally a patient saver. They check the prescriptions and they are authorized. I repeat my words, they are also authorized to check and possibly consult and correct the prescription for better patient and therapeutic outcomes. So thank you for that question.
Thank you, sir. Thank you so much for your valuable suggestions and words. If any of the other participant is having any other queries, the floor is open for you. There is a question from me, sir, like as a general audience. So what do you think, how a person can know about microbial resistant as a layman person?
Right. Thank you for that question. It's also a very important one. Antimicrobial resistance is very complicated as a subject to understand. I'll try my best to talk from the patient perspective. A person who is undergoing any infective disease, any infection, it could be a urinary tract infection, it could be upper respiratory tract infection. Any of these infections require antimicrobials. They were the magic bullets from the times of Sir Alexander Fleming and Paul Ehrlich. All of these were magic bullets. But bacteria undergo mutations and evolution. They have been here in this planet far longer than human beings, we Homo sapiens, have been. So they will always mutate, as we have seen during the COVID times. The COVID virus also mutated. And we had to outdo it as scientists, as researchers, as patients, as laypersons. We had to outdo the virus. Similarly here, in case of bacterial infections, prophylaxis is the key. We have all heard this term that prevention is better than cure. So hand washing facilities should be there everywhere, not only in healthcare facilities, but also other places, normal offices, where people should do the hand hygiene. You will be surprised that according to the WHO results and findings, a large proportions of lives can be saved by just the hand washing. It's as simple as that. The 6 moments of hand hygiene can actually save lives. So as small as that, we could actually make a difference in someone's lives. It could mean the difference between them contracting a disease, the oral etiquettes, the cough hygiene. Social distancing, I'm sure, is not there anymore after the pandemic, but we have all again assembled back. But still, the hand hygiene, the cough etiquette, all of these can go a long way in stopping the transmission dynamics of this bacterial and viral pathogenesis. So as a common person, one can always do a blood culture if required, obviously after consultation with a physician, or a urine culture for the case of a suspected UTI or a urinary tract infection. This would give us a sense of where we lie in terms of resistance. It can be either susceptible, intermediate resistant, or a resistant bug. So in either of these cases, there are therapies available. We have a lot of cases coming from Delhi and Hyderabad in India. And it has also got to do with the genomics and the evolution of the pathogenic strains also. So it's not always that human beings are anthropogenic causes are causing antimicrobial resistance, but yes, we do contribute fairly highly to our own detriment in society. So I think I answered a part of it.
Yes, sir. Thank you. Thank you so much for your valuable words, sir.
Thank you.
If any other persons is having any queries, kindly let us know. And now I will move to the next keynote presentation of our day. Professor Dr. Bharati S., Vice Principal and Dean of Academics, Professor and Head Department of Microbiology, the Oxford College of Science, India. Ma'am, over to you, please.
Hello, good morning, all of you.
Good morning, ma'am.
I'm audible, right?
Yes, ma'am, you're audible.
Yeah. I'll just share my PPT.
Yes, ma'am. Before that, one of the part. Okay, ma'am, we'll go ahead.
Video. Is my PPT seen?
Yes, ma'am, your PPT is visible, ma'am.
Okay. Let me switch on my video just a minute.
Yes, sure, ma'am.
Yeah. There's the PPT. Yeah. Okay, a very good morning to all the participants. First of all, I would like to thank the BioLeaks for giving me an opportunity and also creating a platform for students and academicians to have a very knowledgeable discussion and also to share, bringing the people around together on the single platform. The topic, that means, since I'm a food microbiologist, I'm a graduate from CFTRI Mysore. So it forces me to talk about the foods, and of course, which will be in relation with your topic of your conference. Now, food is medicine, and the way we eat is the way we live. So it is already realized by many of the people, and most of the people are concentrating on eating right things. So there comes the importance of food as medicine. So there's a saying in our side that the person who knows to eat rarely visits doctors. So we should be knowing how to eat and what to eat. And today I have chosen the topic probiotics as nutraceuticals. So this is the new way of future food supplements. Every research is going on how to improve the gut health and how to be more healthier by eating healthy. What do you understand by nutraceuticals? By definition, nutraceuticals are nothing but a part of the food, or it can be a nutrient component that provides lots and lots of health benefits, including either it is preventing the diseases, or if the disease is already set, it is involved in the treatment of the disease. So that is called nutraceutical. So any food for that matter, it is based on regions and the cultures, what we eat. All over India, we have an amazing array of food, which will surprise us every day to know that everybody has designed their own food. And believe me, everybody is concentrating on healthy foods only. So many a times you would have been wondering whether we are introduced to nutraceuticals in our daily diet. Yes, of course, we have been doing that. See, for allergy reliefs, we are depending on ginkgo biloba. This is nothing but your gymnosperm. It is a plant which is having a very medicinal value. Of the leaf extracts of these are taken as nutraceuticals to get relief from the allergy problems. And when we have arthritis, this glucosamine is used. Glucosamine is an amino sugar, which is actually 2 parts of, I think, polysaccharides. And this is always helpful for our arthritis problems. And cancer prevention, flax seeds, lots of flax seeds are used in our diet now. Green tea, our Indian Prime Minister actually told during COVID to have green tea as a preventive measure and to strengthen our immune system. That's very nice. And cardiac diseases always are fought with the garlic as a very good nutraceuticals. Even it helps in our cholesterol lowering. And a lot of jeera seeds we do take in, especially in India, we do use jeera a lot of in a lot of our cuisines. That is helping us in digestion. And diabetic management is once again done by garlic, and ginseng is used. Ginseng tea we have for immunomodulators. So these are all the nutraceuticals which we are always known to, and we have been using in our daily life and in our daily diet. So if I have to go for classification of your nutraceuticals, the food sources which are used as nutraceuticals are all natural. That is to be underlined. They are not chemically synthesized. And this can be categorized into like your dietary fibers, your probiotics, your prebiotics, your symbiotics, then PUFA, that is polyunsaturated fatty acids, antioxidant vitamins, polyphenol, spices. These are all, many food comes under these different categories. So now in today's talk, I'm going to only concentrate on probiotics, because a lot of intersection of nutrition and pharmaceutical continues to pay a lot of weight to innovative health solutions. So how these probiotics have emerged as a cornerstone of this dynamic field. And these organisms, how they help to be very friendly organisms, we are going to deal with this. By World Health Organization, the definition of probiotics is these are the live microorganisms which, when administered in adequate amounts, in the right quantities, confer a very good health benefit on the host. This is the definition for your probiotics. And a little bit of history on probiotics, if I have to speak on, it all involved these 5 great men in bringing probiotics as our gut-friendly microorganisms. It all started from 18th century, and Louis Pasteur, who was is potentially known, for your father of food microbiology, and he was the one who spoke about the potential benefits of lactobacillus. And Joseph Lister isolated the lactobacillus in 1878. And one gentleman, Henry Tisser, who was a doctor actually, he observed the infants who come to him for, he was a pediatrician, and who comes to him with a lot of diarrheal problems. He cultured the material, your excreta, and he also found that bifidobacteria is less in those infants. Then he understood the gut should have bifidobacteria more in number, that is a Y-shaped microorganisms. He understood that it should be more in number, then only the health of the gut will be very good. Then we have here the Elie Metchnikoff, who is the man behind the concepts of probiotics. He also told that probiotics can be taken orally, and the organisms which are good microorganisms help a lot of our gut health. And we have Shirota, that is Minoru Shirota, who was Japanese, and who was also, he is the man behind introducing Yakult. Yakult is a drink, a probiotic drink, which is available in market now throughout the world, I guess, because even in India it is available, and even in other places it will be available. So he is the CEO of that company, and he is a microbiologist, and he's an MD, and he was the one who first isolated the stable culture of your probiotics. Coming to the definitions of what are probiotics, prebiotics, and symbiotics, let me tell you that these prebiotics are the substance that cannot be digested, but they promote the growth. I'm sorry for that, the recorded version came off. So, just a minute. Definitions of what are probiotics, prebiotics, and symbiotics. Let me tell you that these prebiotics are the substance that cannot be digested, but they promote the growth of beneficial bacteria and probiotics. Okay, these are non-digestible substances that provide a very beneficial physiological effect for the host by selectively stimulating the favorable growth of the limited number of indigenous bacteria like oligofructoinulin, galacto-oligosaccharide, lactulose. These are all the examples of your prebiotics. What I mean to say in simple language is prebiotics, though not digested by the living beings, they serve as a substrate for your probiotics. That is, for those beneficial organisms which are making a colony in your gut region, and they live on these prebiotics. And then, if the number of those probiotics are number of those bacteria. So these are all the examples. Coming to I'm sorry about that. Just a minute. Very healthy benefits on the host. That is what your definition, which I have already told you. Coming to symbiotics, symbiotics is a combination of both probiotics and prebiotics. So substance containing both the above. What is it? For example, yogurt and banana are an ideal example for symbiotic. Okay, then yogurt has organisms, good bacteria. Banana is for serving as a prebiotics. So your bananas, asparagus, all these are prebiotics. So it is a good combination of, and so they make a good symbiotic. One more example is whole wheat bread, that is whole grain bread, along with sauerkraut. Sauerkraut being the fermented cabbage. It's very good food for your, it's a fermented food, that is a probiotic, and whole grain bread is a prebiotic. And those two together make a symbiotic. Soya bean is also a prebiotic. With the soya bean, miso soup, miso is a and these two make up a very good symbiotic. Okay, so there are lots and lots of examples like for symbiotic, which is a combination of both prebiotics and probiotics. Microbiologically speaking, the gut has three principal regions. Hello, I'm sorry. There is a technical glitch which is muting me. Am I audible now?
Yes, ma'am, you're audible, please.
Sorry. So we know that it is always like a stomach picture I have shown there, wherein you have a small intestine and a large intestine and all shown there. This part is very important for us when we are talking about the probiotics, because we are concentrating on gut health. So we have stomach, we have small intestine, and we have colon region, wherein good bacteria and bad bacteria reside. So I have shown the good and the bad bacterial flora, wherein bifidobacteria, Escherichia coli, and lactobacilli are in the good bacterial mode, and Campylobacter, Enterococcus faecalis, and Clostridium, and all these are in the bad bacterial region. So now our duty is to improve or increase the good bacterial count in our intestine, wherein it helps in lot of other functions. So the stomach has very, very low bacterial numbers for the very reason because they are having a high acidic environment, because they will not be growing there. It's not suitable, because we all know bacteria grows in the neutral pH, and since the stomach is having acids formation there for the digestion of the food, it is not that good area for the organisms to dwell there. It is when they come to the small intestine, it is the absorption capacity which makes the bacteria stay there for a longer time. And who are at the risk of, like, why we do need a good bacteria in our stomach always, in our intestine always, is because the very fact, the diet we are following, especially when we talk of Gen Z, the diet which is followed by Gen Z is very, very alarming. Highly processed food is always preferred nowadays, and the fiber foods are vanishing from their diet. Low fiber foods we take because we need everything ready-made and easy, less time consuming, all those. So the highly processed food which are preserved for a longer time, and those type of foods are preferred by Gen Z, and the diet is very poor. That type of people require to concentrate on the gut health. Lot of microbial infections nowadays. We all have gone through the COVID period. We all generally go through the infections, whether it is stomach infection or general infections we have. So that is also prevailing most of the time. And just now the previous speaker spoke about the antibiotic resistance which we are developing now. That's also a major factor for us, antibiotics and other drug intake. What we do, lot of times we take self-medication, and antibiotics are taken more in quantity than required. And that is how we are all developing antibiotic resistance also nowadays. Those type of people who are more prone to this, killing the good bacteria in the human body is also a risk factor. Chronic diarrhea is one of the risk factors. Stress, which we all think it is very, very away from the clinical aspects. No, that is the main thing in the clinical diagnosis now. Lot of stress actually induces a lot of other problems. So that is also a risk factor. Chlorinated water, the water supply nowadays, what we have is not so good and natural nowadays. It is treated, and more of chlorine is there. And the radiation and chemotherapy, not to mention, but 90% of the population are undergoing treatment through radiation and chemotherapy for the cancer, one or the other cancer. So when the lifestyle is like this, wherein the diet is poor, microbial infection is more, antibiotics intake is more, we are suffering from chronic diarrhea, we are having unwanted stress in our life with not proper water to drink, and lot of fight against cancer, the whole of the population comes to the risk population. Now I have just made why are probiotics important for human health. Let me explain that to you in quickly in just a minute. So antibiotics, what is taken into the body is always destroying the gut of the microflora, and the problems are created. And alcohol consumption is also a fashion, and people, youngsters or old people, everybody takes alcohol. So they also, with the stress, to beat the stress, alcohol consumption is happening, and alcohol consumption is actually creating a lot of stress with the health. So emergence of antibiotic strains are there, and these are all the important reasons for having probiotics in human health. They say a happy colon is a healthy colon. Many problems, both physical and perhaps even the mental or the emotional problems, stems from a very poor digestion in the human being. So here is the thing what probiotic does for keeping up your happy colon, and that is immunity is developed, digestion is channelized, vitamin B12 absorption, we have healthier skins, oral health is good, and healing from irritable bowel syndrome, that is a syndrome in our colon, that is we call it as IBS, that is also helping the human health. So this is not required for you. What it does is now we will see what are the probiotic strains which are actually used as probiotics. It's we all know that lactobacillus, bifidobacterium, streptococcus, saccharomyces, these are the organisms which are used. In lactobacillus species, these are the species which are used, like this acidophilus, casein, and reuteri, bulgaricus, plantarum, lactis. These are all from lactobacillus species. A lot of bifidobacterium species like bifidum, longum, breve, infantis, lactis, and adolescens. This is also used, and many of the bacillus species is used, wherein infantis is used, and a very rare one that is non-pathogenic Escherichia coli is used, because what we use in lab is non-pathogenic only. Saccharomyces cerevisiae and Leuconostoc mesenteroides is used, Enterococcus faecalis, Streptococcus thermophilus. These are all the different strains. Not only these, many other strains are all there which are used as a candidate for probiotic strains. So which are the foods containing probiotics? We all are taking these foods in our daily diet. Just we need to know what are the fruits which contain, and try to take more in quantity. Milk is also a probiotic, and soya milk is much better a probiotic than milk. Very familiar with this type of food, and you might have seen the Bulgarians live longer. Like we have people living for 100 years and more than 100 years there, because they all take fermented products as their daily diet. Milk products, sour cream, buttermilk, then your yogurt is a very biggest probiotic. That is why we all have yogurt as a part of our lunch or dinner in our culture. And we have if I have to speak about India, and especially South Indian foods like idlis, dosas, which has become North Indian food also now, doklas, vadas, curry, all these are fermented foods which are probiotics in nature. So the key features of probiotic microorganisms, what should be there as for an organism or for a food to become a probiotic food? They have to be able to maintain a good viability, because they need to get inside the human stomach and they have to stay there in that environment. So a very good viability is the key parameter for an organism to become a probiotic organism. They should be able to survive the passage to the digestive system, from esophagus to your stomach to your colon, all those things. They should be live until they pass through the digestive system. They should be able to attach to the intestinal epithelia and colonize there, stand there, and do good things for immunology. So their sustainability is very important. They carry and they should not carry any antibiotic resistance genes that can be transferred to pathogens. Very important. And they should be able to utilize the nutrients and substrates in a normal diet. They also should be capable of exerting a beneficial effect on the host. That is the key criteria which we take probiotics for. They should be non-pathogenic, non-toxic, and of course, very importantly, non-allergic to human beings. And they also should exhibit antagonistic effect against carcinogens or pathogenic microorganisms. Antagonistic effect means they should outgrow those pathogenic organisms. So effects of probiotics on pathogenic, what will be the effect of probiotic on a pathogenic bacteria? In fact, the probiotics reduces the levels of bacterial endotoxins produced in bacteria, and they inhibit by translocation of bacteria across the gastrointestinal lumen into the bloodstream. Very good job they are doing that. Probiotics disallow the colonization of disease-provoking bacteria through competition, that is antagonism, again, competition for nutrients, immune system, upregulation, production of antitoxins, and also upregulation of intestinal mucin genes. So they increase the nutrient absorption and other things. And here is something on lactose intolerance. You might have heard about this condition in many of the, it is not that everybody are lactose intolerant, but some people are having this problem. They are lactose intolerant. That means they simply cannot digest milk and milk products. So there is a malabsorption in most cases. The most common type of carbohydrate malabsorption is lactose intolerance. You will be surprised, some kids, newly born kids will be lactose intolerant. Very difficult because kids survive on the milk only. So that will be the problem there. And they are associated with the inability to digest lactose into its constituents, that is glucose and galactose. So that is because there are low levels of lactase enzyme activity. So probiotics are commonly used in order to prevent or treat a disease or a condition. This is not actually a disease, it's a condition, and they produce lactase enzyme in the intestine. So those who are having intolerance can be reverted. So how do probiotics prevent antibiotic-associated diarrhea? When we take antibiotics, generally we have diarrhea. That is the diarrhea. That is why we are administered to have vitamin B12 tablets there. Antibiotics disrupt the normal colonic microflora and they consequently alter carbohydrate metabolism and antimicrobial activity in the colon region. This potentially leads to osmotic diarrhea or diarrhea caused by pathogenic bacteria. So here probiotics will prevent this by interrupting either of the potential mechanisms, that is by either making the flora of the gut and ongoing carbohydrate fermentation, or by competitively inhibiting the growth of pathogens. This is the role of probiotics in treating the antibiotic-associated diarrhea. If we have to speak about the immunity with probiotics, let me briefly tell you that both innate immunity and adaptive immunity are taken care here. I have a small, this one animated picture wherein it shows microorganisms on the outer side of the intestinal epithelium. All this I have shown in yellow color is the intestinal epithelium, cells, wherein if at all the in the adhesive region, the probiotics makes an entry into the epithelial cells that will give rise to APC, that is nothing but your antigen presenting cells. Therein the T helper cells are produced. You might have been aware of the B cells and T cells. The both antibody mediated response and cell mediated response are taken care, wherein in antibody mediated response you can see the B cells producing the IgG, IgM and IgE. I have shown the arrows in a different direction to show that IgG and IgM are produced more in number in comparison with IgE, and also IgA is produced. And when you come to cell-mediated response, interleukins and interferons are produced more with the probiotic intake, wherein it will take care of your tumor cells, your viruses, because natural killer cells are produced, macrophages are produced, cytotoxic T lymphocytes are produced, and they will take care of all your infections. In fact, to some extent, even tumor cells are also killed by this. It depends on how you have tamed your gut health. This doesn't happen overnight. You might have seen even the doctors now prescribe, why don't you have probiotics in case when you're having simple diarrhea. I have seen many doctors have started prescribing probiotics for people who are suffering from diabetes. So please do take probiotics for that. So it is having, when we take for 1 month, 2 months, the effect will not have taken it for months together, years together, and you should keep your gut health a very, very prominent one, gut health very nicely, so that all these effects starts. So several mechanisms are there, how lactic acid bacteria will inhibit colon cancer, all those things. How they enhance the host immune response, as I told you in the previous slide. They alter the metabolic activity of the intestinal microflora. They bind and degrade the carcinogens which are there inside. They do not allow them to proliferate. They produce anti-mutagenic compounds, and they alter the physicochemical conditions in the colon region. So you might have seen that many of the good eating habits will always not have more problems with the health regions like this. What is the intestinal discomfort which we regularly, probiotic supplementation activates is your gut protection system, which prevents pathogenic bacteria from proliferating more inside. So these probiotics have shown to reduce the gastric inflammation, which is associated with Helicobacter pylori, which is a very, very notorious organism, and they cause intestinal ulcers and all if Helicobacter pylori is seen in the gut region. So this is also known to reduce allergic symptoms, that is, boosts your immune system and treats the allergies at their source while providing relief from chronic sinus congestion and discomfort. How they modify the structures of antigens itself. They reduce their immunogenicity. Then they reduce intestinal permeability, cannot move at all. So then generation of pro-inflammatory cytokinins that are elevated in patients with a variety of allergic disorders. Some people will have multiple allergies, which needs to be addressed. And I told in cholesterol also. So how this stabilizes blood cholesterol? Studies have actually shown that the probiotics binds to the cholesterol in the lower intestine, which prevents it from being absorbed into your main bloodstream. That itself is a big deal for us. So regular probiotic supplementation was shown to reduce the LDL, that is bad cholesterol levels, as much as 12%. Very, very promising percentage. So this lactic acid bacilli increases the production of short chain fatty acid and inhibits cholesterol synthesis by liver and decreases the blood cholesterol. Bile synthesis is increased, more cholesterol is used up and decreases plasma cholesterol here. And also I was telling about diabetes mellitus, probiotic commonly found in the gut is to secrete a peptide, that is a hormone that releases insulin in response to the food entry into the body. Wonderful. So they can be without in oral extra insulin there. So these probiotic bacteria are isolated from fermented foods, which I told even the Northeast people and the South Indians are more having habit of this. They have demonstrated antioxidant potential studied through different assays. And they have seen that nutraceuticals, the application of probiotic as nutraceuticals with antioxidant potential and may find therapeutic potential in the management of degenerative diseases also. So in oral health also it is seen, in dental health, the probiotics are actually helping the people because they compete for the addition sites when the addition sites. So this is the foods which are available globally, which are which are probiotic in nature. There is Yakult, there is Danone, Nestle company, Amul company has come out with this, and these are all the foods which we have to take it, I feel so. So Indian and global probiotic manufacturers, I just wanted to introduce you. Myrobags in Hyderabad, and Unique Biotechnology, Alliance Biosciences, AgriLife, all these companies are working on probiotics. And the international one is UAS Laboratories and Shandong in China, and Hansen in Denmark, Yakult, Danone, then VSL Pharmaceuticals and Alchem Health Food. These are all working on food, probiotics and prebiotics. And the institutes which are engaged in probiotic research is Central Food Technological Research Institute, Mysore, that is where do I come from, and National Dairy Research Institute in Karnal, Institute of Microbial Technology in Chandigarh, National Dairy Development Board in Anand, and Nestle Private Limited. And these are all the companies and institutes which are actually working on probiotics. So with all this knowledge, let us practice a very good diet, and let us know what to eat and what not to eat, because let food be thy medicine, and let medicine be thy food. Instead of taking lots of medicine, we should try to take lots of good food, which will be always helpful to us. So hope I have I have not exceeded my time, or I am hold just a minute. So this is about the probiotics, which are helpful to our healthy lifestyles. And if there are any questions, I can take the questions. Hello.
Yes, ma'am. Thank you. The floor is open for the questions. If any of the participant is having any queries, kindly text in the chat box or unmute yourself. I repeat myself, if any of the participant is having any queries, kindly text in the chat box. Yes, ma'am, there is a question in the chat box that can probiotics help in decreasing the infections or diseases?
Definitely, probiotics will definitely help, because it is directly acting on your immune system. So infections are kept at a bay when when you are on probiotic diet. That is very much known by this, and also it decreases because you are increasing the good bacteria in you. So when the good bacteria dwells in your intestine, it will fight against the pathogenic microorganisms, which is a cause of, for example, if you have, now also I do tell my students, when they have a slight discomfort in their stomach and stomach upset, they tell, when they come, there is no need to go to a doctor for that. You yourself can be on Yakult or any other probiotic food for that matter. Kefir is a very good probiotic food which we can prepare at home, and these type of fermented products will increase the good bacteria, wherein initially the infectious rage can be decreased, and to some extent it can be cured also. Definitely it will help you in decreasing the infections.
Thank you, ma'am. And there is other question that what about the regulations of nutraceuticals in India?
Yeah, nutraceuticals, this is because all nutraceuticals are natural in this one by nature. So there will be no hard and fast rules on the regulation. The regulatory act should be very lenient. And since it is fiber rich, it is more nutritious, there is no problem with the regulation in India. We can take this because only the chemically made formulas need to undergo all these regulations to be released. So since we are talking about natural things, absolutely no problem from the regulatory sides, from not only in India, globally, I think so.
Okay. And the other question is, can we give Yakult like a probiotic to a child daily? Will it be safe?
See, it depends. For a child of what age we should see. Yes, it is healthier. One per day, regularly one unit per day, because you see that it will be around 20 ml or 50 ml or something like that will become. So one per day is good. I would suggest not to cross the quantities, because since organisms are there, organisms are live organisms in probiotics, and they are also metabolizing. Lot of gas issues will be there if you are overdoing that. So one per day is absolutely a welcome dose, not to cross more than that. It will keep your gut health in a very nice way. So it will be very happy. The child will be very happy to enjoy the good gut health. So it is administered.
Is there any chance of development of drug resistant with the regular consumption?
Definitely not. Definitely not. Because it is not at all a drug given there. It is not a drug given there. So it will increase the immune system. It will tune the immune system in such a way that the infections will be hard to get onto the human body.
As I myself a food technologist, I have a question from my side, ma'am. Like, how are probiotics being integrated into clinical practices?
clinical practices, administering the probiotic food itself is this one way for the clinical people to administer, because we cannot formulate with the live organisms. Since I have been continuously telling it's a live organism which is there in the food, we cannot monitor the dosage of that for the clinical aspects. So that is administering the good fermented foods or a probiotic food itself is an act of clinical social. I think so. There is a effect on your. Am I audible?
Yes, ma'am, you are audible, ma'am.
Saying that your audio is affecting the system or something like that. Okay, so that itself is one way. So incorporating this as a medicine, as a capsule, will altogether not solve our problem. We should not take this as a medicine, because it should be a medicine by itself. We are telling the food itself should be a medicine. So let us not think of again. The pharmaceutical industry should not take up the probiotic as its medicine. That is a big one challenge with us.
Yes, ma'am. And there are many misconceptions probiotics are completely taken in a pharmaceutical way. We can't inculcate through foods. This is the misconception I have seen along with so many of the peoples.
Okay, okay. No, no, no. This is not the medicines. That is right. So it is actually opposition. Like pharmaceuticals should not take this as a, capsule making or a tonic making or something like that. Probiotics is in simple words, I can say that a good food will always keep us away from the medicines. So this is the way for wherein organisms are helping us. So we should know how to use the organisms in good way and take it naturally as our food.
As the terms are just nutraceuticals, everyone thinks those are meant for medicines, not as a food.
Yeah, yeah.
Yes, ma'am. Thank you. Thank you so much for your informative session. If any other people having any queries, kindly let us know. I guess these are.
Thank you. Thank you for giving me this opportunity, ma'am. Thank you.
Thank you, ma'am. Have a good day.
You too. Thank
Thank
you.
you, ma'am. Next, I would like to welcome our next keynote speaker of our day, Professor M.B. Yerima, Professor, Department of Microbiology, Faculty of Science, Sokoto State University, Nigeria. Just give us a couple of moments. Sir is facing technical issues. Just give us a moment, please. We are so sorry for the inconvenience. Let us wait for a couple of moments. Sir will be joining. We are so sorry for the inconvenience. Kindly let us wait for a couple of moments. Sorry for the inconvenience. Kindly let us wait another couple of minutes. Before that, I request every participants to be ready with the presentations as the technical session starts in post the keynote talk. I request all the participants kindly be ready with your presentations. This is to inform you all the participants. There is a technical glitch from our keynote speaker side. So we'll be moving ahead with the technical sessions. Before that, I request all the participants kindly be ready with your presentations. I will call out all the participants' names. Let us know if any of your friends has to still join. Dr. Niranjana J. Mohan, Ajay Kumar, Binat Kaur, Mohammad Ansar CK, Deepika Beniwal, Asmita Lohia, Dr. Ismail Y, Dr. Balarao Akil Raj, Dr. Sridharani, Dr. Mohammad Imtiazuddin, Dr. Sheikh Mohammed Uddin Muzam, Dr. Surya Kala, Dr. Shubhagni Sharma, Dr. Abhaya Sharma, Arunima Banerjee, Dr. Neha, Dr. Binish Gulzar, Ais Zetla, Ajay Kumar, Ravi Maurya, Ishu Garg, Harish, Voleti Vijay Kumar, Mr. S. Shyam Sudarshan, Jyoti Guleria, Jafar Sadek, Dr. Sneha Roy, Nihal, Nurul Shakira, Mohammed Shakrin, Syed Abi. I request all the participants kindly be ready with your PPTs and presentations. We will start technical sessions by sharp 12. I repeat myself, all the technical sessions will start by sharp at 12 o'clock. Now I would like to introduce our session chairs of our day. Dr. Mukta Sharma, Professor and Deputy Dean, IIMT University, Meerut, India. Dr. Yati Hemant Kumar Vaidya, Assistant Professor, Sri Alps N. Patel, Postgraduate Institute of Science and Research, India. Now I would like to request Yati ma'am to have a word with the participants.
Good afternoon, one and all. And I just want to say to all the participants, all the best. Give your 100%, and always give honesty to the science. That's it. Thank you, ma'am.
Thank you. So I request every participant kindly stick on to the time. For every participant, there will be 9 minutes of presentation time and 1 minute for the questionnaire. So I request every participants kindly stick on to the time and go ahead with the presentations. Now I would like to call upon our first presenter, Mr. Ajay Kumar, giving an e-oral presentation on topic of presence of NDM1 among carbapenems resistant enterobacteriaceae CRE strains in urine specimens. Mr. Ajay Kumar, are we having you here? Mr. Ajay? Mr. Ajay, are we having you? Okay, we'll move to our next presenter, Dr. Beenat Kaur, on our topic of computational design approach for multi-epitome vaccine targeting nymphovirus and immunoinformatics. Ms. Beenat Kaur? Ms. Beenat Kaur? I request the participants to respond while we are calling your names.
Yes, ma'am. Beankar.
Yeah. Kindly share your screen and start your presentation, please.
Ma'am, just give me a 2 minute. I have to join from the laptop because it's on the charging. I joined from the mobile. Please kindly give me a 2 minutes. Just I had an
Yes.
exam now.
Yeah, sure, please.
Thank you. Thank you.
Okay, meanwhile, we'll go to our next presentation by Mr. Mohammad Ansar CK on a topic of proportion of biofilm production by Candida species isolated from clinical samples in a tertiary care hospital. Dr. Mohammad Ansar CK.
Hello, am I audible now?
Yes, you are audible. Kindly share your screen and start presenting, please.
Is it visible now?
Yes, sir. Please go ahead, please.
Okay. Good afternoon, all. Today, I am so excited to share with you a fascinating topic that has a significant implications for public health. That is proportion of biofilm production by Candida species isolated from various clinical samples in a tertiary care hospital. Candida, it is the most prevalent fungus that affecting the mucosa, skin, appendages, and the internal organs also. And also the most challenging clinical problem is that increased rate of non-albicans Candida and also the rapidly growing of resistance of Candida species. And also the major key factor is the ability to form biofilms. Biofilms are a complex communities of microorganisms that stick together and adhere to the surfaces. And these biofilms will protect Canada from antimicrobial agents, some host responses, and also from the environmental stresses also. So these much things making them a major contributor to causing severe infections or chronic infections. But not all Canada species are producing biofilms at same rate. So that's why I'm determining the proportion of Canada biofilm production among the Canada species. So these are my main aims and objectives. The primary objective is to estimate the proportion of biofilm production by Canada species, and the secondary objective to estimate the proportion of different species among Canada isolates, and also to estimate the proportion of metabolic active among the biofilm producers. And just look at the materials and methodology. It is a hospital-based cross-sectional study, and I have collected 105 Canada isolates from our Central Microbiology Laboratory in our institution after the approval of Human Ethical Committee. And just move on to the protocol. That is, I have identified the Canada species by the help of Multitoph. I have used the Vitec MS instrument. And after the identification of Canada species, done the total biofilm production estimation by the help of crystal violet microtiter plate assay. And also the metabolic activity is done by the help of MTT assay. MTT is that is 3,4,5 dimethyl thiazole 2I 2,5 diphenyl tetrazolium bromide. It is a tetrazolium reduction test. So by these protocols, I have got better results. And just look at the result part. The distribution of Canada species were collected, which were got as tropicalis, that is 26.7, Canada orthopsilosis, Canada lustane intermedia, Canada cipheri, cruci, glavata, that's like shown in the table. And these are all the different species I got. And just look at the BioGold, 65.7% were biofilm producers, and 34.3% were non-biofilm producers. So from this, I have got the result as majority of the isolates were susceptible to biofilm production. And also let's look at the result by the crystal violet microtiter plate assay. It is a we can result as get the result as biofilm production if it is weak producers or moderate producers or strong producers, just like that. So from here I got the majority of the biofilm producers are moderate biofilm producers. So that is 41%. And just look at the correlation between the metabolic activity and the total biofilm producers. I have already told that biofilm production was determined by the crystal violet assay, and the metabolic activity is determined by the MTT assay. So total 69 isolates were got as total biofilm producers, and out of this only 63 were metabolically active. From this, we can say that only 91.3% among the total biofilm producers were metabolically active, and only 8.7% were non-metabolically active. That means majority of the isolates were metabolically active among the biofilms. So that's why it can cause chronic infections. And also just look at the species-wise distribution of this total biofilm production and as well as the metabolic activity. I have already told that the Canada tropical is the most predominant species, that is 60 out of the 105. And from this 60 Canada tropical isolates, 50 were total biofilm producers. It is a huge, 46 were metabolically active. So from here, I can say that majority of the Canada tropicalis isolates were very susceptible to biofilm production, and as well as it is more prone to metabolically active. And also then followed by Canada albicans. 9 out of total 28 isolates of Canada albicans are total biofilm producers, and 7 were metabolically active. And then next species like Parapsilosis, Oris, Optrosis, these all are similar results go for the biofilms and metabolic activity. That denotes that all the biofilm producers are metabolically active. So from here we can say that Candida tropicalis is biofilm produces as 83.3% and metabolic activity is 76.7%. And in case of Candida albicans, that is biofilm produces are 32.1% and metabolic activity is 25%. And all other species of Candida that showed all biofilm producers were metabolically active in the biofilms. And also, just look at the clinically. I have just categorized the total patients or total samples into the two groups, that is patients with diabetes mellitus and patients without diabetes. So total 56 patients or 56 isolates were patients with diabetes mellitus and 49 were without diabetes mellitus. So from these 56 diabetes mellitus patients, 75% were susceptible to biofilm production, and as well as in case of non-diabetes mellitus patients, that is only 55.1%. So when correlating statistically like chi-square test, I have got the result as more biofilm production was seen in the patients with diabetes statistically with the p-value of 0.032. And also in case of urine isolates from catheterized patients and non-catheterized patients, total 68 patients were catheterized and 13 were non-catheterized. So from these catheterized patients, that is 70.6% were more susceptible to biofilm production, and as well as in case of non-catheterized patients, only 30.8%. So from here also, I can confirm that the more biofilm production in the catheterized patients and with the p-value of 0.0064. So these all are my results, and just look at the conclusion part. According to this present study conducted in our institution, Candida tropicalis was the most predominant species isolated, and as well as it is the more susceptible to biofilm production and as well as metabolic activity among biofilm albicans species than Candida albicans. And also the patients with diabetes mellitus and catheterized patients were more susceptible to biofilm production, and the majority of the isolates were metabolically active metabolically active in the biofilm producers. So in the present study has shed light on the significant variation in biofilm production among Candida species. Biofilm production may be a key factor is the persistence and recurrence of Candida infections. So thank you all of you for this attention. These all are my references. Thank you once again.
Yeah, thank you so much, sir. So I would like to call the next presentation from Binat Karur, in which topic computational design approach for multi-epitope vaccine targeting Nipah virus and immunoinformatics. So we'd like to present it over here now. I request Binat Karur to present this abstract over it.
Kindly allowed to join the meeting from the laptop because it's rejected by the visitor. Hello.
Yeah, ma'am, you're audible. You can go over it.
Okay. May I share my presentation on the on a WhatsApp group, so you can share the WhatsApp and I will present here.
Yeah, okay. You can share with us, ma'am. You can share in WhatsApp.
Kindly check it. I shared on the WhatsApp group to the ma'am. That is Priyanka.
Yeah, sure. It will be done, ma'am. Just give me a couple of moments.
Yeah, sure. Thank you, ma'am, to share the screen. Should I start it, ma'am?
Yeah, you can go.
Okay, thank you. Sorry for the inconvenience.
No problem, no problem.
A greeting to all eminent dignitaries and the research scholars present over here. I'm Vedka, PhD scholars in the LPU University, Fagwada in India. First of all, I would like to thank my supervisor, Dr. Anu Gupta, who guided me the best throughout my research. And I would also like my gratitude toward the panel members, organizers, and the conveners of this conference for providing me the opportunity to present my research work in front of you all. So today's my presentation work is basically on the paper of the computational designing approach to the multi-epitope vaccine targeting the Nipah virus, which basically belongs to the newly emerging death-dealing pathogenic virus, that is that brain-damaging virus, belongs to the family Paramoxydidia. And the transmission mode of the transmission of this virus is zoonotically. First, it was documented into the pigs in the Malaysia in 1999. After that, it was discovered into the mammals and the humans, pigs, and the human-to-human transmission also occurs. It's an acute respiratory illness and the fatal encephalitis. This work is totally the software based on the softwares. And recently in 2019 in Kochi, second outbreak was found where the 23 cases was reported, and out of the 2 was only survived, and the 91% was the cases deaths. That based on the epitope-based vaccine, the risk of the association and the attenuation and the time reduce the time and the cost for the vaccine developer occurs. The Nipah outbreak timelines. First of all, it's documented into the Malaysia. After that, there is no case found into the Malaysia. In pigs, it was founded. And after in 1999, in the Singapore, where the person was working under in the pig farm, and after the discovery of the virus in 1999, in Bangladesh 2001 to 2007, in between this found into the date consumption of the date plants that is infected by the carrier of this virus, that is infected by the virus of the Pelothrus, that belongs to the fruit bat of the viruses. And in India, it belongs to the 2001, 7, 2008, and 2018, 19, and 21 recently it found it. And again in Bangladesh, 2015 to 22, it's recovered. And in Philippines, it is recovered into the 2014. And then again 2008 to 2014 it's recovered into the Bangladesh, and recently into the India, Kerala. The 6 cases was founded in the out of the 6, 2 was dead during the reaching of the hospitals because of the working of the brain is totally damaged. The basically morphology of this virus is considered as a single-stranded virus, which have the highest genomic nucleotide, more than the 18,252 nucleotide consumed into the single-stranded virus with the protein codes, that is the spikes and the spikes that are concluded to the glycoprotein, nucleoprotein, matrix protein, phosphoproteins, and fusion protein, which are responsible for the attachment, recognition, viral application and packing, and the budding and the assembly. After that, the phosphorylation, it contains the non-structural accessory protein viruses, which are responsible for the interferons, suppressing on the regulation of the cell signaling pathways. And then the basic of the discovery, basic of the designing of the multi-tope apivirus is the first step is the retrieval of the protein viruses, proteoma, that is the from the uniport data. That is the uniport data which is used for the retrieval of the proteins. We are included the 8 polypeptides. Out of the 8 polypeptides, we find with the help of the Vaccegen server 2.0, where the threshold value of the Vaccegen server is the 4, and for the viruses, and the antigenic score is more than the 0.5. Only the 7 polyprotein was found to be the for further reconstruction of the vaccine. Then after that, the T cell and the B cell epitopes prediction for their antigenicity, allergenicity, and the toxicity with the help of the immunoepitope database analysis resources, which help to predict the T helper cells epitopes with the help of the Nipah virus marker protein of the MHC1, with the prediction version of the IEDB recommended prediction version to 2020 to 09. And MHC2 molecules also predicted with the help of the same database of the, but the version is the different. After that, the construction of the the prediction of these epitopes, B cell epitopes and the T cell epitopes, which include the MHC class 1 and the MHC class 2 types of the epitopes, passes into the different criteria for determine which are the suitable for the construction of the vaccine. Vaccigenic V 2.0 web server. In this, the B cells are included, those included with the length of the 10 to 18, not more than the 50 of the length was considered. After that, the prediction of the MHC1 and MHC2 molecule, then the prediction of the interferons epitopes, IFE4 PREDs, IF10 PREDs were used to develop the diagnosis. The epitope was selected criteria is it's highly antigenic in nature, non-toxic in nature, non-allergenicity, and the non-homologous to the human body, and the other genomic of the construction. Then after that, the construction of the multi-epitope vaccine against the Nipah virus with the help of the linkers, adjuvants, and the fusion proteins. Adjuvants is responsible for the attack. Sorry. Hello.
Please go ahead with your presentation.
Okay. Then the multi-epitope vaccine constructed with the help of the joining of the linkers. B cell was joined with the KK linkers, and the HTL molecules epitope molecules was joined with the AAY linkers and the cytotoxic T lymphocytes, which are responsible for the production of the suitable vaccine for the using for against the Nipah virus infection. That is the GPG server, GPG linkers. And the 6 fusion protein, that is the His protein, was responsible. Adjuvants, ribosomal protein 50S L7 by L20 was used because it provides the significant importance into the immunogenicity or enhancement of the immunogenic response during the infection of the virus. This multi-epitope vaccine was passes to the different criteria for the physiological properties. This virus was passed to the ProTPRM online tool server, from where we have to check its physiological properties, antigenicity, theoretical PI value, gravity scores, and the index, refractive index, all find into the. After that, the ProTPRM server, it passes into the SPOMA or CyPRED servers, from where we construct the secondary structure of the viruses, and the 3 characters of the beta strand, alpha strand, coils, and the turn structure formation was predicted to the secondary structure with the high accuracy. This accuracy is 89%. And further that, it was passes into the ITRASER server, which is a very effective server for the prediction of the 3D structure of the constructed vaccine. there are some results that I also shared with the construction with the slides. These are the first one is the construction of the vaccine, that is the 395 amino acid sequence with including the linkers and the adjuvants. Only the 16 epitopes was selected out of the 2100 epitopes from the single MSc1 and MSc class 2. And from the B cell epitopes, only the two epitopes were selected for the construction of the vaccine, because if the length of the vaccine is more than the 500, then it will be not considered for the good multi-epitope vaccine. Then further, there is a secondary structure which show the protein sequencing, coils, strands, and the helix form of the different color with the cyprid and the spoma sheet. With the help of the ITSR, there is a structure of the 3D structure of the constructed, that is on the basis of the protein database, where the C value shows the 1.64. That means this constructed vaccine will be the good for the wet lab process. But this is a basically global population coverage. After that, the 3D vaccine construction, the tertiary structure, we will go for the molecular docking and the molecular dynamic simulation and the analysis of the phylogenetic tree. This research is going, but we have to check out with the help of the ELIPRO server, where we find this constructed vaccine cover the word class, basically MHC class 1, which covers the MSc1 and MSc class 2. It goes to the 68.62% of the global population, which will be effective. After the research of this multi-epitope vaccine with the help of the software, we will go for the wet lab experiment so can validate this vaccine. Thank you so much.
Bennett, I just want to know that Vaccinogen use which type of algorithm for scoring?
Basically, it's interrupted your voice. Sorry, ma'am.
I just want to ask that Vaccinogen software use which type of algorithm for scoring?
It's a PAM server that was used for the. There is a lot of the server, but we use the proton, that is basically on the basis of their construction.
Okay. Thank you.
Thank you, ma'am. We are so sorry for the inconvenience that the previous keynote talk we didn't had. Now we will be having that keynote talk by Professor M.B. Erima, Professor, Department of Microbiology, Faculty of Science, Sokoto State University, Nigeria. Professor Erima, are we audible?
Hello, good afternoon, all.
Good afternoon.
Yeah, so can you hear me?
Yes, sir.
Yeah, so first of all, I want to apologize. I have gone through a lot of stress. In my previous location, I think the network internet connection was very poor. So after a lot of frustration, I have to move to another location to join. I'm sorry sincerely for all this. And the time that is left for me now, I think, is just to share an idea based on the topic of presentation. I'm sorry, we are early morning here. I spent the last 4 or 5 hours to prepare some slides to share with you. I know India is 4.5 hours ahead of us. So I think what is important is just to talk about my topic, not necessarily going through all the slides. The topic is actually the good side of pathogens. This is a clinical microbiology conference, and the notion by most of us is that pathogenic microorganisms actually are very, very dangerous, very hazardous. But my presentation will really give a highlight that in the present context of the world, pathogenic microorganisms can also be useful, especially in the production and development of test kits, rapid test kits for diagnosis or diagnostic kits, and also for the purpose of production of vaccine and so on and so forth. So I will just go through to say the infectious microbes actually are found everywhere, a diverse group of organisms, and they include both gram-negative and gram-positive. But I must confess that the soil environment is the reservoir of all microorganisms, including pathogenic microorganisms. So the groups of bacteria, fungi, and protozoa, as well as viruses, are really categorized as representative members or representative groups from which pathogenic microorganisms can be found. You could recall the recent happenings in the world, the COVID-19 pandemic. A single virus was able to hold the whole world to ransom. So actually, I will be talking about the use of these organisms in a positive way, not in a negative way as they are known for. I want you to know that out of these groups, we have the algae. Members of the algae are not typically pathogenic because they don't look for nutrition from their hosts. They are actually autotrophic, they can photosynthesize. So many of them are not likely to be pathogenic. But there are some of them, especially the cyanobacteria. Cyanobacteria is known to be toxigenic. Some species of cyanobacteria are known to be toxigenic. In Africa here, there are instances where cattle, as they move and they drink water from ponds that is contaminated by cyanobacteria, many of them die due to intoxication. So these disease agents, they spread widely, and a recent happening, like I said, with COVID-19 is a clear indication that pathogenic organisms can really make our lives miserable. COVID-19 made our life miserable recently. The whole world was shut down and so on and so forth. So each pathogenic microorganism actually can develop efficient means to inactivate the various layers of host defense and to cross innate as well as adaptive immune defense. So in as much as our body immune system try to fight them, they also devise means of defying that and making sure that they infect our body system and so on and so forth. As far as they are concerned, they also have the right to live. So they never knew they are causing diseases to us. They never knew that they are causing discomfort to us, and so on and so forth. It is we human beings that interpret it in such a way that we are diseased, we are that, we are that. So pathogenic microorganisms are actually seen to many to be hazardous, like I said, and so on and so forth. So in this changing world and the changing scenario, activities of man actually leads to a lot of changes. And the problem of sanitation actually leads to emerging and re-emerging infectious diseases. So pathogenic microorganisms are actually seen to be useful in the production of diagnostic tools, like I said, and other biologics. They can also be used in for analytical purposes, because new antibiotics are still being developed. So the new generation's antibiotic, actually after production, they need to be tested. So in testing them to know whether they are efficacious or not, pathogenic microorganisms are usually used as test organisms to generate data of efficacy, so that they can be confirmed to be useful to man in terms of medication. So I want us to believe that pathogenic microorganisms are not as fearsome, and then if we handle them with caution under laboratory conditions, we can get the best out of them. So I want you to know that also there are recent advances in basic scientific research, and together with the development of molecular techniques, that has not only improved infectious disease diagnosis, but have also provided relevant data about pathogenesis and epidemiology. Because one of the major problems we encounter is the fact that diseases are not properly diagnosed. And the moment diseases are not properly diagnosed, then the choice of drugs for such diseases will be a matter of experimentation. In the developing countries, like in Africa, where I belong, I know most of the problems we encounter in clinics and hospitals is because the medical doctors that are not well trained in medical microbiology will actually begin to treat patients on the basis of experimentation. A situation where a doctor will give you a drug and tell you to go and use it, and come back to tell him or her after 3 days how you feel. It appears the doctor does not even know the actual cause of the disease, and I think this is responsible for a number of deaths. So the molecular techniques that have become more important now, they are very specific and sensitive, and are now indispensable in developed countries as well as even the developing countries, in order to run diagnostic tests. So tests such as FIM-RA, that is biometrics, and gene experts are actually, and of course PCR, are actually used routinely in some of our clinics and laboratories now because of the high precision. Once an organism is identified using this, the level of precision is high, and that can actually guide in the choice of drug for treatment of diseases caused by these things. So the presence of pathogens in the clinical samples after amplification and housekeeping genes provide a signal for testing. You could recall that during COVID-19, when the pandemic subsided, when some countries began to open for immigrants, right at the hospital, PCR tests are usually done to confirm whether a particular passenger is carrying the virus or not. So that is due to the molecular techniques of identification. And in using the PCR, usually you target the virulence genes. Pathogenic organisms are known to have virulent genes and some signature sequences. And these are used as a basis of detecting such pathogens in patients, especially where immediate results is required. So the signature sequences of the housekeeping genes are used. Each and every organism, each and every pathogenic microbe has those signature sequences that are usually used for the purpose of identifying it for diagnostic purpose. So thanks to molecular biology technique, otherwise, many of us will continue to take drugs that have no business in their system. And in fact, this is what is responsible for antimicrobial resistance. The antimicrobial resistance, AMR, is a major killer today. It's a major killer because you can imagine taking a drug that has no activity in your system. And with time, such drugs can be, familiar to the organisms. And by the time you really need them, you may not get their real action. So I think it is important that we should try not to be taking drugs without proper subscription. In Africa and developing countries, that is our major problem. Anybody can go to a drugstore after feeling some signs of headache, whatever, and then he will buy drugs on his own. So it's a very, very serious issue here. I know in India and Indonesia and other parts of Asia, you are well enlightened, and I think such kind of practices are not allowed. Drugs are not sold without doctor's prescription. Just this January, in January, I was in Mumbai. I felt that I had malaria, so I thought I was in Nigeria. I went straight to one drugstore and I wanted the lady to give me anti-malarial drugs. She now requested for doctor's prescription. I said, no, just give me, I think I need to take it. She looked at me and she refused me that drug simply because I was not carrying doctor's prescription. So I think that is a good one. If we can imbibe that in Africa or Nigeria, a lot can happen. So I told you the successes of molecular techniques that are used in diagnosis is because the pathogens can give them something or possess something that can make the test to run. For example, in antibody-based tests, in latex agglutination and immunofluorescence tests, they allow the relatively fast determination of pathogens in clinical samples. ELISA also is used, mostly ELISA assays, in clinical settings for over 50 years to detect antigens. You see, these antigens from pathogens is what give the opportunity for such a test to be carried out. So you can see on this slide, pathogenic microorganisms can develop that. And in the traditional production of vaccines are merely extracts from pathogenic organisms that are used to stimulate the immune system to react. So in the production of vaccine, even the traditional method, not to talk of the mRNA vaccine against COVID, extracts from pathogens are used because they retain their antigenicity and so on and so forth. And it is on that basis that, vaccines are produced. So another molecular technique is the biosensor. A biosensor is a device that contains a bioreceptor that is biorecognized and a transducer substance. This transducer substance can recognize the target analytes derived from pathogens. So whatever you are targeting to analyze on the pathogens to serve at the basis of diagnosis, you can use biosensors to be able to detect that. So again here, it is the pathogenic organism that give the opportunity for such a test to be carried out, because the target analytes they contain makes it possible. So essentially, the bioreceptor recognizes the biological event taking place, and the transducer substance actually convert the biological reaction into measurable signal. So that measurable signals can indicate whether the pathogen in question is present in the clinical sample or not. Then I just want to share a table with you which has been used on antibody substances and using the biosensors to detect some pathogenic organisms, Salmonella typhimurium and Salmonella enterica, Escherichia coli, and Streptococcus epidermidis, Bacillus subtilis, and other things. In the next table, we can see the same biosensors that are used to detect one of the most dreadful strain of Escherichia coli. This strain, Escherichia coli 0157H7, is a strain that is really responsible for a lot of mortality, especially in infants and small children in Nigeria. So it is a pathogen of concern, and it is commonly found in vegetables and so on and so forth. So again here, because of the technology, the biosensor detection mechanism can also be linked to our smartphones. So there are smartphones that are designed to also carry out biosensing activity to detect pathogens and so on and so forth. So this table also is showing you the point of care, because healthcare delivery now is such that it can be carried out seamlessly. You don't need to, leave your home and visit hospitals or clinic that are at a very long distance from your residence. So there are point of cares, there are kits with this diagnostic kits that make things much easier. You can establish point of care, healthcare delivery system. And that was really what happened during COVID-19. Inside airports, you have dedicated points that are actually designated for the purpose of rapid diagnosis. So one of these methods of point of care is use of quantum dots. The use of quantum dots is yet another biosensor technique used in pathogen detection. Genomic RNA from viable Listeria, for example, was extracted, and this assay had a high specificity and gave the results within 15 minutes. So it was tested on real samples like milk and cheese, and also detect foodborne pathogens and other bacteria. This is a mechanism of quantum dots I got from the net, which I want to share with you. And the quantum dots, like it is so-called, the target substance in the pathogen is really quantified, and then that is what forms the basis of detection and so on. And this is the details mechanism of laboratory procedure on how you can carry out the quantum dot test. At the moment, vaccine development focuses on pathogens that represent a global threat to human health. You could recall during COVID-19, vaccine production companies all over the world actually had a good business because we all knew that COVID-19 was not a kind of pathogen that can be handled by traditional vaccines. So many, genomics companies, whatever vaccine companies came up with the mRNA vaccine. So all of these things are made possible because the pathogen, which is the coronavirus in this case, can offer something for detection. Because you could recall the virulence factors in COVID-19, spike protein and 3CL protease. These are products that are encoded by their genetic material, which they use to produce disease. So one can target such proteins and then develop a messenger RNA construct on the basis of which molecular vaccines can be. So if coronavirus do not have such spike proteins and the enzyme which they use, then definitely such detection or diagnosis will not have been possible. And we thank God that those vaccines that were used all over the world, I mean, the mRNA vaccines from different companies really helped to suppress the spread of the coronavirus. Countries have now opened and many of us travel from one place to another. So another promising area of vaccine development actually includes those that are based on DNA, with important progress to enhance their immunogenicity and delivery. You see, as we all know, every disease, whether in humans or in plants, have a genetic basis. So also, pathogenic microorganisms use their genetic machinery to produce substances which they use in causing disease. These substances are usually called virulence factors, and molecular analysis have shown that they are mostly protein in nature. So you see, based on the theory of central dogma in genetics or molecular genetics, most proteins of biological importance are synthesized using the genetic information residing in the DNA. So I think my presentation is a food for thought, is to make us understand really that pathogenic organisms also have their own positive sides. In fact, the world we live in today, a lot of challenges, a lot of diseases that are ravaging mankind, that are posing a lot of threats to man, can only be handled using the molecular techniques. So, and molecular techniques, like I said, are only possible because the pathogenic organisms are capable of providing some targets for procedures to be developed, and then we ride on. So like I said in the beginning, I want to sincerely apologize. I thank you for listening, but I want to apologize again because I was on transit. The slides I actually made for this presentation are not the ones I presented. I just tried to develop these slides because I don't want to fail BioLeaks. I'm scheduled today, so I have to at least say something. So we are looking forward to the next event where we will do justice to whatever topic is given to us. Thank you very much. Shukriya. Remain blessed.
Thank you so much, sir. We are very much happy to have you, even you have faced so many technical issues. And I would like to
Yeah.
ask if any other participants are having any other doubts or question part, you can get clarified with our sir.
Yeah.
I repeat again, if any other doubts or question part, if you are having from your side, you can make unmute and you can raise the question.
I hope they have heard me because you are in a different location. I was talking.
Okay, sir. I think we don't have any question part from our participants. Anyway, thank you so much for being a part of this conference. So now we would like to move on and continue the technical sessions. So now I would like to call from Ajay Kumar to present the topic of presence of NDM among carbons resistance enteromicrobial stains in urine specimens. Ajay Kumar,
Shazali.
Ajay Kumar, you can unmute yourself and you can go over it. Okay. So I think he's not audible now. So we can move on the next participant from Deepika Benival from the topic of antimicrobial activity of bacteriocins from Bacillus subtilis from the NTC CC305 Lactobacillus penetrans, MTCC 2621 Citrobacter fideo, MTCC 1658 and Escherichia coli, MTCC 443 against microbes associated with street food. Yes, Deepika Benival, you can go over it.
And just give me 1 minute.
Sure, ma'am.
I hope my screen is visible now. Hello?
No, ma'am, we can't able to get your screen. Can you please check once?
Yeah, just
Yeah, ma'am, now it's fine. Yeah, now it's fine. You can go over it.
yeah, okay. Thank you. Good afternoon, everyone. My name is Deepika, and I am honored to represent some part of my research on antimicrobial activity of bacteriocin against street food. Excuse me, there is some issue. Yeah. This study evaluates the effectiveness of partially purified bacteriocin produced from Bacillus subtilis, Lactobacillus plantarum, Citrobacter feridae, and E. coli against microbes isolated from street food samples. This research addresses the growing consumer demand for natural biopreservatives and food safety solutions to combat foodborne infection. So let's start with bacteriocin. Bacteriocins are the antimicrobial peptide produced by bacteria which inhibit the growth of closely related or even distant bacterial strain. They are considered promising alternative to traditional antibiotics due to their specificity and the lower likelihood of resistance development. Bacteriocins have potential application in food preservation and medicine as natural preservative and therapeutic agent. Research design we have followed. In this study, we have collected street food samples and raw food samples for microbiological analysis. It involves isolating and identifying bacterial isolates from these samples. Food samples used in our study: raw food sample, orange, pomegranates, broccoli, banana, etc. Street food samples like tomato ketchup, green chutney, pani puri, pickle, chaach, etc. Collection sample gathered from local vendors using sterile instruments and stored in label sterile containers. Experimental design for the analysis of bactericidal activity of bacteriocin on foodborne bacteria, production of bacteriocin, purification of bacteriocin, and analysis of bactericidal activity by well diffusion method. Production of purification from indicator strains like Bacillus subtilis, Lactobacillus plantarum, Citrobacter pyrrhudi, and E. coli. The production is followed by purification of bacteriocin. The cell-free supernatant containing bacteriocin was sterilized and the bacteriocin were precipitated using ammonium sulfate. The purified bacteriocin further used for analysis. Bactericidal activity of bacteriocin against foodborne bacteria was done by well diffusion method. In this method, we use the MHA agar plate inoculated with the bacterial strain, punched with the 3 mm well, were filled with the 75 UL of bacteriocin. After the incubation period, the diameter of zone of inhibition was measured. The result of microbiological analysis of food samples, we identified 65 gram positive and 20 gram negative bacteria from the food samples on the basis of the morphological, microscopic, and biochemical analysis, followed by Burge's Manual of Systematic Bacteriology. This figure represents the percent incidence of gram-positive bacteria. In figure A, we have Bacillus, which is 3%, Streptococcus 7%, Micrococcus 30%, Lactobacillus 2%, Neisseria 2%, and Staphylococcus is 50%. In figure B, the percent incidence of gram-negative bacteria is Proteus 25%, Pseudomonas 15%, Enterobacter 15%, Escherichia 10%, Citrobacter 10%, Moraxilla 5%, and Actinobacter is 20%. This table shows the identified gram-positive and gram-negative bacteria isolate presented in the raw and the street food samples. The bactericidal activity of bacteriocin against foodborne bacteria. In this, the Bacillus subtilis demonstrated the highest bactericidal activity. The Bacillus subtilis was particularly effective against both gram-positive and gram-negative bacteria. E. coli also showed the significant antimicrobial activity. The bacteriocin from the Lactobacillus and Citrobacter pyrrhudi had the lower antimicrobial effect. In this graph, the figure is shown the mean and the standard deviation of zone of bacterial growth inhibition in mm, resulting from the well diffusion method. The bar in the figure represents the mean of zone of inhibition and the error bar represented the standard deviation. The method was conducted in set of triplicates and the significance value p less than 0.001. This table shown the zone of inhibition. Bacillus subtilis between 11 to 18 mm, E. coli 8 to 14 mm zone of inhibition, Lactobacillus plantarum 8 to 14 mm zone of inhibition, Citrobacter pyrrhudi 7 to 13 mm zone of inhibition. These all bacteriocins are compared with the standard antibiotic chloramphenicol, which showed between 14 to 23 mm zone of inhibition. Our findings reveal that the Bacillus subtilis exhibited the highest bactericidal activity, followed by E. coli. Lactobacillus plantarum and Citrobacter pyrrhudi saw moderate effectiveness. The statistical analysis indicated the significant difference among the bacteriocin tested, highlighting the potential use in enhancing the food safety. In conclusion, the study highlights the potential of bacteriocin from Bacillus subtilis and E. coli as the natural antimicrobial against foodborne pathogen. Lactobacillus plantarum Citrobacter pyrrhudi may also contribute to the food preservation when used in the combination with the other method. These findings align with the increasing consumer preference for natural food preservatives. Significance of our research supports the exploration of bacteriocin as safer natural alternative for food preservation. The benefit of using bacteriocin include their effectiveness in the controlling pathogenic bacteria, extending shelf life, and reducing the risk of foodborne illness while meeting consumer demand for the natural preservative. Future research direction should focus on improving bacteriocin production method, exploring synergistic effect and other preservatives, investigation potential resistance, development in pathogens, and developing cost-effective purification techniques. Thank you. These are my reference. Thank you for your attention. I would be happy to answer any question you may have.
Which class of bacteriocin you used? Or you purified?
Yeah, partially purified. And we use them as a crude one.
No, I just want to ask that which class of bacteriocin, class 1, 2, or 3?
It was not specified now because it will be specified after the molecular, because after the class 1, class 2, class 3 will be classified according to their molecular weight.
Okay.
Yeah.
Thank you.
Yeah, so thank you so much for presenting your
Hello,
paper, ma'am.
Deepika.
Yes,
Am I
ma'am.
audible?
Yes, ma'am, you are audible.
Hi. Deepika, I have a question.
Yes, ma'am.
Have you heard about the niacin?
Yes, ma'am.
So, how your bacteriocin is better than niacin?
Actually, ma'am, I didn't work on the niacin. I'm working on the bacteriocin. Actually, my research work is basically on the, I'm working on the potency of bacteriocin and as well as essential oil parallelly together, that which one is more potent. That is why I'm not going on niacin.
You are focusing your work on the essential oil and
Essential oil as well as bacteriocin
as
potency
well as
in the
the
food preservation.
bacteriocin. Okay.
Yeah.
So, how will you purify this bacteriocin in future?
Actually, sir, ma'am, I have purified this bacteriocin by using the ammonium sulfate method. And
Okay.
in future, I am also going through this same technique. But if there is anything better than this for the purification, then I will research and study on that.
Okay.
Yeah.
So, will you use the fermenter for the production, for the commercial production of this bacteriocin?
No. This is basically, I have not used any fermentation. I have just a cell-based culture. And
Okay.
then after centrifugation, I have produced that
You will
cell-free
purify with the
supernatant.
help of the column chromatography and all.
No, I have purified with the ammonium sulfate.
Only ammonium
method.
sulfate. Okay, fine.
Ammonium sulfate method. Yes,
Okay,
ma'am.
thank you.
Hello, Deepika. Can you hear me?
Yes, sir.
Hello, Deepika. Yeah.
Yes,
So I
sir.
just want to make, I want to make an observation, and then so that you can add value, to your presentation. You have been
Okay.
saying Lactobacillus plantarum. Is it another species? Because what I know is Lactobacillus plantarum.
Actually, sir,
So
I have
you are
choose
changing.
this. I have choose these strains from the reference papers, which is already worked on, but their
No,
work
I
is
think,
different.
yeah. So what I'm saying, except if it is a different species, the right name is Lactobacillus plantarum, not plantarum.
Okay. Might be
Yeah.
that is mispronounced.
Secondly, on your data, standard deviation and mean are meaningless when it comes
Which
to your data, the data you generated on zone of inhibition.
which one, sir?
You are presenting mean and standard deviation, right?
Yeah.
So can you tell us the actual statistical tests you use in analyzing the data to conclude that significant differences or otherwise exist? Because you mentioned that there are significant differences between so which statistical analysis did you use actually?
One way ANOVA technique, sir.
Or one way ANOVA?
Yes, sir.
So what was the basis of using one way ANOVA?
Actually, sir, this is between the two groups, which is showing the two groups mean difference.
Two groups?
Mean differences.
Yeah, but that's why I come in. If you are analyzing, if your experimental design is generating data on the basis of two groups, ANOVA is not the appropriate statistical test. Okay, I
Then,
think you better use
okay,
student two test. Yeah, ANOVA
okay.
can only be used when you have more than two groups to analyze in
Actually,
your
sir, I
design.
have more than two groups to analyze because I have 12 bacterial isolates and with the 4 bacterial strains of
Yeah. So
bacteria.
what is the So what are you trying to? Which constitute the treatment? Is it the bacteria, different bacteria, or different concentrations of
What?
bacteria?
What's so I have used 75 UL crude bacteriocin. I didn't work on the different concentration of bacteriocin.
Okay, so the bacteria, the different types of bacteria constitute your treatment. Is that what you are saying?
What's actually I don't
So
understand what you are
Deepika,
trying to ask.
no, Deepika, just get back to your data, get back to your experimental design, try to know the research
Okay.
questions you are trying to answer, and from there
Okay.
you can justify why you use analysis of variance. Thank you.
Actually, sir, this is a small part of my research work. I'm basically working on essential oil, and I just wanted to show which is more potent, like essential oil or bacteriocin. After that, this is just a production and purification and
You are there.
antimicrobial active.
No, you are there. Essential oils and bacteriocins. You compare them, right?
Bacteriocin, yeah.
So, you succeeded in comparing only two,
Actually, sir, this
things.
is
Is
the
that not?
75% work has been done, and few more
No,
steps
get
should.
me the
Yeah.
Deepika. Get me very well. Get me Deepika. The essential oils and bacteriocins are the substances you use to generate the data on zone of inhibition, right?
Yes.
You will use only
But
two
that
of
is
them.
separate data. Essential oil with these isolate, I have. Matlab, that is the different thing, and this is the basically on bacteriocin and the bactericidal activities on the
Okay.
isolates which we isolated from the food.
So my advice: go back to your supervisor, sit down together, look at the design of the work and the research questions. Perhaps
Okay.
you can improve on that. Thank you.
Okay. Okay. Thank you, sir.
Yeah. Thank you so much, ma'am, for your present over it. And next, we would like to call Ashmita Lohia, who is going to present with the topic of analyzing the potential of sugars as biofilm agents towards TP. Yeah, Ashmita Lohia, you can go over it.
Okay, I'm trying to, am I audible to you?
Yeah, it's audible. You can go over it.
Just let me share the screen. Actually, I'm unable to share the screen because somebody else is sharing it right now.
I request Deepika to stop sharing your screen, and it will be fine to share the other person to go over it. Yeah, you can go over it, Sneha.
Sure. Thank you so much. Is my screen visible now?
Yeah, we can see over it. You can go.
Okay, thank you so much. Okay, hi, good afternoon. My name is Asmita, and I'm here to present the work that I did with my team on analyzing the potential of sugars as biofilm agents towards S-typhi. So I would like to start by presenting a data on incident rates per 100,000 of typhoid and paratyphoid fevers. So this data is present country by country for the year 2017. So as can be clearly seen through the figures, typhoid and paratyphoid fever is relatively common in countries with poor water supply and sanitation. The developing countries, especially the South Asians, Southeast Asia, Sub-Saharan Africa, and these are the major cause of disability, especially among the children. As you can see, the cases of typhoid are greater than 50, are almost 50 to greater than 80 in India, and about 40 to greater than 50 in its surrounding countries. And there are almost 0 to less than 1% cases of typhoid in the developed countries like the United States of America, Russia, and others. So I would like to give a brief introduction of what typhoid is. The typhoid and paratyphoid fevers are collectively referred to as enteric fevers since they affect the intestines, and these are caused by systematic infection with Salmonella enterica subspecies Serovar typhi and paratyphi A, B, and C. So Salmonella can produce either typhoidal or non-typhoidal kind of infections. Infection with non-typhoidal Salmonella species produces diarrheal illness, and it rarely causes bloodstream infections, while the typhoid and paratyphoid fevers produce primarily bacteremic febrile illness, whose symptoms are prolonged high fever, malaise, and headache. Without proper treatment, typhoid and paratyphoid fevers can lead to altered mental states, ileus and gastrointestinal bleeding, intestinal perforation, septic shock, and even death. So this is a graph that represents the incidence of typhoid along with the age, and the mortality along with the age. So as can be clearly seen from the data, the incidence of typhoid is higher in the young children, especially in the age of 5 to 9, and so is mortality, while it keeps on decreasing as you age. Several other factors that are found to play a prominent role in typhoid is age, cause, and the income. And the cases of fatality for typhoid fever were found to be 1.89 times higher than that for the paratyphoid fever. So this is the pathophysiology of typhoid fever, where we have a gram-negative bacillus that is ingested through food or water, and it ultimately goes to the intestine, from where it enters into the blood, and the body can hardly do anything to fight the bacterium, and it is then released from the body through feces of a person, and it can come in contact with the water stream, and some healthy person can intake that water, and the same process goes in with them. So if we talk about the treatment of typhoid, it is primarily treated with antibiotics, which are essential in combating the infection. The choice of antibiotic may vary depending on the strain of bacteria and its resistance pattern. The commonly used antibiotics are fluoroquinones, which are often the first choice. Then we have cephalosporins, which are used if resistance is suspected. Then we move to macrolides, which are used in case of resistance. And the last choice for treatment is carbamazepines if the patient is not responding to any of the above mentioned treatments. So several challenges that are faced in treating typhoid are the emergence of multi-resistant strains. The emergence of multi-drug resistant strains of Salmonella enterica Serovar typhi is seen in a lot of cases, and it makes the use of antibiotics difficult. And one of the reasons for the development of multi-drug resistant strains is also the development of biofilms, which causes long-term colonization of bacteria in the host. So the studies have revealed that multi-drug resistant S. typhi isolates are typically high-grade biofilm producers. After infection, the non-biofilm producing bacteria can exhibit bacterial shedding in feces up to 17 days, but the biofilm producers may experience this phenomenon for up to 50 days, which indicates that the duration of carrier state in a patient following infection may be connected to the formation of biofilm by S. typhi. So biofilm is basically a bacterial-initiated matrix, and that envelops a community of bacteria which adheres to a surface on the body. So these biofilms allow the bacteria to survive harsh environments like acid exposure, salt dehydration, metal toxicity, exposure to UV radiations, the body's immune system, basically anything that can be very dangerous for a single bacteria, but has no effect on a bacterial biofilm. So this is the life cycle of the Salmonella biofilm. It has 3 stages: attachment, maturation, and dispersion. During attachment, the cells find a surface to which it can attach. After attaching to a surface, a cluster of cells, they are enclosed by an exopolysaccharide layer, which causes a biofilm, and this stage is called maturation. As is indicated here in the maturation, these are the molecules which play a role in the biofilm. We have cellulose, we have teichoic acid, there's DNA. These are the processes that happen within the biofilm. We have quorum sensing, which enables the cells to communicate with each other within a biofilm. And there is this horizontal gene transfer happening, which is supposed to cause the antimicrobial resistance within the biofilm. So this biofilm actually tries to utilize all the nutrients that it can find in its surrounding. And once it is done with all the nutrients that are there, it just disperses, which is the final stage of the life cycle of biofilm dispersion, where each of the cells of biofilm starts gaining its planktonic stage again. And this causes the chronic stage of typhoid as well. So coming to our topic, the effect of different sugars on biofilm. So sugars are primarily linked to the biofilm formation and their role in pathogenicity. Sugars serve as essential nutrients which can enhance the growth of biofilm formation, and the presence of sugars specifically promotes the synthesis of exopolysaccharide membrane. Sugars, particularly those that are easily fermentable, can enhance the biofilm structural integrity and acidogenicity, which may contribute to the pathogenic potential of the bacteria. So in our study, we took 3 distinct types of sugars, namely sucrose, fructose, and dextrose in the concentrations of 50, 75, 100, and 200 millimolar, sorry. The biofilm was allowed to form over a duration of 5 days. We added 100 microliter of bacterial culture in the 9612 microtiter plate containing nutrient broth along with sugars at different concentrations. The plate was incubated at 37 degrees Celsius for 24 hours, and the culture was replaced with a fresh culture after every 24 hours throughout the duration of experiment to provide optimum growth conditions. And the biofilms were quantified using 0.1% of crystal violet solution for 15 minutes, and it was visualized under a spectrophotometer at 590 nanometer. And the statistical analysis was performed using GraphPad Prism. So these were the results that were obtained. Here in the x-axis, we can see the optical density at 590 nanometer, and y-axis depicts the concentration of the sugars that were used. So the first graph depicts the pattern of biofilm formation in the presence of sucrose on day 1, which is represented by blue colored dots and lines, and the day 5 is represented by orange color. And the same is the case with dextrose and finally fructose, where day 1 is represented by the blue colored line and day 5 is represented by the red colored line. So high concentration of sugars were found to promote the growth of biofilms by S. typhi, and all the sugars showed significant differences in the density of biofilm on the first day and 5th day, having the p-values of 0.0386, 0.0062, and 0.0114 for dextrose, fructose, and sucrose respectively. In case of dextrose and fructose, a continuous increase in the density of biofilm was observed, whereas in case of sucrose, biofilm grew up to a concentration of 100 millimeter, after which a sudden decrease in its density was observed. Out of the 3 sugars that were tested, fructose was found to support the growth of biofilms the most. So the conclusion drawn from it was that the sugars are a crucial dietary element in the typhoid treatment regime, since they give energy to the body and helps it to overcome the weakness. But the concentration and the kind of sugar can also influence the recovery from typhoid due to their direct influence on the growth of S. typhi biofilms. According to our team study, fructose was the most effective in promoting the growth of biofilms, whereas dextrose was least effective. Therefore, consuming fruits rich in fructose, like certain fruits and fruit juices, should be avoided in case of typhoid. So that's it from my end. Thank you so much. These are my references, and thank you.
Hello, thank you. Can I speak?
Yeah, sure.
Yeah, thank you for your presentation. You know, I'm a statistical person.
Yes, sir.
You made some assertion that you got some probability value 0.0. What was the basis of that?
Sir, we applied stats on it. So we had
Yeah.
certain data. We had data for all the days, day 1 to day 5. We just considered the data for day 1 and day 5, and then we applied the student t-test on it. And
Okay.
we saw if this difference was significant or not.
Yeah, so the point I'm trying to raise is that all the probability values you mentioned are actually less than 0.05, right?
Yeah.
Yeah, so I think it's better to say that you just go ahead to say significant differences exist or not on the
Yes.
basis of that level of significance, 0.05.
Yes,
You
sir.
don't have to mention the probability values individually
Okay.
because
Okay,
all
sure.
of them are less than 0.05. If
Okay.
5% level of significance is your chosen level of significance, then you now say, you now make a judgment that significant differences, since they are less than 0.05, it means there are significant differences. You have observed significant differences in the data. So I think that is my
Yes, sir.
contribution. Thank you.
Okay, thank you so much for the feedback, sir.
Thank you.
Yeah, thank you so much, Ashmita Lohia, to present over your paper here. So next, I would like to call Dr. Ismail to present the title of Review on Nutraceuticals and its Impact on Healthcare.
Yeah, good afternoon, ma'am. Can I share my screen?
Yeah, sure, you can go over it.
Ma'am, my screen is visible?
No, still we didn't get any screen.
1 minute. Now it's visible, ma'am.
No, so still we are not getting any screen over it. If not possible, if you're facing some technical issues, you can share through the WhatsApp so that we can help you out by sharing from our side.
Can I share you the screen now?
Yeah, you
PPT
can
now?
share in, yeah, you can share me in WhatsApp.
Yeah. Yeah, I have shared.
Okay. I hope everyone is visible towards the screen, right?
Yes.
Okay, thank you so much.
Yeah, yeah.
Yes, Ismail, you can go over it.
Yeah, thank you, ma'am. Yeah. Yeah, I am an associate professor from Crescent School of Pharmacy, B.S. Abdul Rahman, Crescent Institute of Science and Technology. Today, I'm going to tell a review article on nutraceuticals and its impact on the healthcare properties. Okay, next slide, ma'am. Yeah, from ma'am, next slide. First, the word nutrition from the coin from the 1989 century by Sefton, and the founder and chairman of the innovation of medicine, and how the nutraceuticals are produced and derived from the source and how it provide the extra health benefit towards the nutraceuticals, and how the value is founded. So it depends on how it claims, how the product claims to prevent the diseases, and how to improve our health day by day, or any the processes were delaying again. So it will see the structure and bodies of the function to support our health. Okay, next slide. Yeah. See, first we will see what is the reason towards the nutraceuticals. First, it should increase the number of consumers, and yeah, we should be concerned about the healthcare cost. So, what are the difficulties with pharmaceutical agents means in promoting the healthcare system, or turning the nutraceuticals to improve their health and prevent the chronic diseases. So, and how the more than cure, and how to take the disease into and sent into a good manner, and how to find a solution for that in allopathic medicines, and we can also challenge it in a economically patient. Next slide, ma'am. Yeah. First, the concept of nutraceuticals is we have to need health. That is, we have to need nutrition. We have to take the required nutrition for our health, and what are the remedies we have to take for the sickness or injury. So it prevents and it gives the medical approaches towards us. Next slide. Yeah. We can see difference between pharmaceutical and nutraceuticals. The main difference between these two is that they primarily use, as well as being, pharmaceuticals are developed to diagnose, treat, prevent, and specifically medical condition. So both nutraceuticals and pharmaceuticals are crucial in our healthcare system. So there are some differences of these two. Under nutraceuticals, we can see that nutraceuticals are dietary supplements or food components that provide the health benefits behind the basic nutrition, and it promotes the general health complementary dietary needs towards our wellness support, and it often available over the counter, and it can be purchased within the prescription in retail settings. Okay.
Okay.
Under
मैं इस पे क्लिक करूंगी तो
pharmaceuticals,
डायरेक्ट आ जाएगा
the
क्या यहाँ से?
pharmaceutical medication specifically formulated to diagnose or treat, cure, or prevent, that is ADME process, and we can also diagnose the medical conditions from infection to from the disease. Next slide, ma'am. Yeah. Here are the categories based on the natural source. See, first, under nutraceutical category, it is been categorized into non-traditional, traditional, commercial nutraceuticals. Under that, there is nutrition, nutraceuticals, and recommendation nutraceuticals, and prohibited microscopic organism or chemical constitution. Under chemical constitution, there are three: one is nutrition, herbal, and phytochemicals. And under commercial nutraceuticals, they are functional food and yeah, next slide. Yeah, and it has been classified, and the example has been given based on this. For the example is green tree and fish oil and prohibits based on chemical composition, vitamin C, vitamin D. Example for iron, calcium, amino acid, fatty acid. Example omega-3 and conjugated acid based on mode of action, oxide and toxide, and we can say as inflammatory agents, immune boosters, and all. And based on the health benefits, the heart health, for example, we can say omega-3 fatty acid, or garlic, or bone health, or brain health. Next slide. Yeah. See, and the health benefits of nutraceuticals.
Yeah, okay. Are you ready with your presentation
have
list?
Yeah, ma'am.
Yeah, you can go over it.
Yeah, okay, sorry. Yeah, nutraceutical have gained a significant attention in many years due to the potential health benefits. We can say that is to improve the heart health, to enhance the body health and better brain health conditions, and to boost the immune system or anti-inflammatory system. Yeah, next slide. These are the main roles of nutraceutical in various diseases. That is, first one is cardiovascular disease, next one is oxidative stress, cancer, inflammation, gastrointestinal disease, allergic conditions, and heart disease and osteoporosis. Next slide. Under vitamin A, the health benefits can be antioxidant essential for growth. So it develops the treatment of certain skin disorder, mainly for the skin disorder. Under vitamin E, the antioxidant help forms the blood cells and it boosts the immune system. Under vitamin C, the antioxidant for health bones in owning and it healing the prevent common cold. Under vitamin B1, it helps to convert the food into energy essential of neurological functions. So it can maintain the beta under vitamin B3. It helps to convert food into the energy and it maintains the proper brain to be function. Next is folic acid. It produces the genetic materials of cells, RBC formation, and protects against the heart diseases. Yeah, next slide, ma'am. Ma'am, next slide. These are the diseases prevention and nutrient, that is cardiovascular disease. You can say blackberries, cherries, berries, and apple. For diabetes mellitus, omega-3 fatty acids. This is like fenugreek seeds, cinnamon, and beta melon and pomegranate. Next one is cancer. For this sulfur compound like garlic and all, is beta carbonate and all. Oral disease, ginger or green tea or grapes. For obesity, we can take herbal nutrition like caffeine, fenugreek, vitamin C, and green. For algae, we can take folic acid. Can take folic acid, beta, and vitamin B12. Yeah, ma'am. Next one. On this is recalled of supplements of the nutraceutical. So on March 9, 2023, a recall was announced that 62 brands of omega-3 supplements intimated for cat and dog. So this recall was promoted by a report from customers. So in this, vitamin A toxicity have been after being consumed one of the affected products. So vitamin A toxicity symptoms in pet can vary from mild, such as feelings of illness, loss of appetite, and many severe indication like peeling skin, muscle weakness, etc. On November 12, 2021, a manufacturer of dietary supplements intimated. If in this, this recall may pose a potential health risk to individuals who are allergic or highly sensitive to milk. So this concepts the product that result in the seriousness of life-threatening and allergic reaction. Ma'am, next slide. These are the contamination in nutraceuticals, that is under source, raw material, manufacturing process, packing and storaging, cross contamination, and adulteration. Okay, next one, ma'am. For past few years over the market, the nutraceutical market has a global value of 4.5 billion in 2021, and it is projected to grow as 9% from 2021 to 2030. So the demand of the dietary supplements, how the functional food is expertized to the major drive of market growth, and so there is a growing interest in a medicinal nutrition of cardiovascular disorder and malnutrition, which is expected to further boost the demand of dietary supplements. So this is the bar graph of the overview of the market, how it has been raised. Next slide, ma'am. And regulation and standard in India. So these are the full form, that is Food Safety and Standard Act, Indian Pharmacopoeia, Federation of Indian Chamber of Commerce and Industry, Center of Food Safety and Applied Nutrition, Healthy Food and Dietary Supplement Association, National Institute of Nutrition, Food and Drug Toxicology Research Center, National Nutrition and Monitoring Bureau, Indian Health Food and Dietary Supplement Association, Indian Council of Medical Research, and the Food Safety of India. Next slide. the roles of research and development in what are the roles of research and development in nutraceutical? First of all, we should test the safety, purity, and potency of the product. And next, you have to develop the more effectiveness and efficacy in the product and ingredients used in the product, and to develop the testing method to ensure and verify the consistency of the dosage of the ingredients included in the company's product. And the next one is the new product, either by combining existing ingredients used in the nutraceuticals, supplements, or identifying the new ingredients. Yeah, next slide. Okay, I conclude that nutraceutical provide all the essential substances that should be present in the healthy diet for human. So this study, it can be concluded that various chemical constitution from natural source can be obtained and prepared into various obtained safe, stable formulation for the treatment and diagnosis. And most of the nutraceuticals are from either mineral origin, animal origin, or vegetable origin, or like etc. So these constitution are prepared into dosage forms, such as topical, oral, etc., creams, lotions, ointments, emulsions, and formulations, etc. So this study indicates that demand and consumption of the nutraceuticals are now going on increasing due to safety, therapeutic efficacy, and stability of formulation. Yeah, next slide. Next slide. Yeah, these are my references which have been taken from the article. Yeah, ma'am, next slide. Yeah, thank you. And I thank Clinical and Applied Microbiology and BioLeaks for giving me this opportunity to present in this international conference. Once again, thank you.
Yeah, thank you so much, sir. So I just want to call the next presentation is done by Dr. Baliro Akhil Raj in the topic of antibiotic resistance airborne bacteria and their multi-drug resistance pattern at a tertiary care hospital.
So very good afternoon. Is my screen visible?
Yes, Akhil, is it visible? Yeah, I can see it.
Good afternoon to all the delegates and dignitaries. I'm Dr. Balero Akhil Raj, Junior Resident, Department of Microbiology, GMC Nalgonda. My topic of presentation is antibiotic-resistant airborne bacteria and the multi-drug resistance pattern at the tertiary care hospital. Nosocomial infections are infections occurring in patients in a hospital or other healthcare facility in whom the infection was not present or incubating at the time of admission. Hospitals are reservoir of microorganisms, many of which are multi-drug resistant and are an increasing problem to infection treatment. In view of this, the microbiological quality of air can be considered as a mirror of hygienic condition for hospital wards. A cross-sectional study was conducted in a hospital from Jan to May 2024 in various wards that includes gynecology and obstetrics OTs, pediatric ICUs and OTs, surgical ICUs and OTs, post-operative wards and medical inpatient wards of tertiary care hospital for assessing bacteriological quality of indoor air and to determine the antibiotic susceptibility of the isolates. Air samples of the selected wards were collected using slit air sampler, that is the most latest version, air Petri sampling system Mark II, and sample processing and antimicrobial susceptibility testing were done following conventional bacteriological techniques as per CLSI guidelines. So here, this is the air Petri sampling system Mark II, where we can see the impactor. This inside the impactor will keep the media plate, and through internal pump for 10 minutes, there will be 10 liters of air which will be sucked in. And if there are any organisms which are present in the air, those will be trapped inside the impactor, the plate which is kept inside the impactor. After incubation, overnight incubation at 37 degrees C, on the plate, we can see 378 wells. And if there is an organism growth, that will be captured or grown in those wells. We can see various isolates of organisms which were grown on the nutrient agar plates. Next, the evaluation of the result. The number of colony forming units that are counted on the Petri dish after appropriate incubation needs a statistical correction. then related to the number of organism per cubic meter of air sample. This kind of correction was first described by Feller, a mathematician in 1950. The following formula describes the presumption that as the number of viable particles being impinged on a given plate increases, the probability of the next particle going into an empty hole decreases. And this is the formula which has been used, and the total number of wells which are formed are 378. And this is the conversion table chart which is given by the high media along with the equipment. And we can see if there is one colony which is formed on the plate, that is equivalent to 3 colony forming units. And if there are 4, then it is 6. So according to this chart, we can consider the colony forming units. Interpretation. If the sample volume is 500 liters, that is 0.5 meter cube, that is for 300 seconds or 5 minutes, and the number of colonies formed are 126, then the corrected number of colonies using Feller's table will be 156. Then the colony forming units per meter cube will be 312 colony forming units per meter cube. Results. In our study, total of 153 indoor air samples were collected from various wards of the hospitals, and in that 124 were culture positive, resulting in 150 isolates of 9 genera of bacteria. In that, gram-negative and gram-positive bacteria accounted for 61.3% and 38.6% accordingly. The bacteria isolated were Staph aureus, which was 25.4%, Klebsiella species, which included 26.8%, Micrococcus species, 8.8%, Coagulus negative streptococci, which included 11.5%, Proteus species, 1.8%, Streptococcus species, 15.6%, E. coli, 7.6%, Citrobacter, 1.2%, and Enterobacter, 1.2%. So the most common organisms were Staph aureus and Klebsiella species. Maximum sensitivity, that is 90%, was seen with imipenem, followed by moderate activity with piperacillin tazobactam, which was 85.5%, and amikacin 45.5%, and cotrimoxazole 42.5%, and poor susceptibility patterns with the remainder of the drugs. Conclusion. High level of antimicrobial resistance was observed among the airborne isolates obtained in the study. That is 91.9% were resistant to one or more antibiotics, and 71.8% were multi-drug resistant to commonly used antibiotics in the hospital. So air quality of the hospital environment in the restricted settings deserves attention and requires long-term surveillance to protect both patients and healthcare workers. Thank you.
Akhil,
Yes, ma'am.
can you tell me what are the 4 type of antibiotic resistance? Can you tell me the categories of 4 main type of antibiotic resistance?
4 main type of antibiotic resistance, you mean? Okay. Extended spectrum beta lactamases, metallo-beta lactamases, carbapenemase resistance. We have AMC resistance.
These are the type of antibiotic, but the mechanism of type of antibiotic resistance. But I am telling the 4 main category, मतलब 4 type of antibiotic resistance. कि किस 4 जो types होते हैं, वो आप बताइए.
4 types of antibiotic resistance, ma'am.
Yes.
In the Hello. Ma'am, you mean antibiotic F-lex? Hello. Hello, ma'am.
Hello, am I audible?
Yes, ma'am.
Yes, please.
Is it intrinsic resistance, acquired resistance, genetic change, DNA transfer resistance? Am I going in the right way, ma'am? Hello?
Hello, 4 categories. Like first is the limiting uptake of the drug. Second is the modifying the target drug. These type of category I am asking you.
Okay.
Okay, no problem. Can you tell me what are the ways that antibiotic become resistant?
Ma'am, there
How
are
many ways? How many ways antibiotics may become resistant?
according to AMSP, ma'am, antimicrobial stewardship program. So that is to combat the resistance. Right drug, right dosage, right administration, right way of giving it, and these are the ways by which the drug has to be given. If if they are not according to those guidelines, if they are not given or followed, then that could lead to resistance where the antibiotics can escape from the drug and that become threshold after multiplying and could cause resistance. Even through veterinary usage can also, the drugs can become resistant.
Okay, fine. Okay, thank you.
Thank you.
Okay, thank you, sir. So I just want to inform the session chair and the other participant that Dr. Sri Dharani Mungala and Dr. Mohammad Imazin will be joining us later as they are having some technical issues. So now I would like to call Dr. Shak Mahmoudin Momzan to present the topic over prevalence of vancomycin resistance enterococcus at the Tertiary Care Hospital. I would like to request Shak Muzam to join to do the presentations.
Mr. Sheikh Musa, we couldn't hear you. Kindly text us if you are with us.
Hello.
Yes, you're pretty audible now. Kindly share your screen and start the
Yeah.
presentation, please.
Sir, I just want to know that you're facing any technical issues so that we can help you out from your.
Hello. Yeah, I have had an issue with the sharing of my screen. There is some issue going on.
Okay, sir, if not possible, you can share to the WhatsApp number so that we can help you out from your.
Okay, just a second. I've just shared you, ma'am.
Yeah, we have get your screen. So I'll just share you now.
Very good afternoon to all the delegates here. I'm Dr. Sheikh Mahmooduddin Mozam, a first year postgraduate resident from the Department of Microbiology, GMC Nalgonda. My topic of presentation is prevalence of vancomycin resistance enterococci at a tertiary care hospital, Telangana. Coming to the introduction, the enterococci are gram-positives. They are facultative anaerobes, often are recognized as a spectacle-shaped, which are present in either singles, pairs, or short chains on a smear. Despite being commensals of adult feces, enterococci continue to be a significant nosocomial pathogen. They are commensals of the intestine, the female genital tract, the oral cavity, including the humans and the animals. There are about 35 species known to exist, but about 85 to 90% are Enterococcus faecalis, and 10 to 15% include the Enterococcus faecium. These organisms have gained recent importance due to the acquired glycopeptide resistant, which is not only limited to the therapeutic drugs, but also to the transfer of other organisms, like example is MRSA. And according to the recent studies, the nosocomial infections, Enterococci ranked the third in terms of bacteremia and second in terms of UTIs. Next slide, ma'am. Next slide, ma'am. Yeah. Continuing with the introduction, among the hospital-associated enterococcal infections, UTIs infections and remain the highest, followed by the surgical site infection, and next comes the bacteremia. The species Enterococcus faecium, which has gained resistance to vancomycin and penicillin, has become a therapeutically challenging organisms at the hospitals. The drug resistance, which occurs due to the alteration of the drug target sites, that is from D-alanine to D-alanine D-lactate. The emerging multi-drug resistance, especially the vancomycin resistance among the enterococci, is spread. It has been spread in response for many outbreaks in the world, with faecium and faecalis becoming the species of international concern. Next slide, ma'am. So my aim of the study is to ascertain the prevalence, the antimicrobial susceptibility patterns, and the associated factors of vancomycin resistance enterococci in a variety of clinical specimens from the patients which have been received at the tertiary care hospital. Next slide, ma'am. Coming to the materials and methodology, a cross-sectional prospective study was carried out between December 2023 and May 2024 in the Department of Microbiology at the Tertiary Care Hospital. Approximately 76 distinct clinical isolates of enterococci were obtained from various clinical samples, which includes blood samples, pus, urine, and also the tissue fluids from both the inpatients and the outpatients coming to the hospital over the course of the study. The isolates were identified using both an antibiotic susceptibility testing and a standard biochemical test according to the CLSI guidelines. And the conventional techniques for identifying the VRE were the minimum inhibitory concentration test and the Kirby-Bar disc diffusion approach using an MHA agar. Next slide, ma'am. This is the pics demonstrating the bacterial isolation and the identification. The first one shows a enterococci from a urine sample, and the second one is a bile escalin hydrolysis test, which is positive for enterococci. Next slide, ma'am. Coming to the results, out of the 76 isolates which were taken, the majority, that is 48.06%, came from the urine samples, while the blood culture showed about 33.08% and the pus showed about 5.66%, coming the second and the third variety respectively. And the susceptibility testing came out to be 11.13% of the people who are shown to be having vancomycin resistance enterococci. Next slide. This is the graph showing the distribution of the VRE among the various clinical specimens. As shown, urine had the highest percentage. Coming to the next is blood, then with the pus and the remaining isolates showing about 13.2%. Next slide. This is the slide showing the various antibiotics susceptibility pattern coming of the various drugs as shown below, like linezolid, aminocycline, teicoplanin, chloramphenicol, Tetracyclines, erythromycins, ampicillins, norfloxacin, nitrofurantoin, and clindamycin, where higher sensitivity was seen with ampicillin and norfloxacin. Next slide. Next slide. Coming to the discussion, the present study had shown the prevalence of to be about 11.13%, showing the VRE. Mellis et al. Conducted a systemic review and meta-analysis, which included 20 studies from PubMed, Embase, Google Scholar, and African Journal Online, and other sources, where the pool prevalence was found to be 14.8%. In my study at the tertiary care, in a study conducted at tertiary care center in North India, the prevalence was about 7.9%. And the highest percentage of isolates came from urine samples, that is 48.1% in my investigation, which matched the prior investigation by Mukherjee et al. Yeah, coming to the next, in that investigation, urine accounted for about 80%. Previous slide, no? Previous slide. Yeah, the last one accounted 80% of the isolates, with the pus accounting for 16% and the blood for about 3.3%. Next slide. Continuing with that, according to the study, about 2.4% of VRE isolates were seen in the University of Manitoba, which were resistant to chloramphenicol, 47.6% to gentamicin, and 85.8% to ampicillin, and 3% to linezolid. In contrast to the findings, we found that that VRE isolates were 100% susceptible to ampicillin and norfloxacin. Next slide. So my conclusions and recommendations would be the increased prevalence of VRE in the hospital settings is largely due to an inappropriate sanitation practices and the careless use of broad spectrum antimicrobial agents. Due to the increased treatment costs, the high frequency of VRE is causing a nosocomial infection and patient morbidity as well. According to this study, VRE is more susceptible to ampicillin and norfloxacin. And this study emphasizes how important it is to run regular surveillance programs in order to quickly identify the VRE in communities and hospitals. And lastly, this emphasizes the necessity of putting strict infection control measures in place, such as appropriate containment, efficient treatment of VRE infections, and sensible utilization of antibiotics, especially limiting the utilization of vancomycin. Thank you from my side. And these all are my references. Thank you.
Yeah, thank you so much, sir. So the next presentation will be done by Dr. Suryakala Elangovan by presenting over the title of a study on prevalence of...
Hello, I have a question.
Yeah, ma'am, you can go over it.
Okay. Can you tell me the most common site for enterococcal infection? I mean, the most common site in the human body in which enterococcus may cause the infections. Am I audible? Roshni madam, am I audible?
Ma'am, you're audible, ma'am. I think he's facing some technical issues, so what he's not answering over it.
Okay, fine. So you may proceed now.
Okay. So the next presentation will be done by Dr. Suryakala Elangovan.
Good afternoon, everyone. Myself, Dr. Surya Kala. My topic is on prevalence of extended spectrum beta lactamase producing Escherichia coli among various clinical samples at a tertiary care hospital, South India. Next slide, ma'am. Coming to the introduction, Escherichia coli has been the most commonly isolated and confined species within the Enterobacteriaceae family. The major risk factors for emergence of multi-drug resistant E. coli infections include prolonged hospital stay, prolonged antibiotic exposure, severe illness, and an unusual use of third-generation cephalosporins. And the principal mechanism of resistance that is offered by cephalosporins and most of the bacteria are ESBL, that is beta-lactamase production. And the principal mechanism of resistance to cephalosporins and penicillin is by the production of beta-lactamases. The extended spectrum beta-lactamases are enzymes that show expanded hydrolysis of the side chains of oxamino-beta-lactams, which includes drugs such as azithromycin, which is a monobactam, ceftriaxone, cefotaxime, and ceftazidime. Therefore, the up-to-date knowledge of prevalence of ESBL generation by one of the most frequently isolated life forms, that is Escherichia coli, is crucial for comprehending the illness burden and implementing crucial precautions to stop the spread. Next slide, ma'am. Ma'am, next slide, please. The aim of the study is to identify the predominance of extended spectrum beta-lactamases among E. coli isolates from various clinical samples. Ma'am, next slide. The materials and methods. A prospective study was conducted over a duration of 6 months, and about 70 non-repetitive clinical segregates of Escherichia coli from various clinical samples, which included urine, blood, pus, and other body fluids, were taken into consideration. And their antimicrobial susceptibility pattern were noted, and they were inoculated on ESBL blaze agar. The double disc synergy test, which uses combination discs containing clavulanic acid and a cephalosporin, was used to validate the ESBL phenotypic detection as per the clinical and laboratory standards guidelines. And an organism was considered to be an ESBL producer when the zone of inhibition around clavulanic acid combination is more than 5 millimeter compared to a cephalosporin alone. Next slide, please. Coming to the results, of the 70 isolates, 36 isolates, that is 51.4% of the isolates, produce extended spectrum beta-lactamases, and 8 among the 36 were resistant to multiple agents. Next slide, please. Among the various samples, urine sample accounted for ESBL production, more for ESBL production, that is 41.4%, and blood was about 25.7%, and wound or pus swabs contributed to 28.5%, and exudates 4.3%. Next slide, please. 90% of the patients showed maximum sensitivity to meropenem, which is followed by piperazolin azobactam, which was 88%, and imipenem, which was 85%. There was moderate activity seen with nitrofurantoin and colistin. Next slide, please. Coming to the discussion, because ESBL producing strains are resistant to azithromycin, trimethoprim, sulfamethoxazole, quinolones, and all penicillins, as well as most of the cephalosporins, which included the 3rd and 4th generation, they pose a particular threat. And effective disease control strategies and appropriate treatment depend on early detection of ESBL production. The increased diversity of drug-resistant bacterial segregates that are resistant to ESBL, they can pose a significant challenge for the clinicians when selecting the restorative drug options. Furthermore, the presence of ESBL producing microbes increases treatment cost, lengthens the hospital stay, and also increases mortality and morbidity. Consequently, controlling illnesses within clinics may lower the frequency of ESBL producing bacteria as well as their dissemination to neighboring communities. The incidence of resistance seem to be decreased by sparing the use of antibiotics, that is by controlling the use of antibiotics. The majority of the segregates in our investigation were susceptibly vulnerable to meropenem, piperazolin tazobactam, imipenem. Next slide, please. And the present study concludes that there is a high prevalence of ESBL generation, and they limit the available options for treatment, and so the appropriate antimicrobial treatment and the implementation of suitable disease control approaches are necessary to prevent the dissemination of these pathogens. The territorial information obtained from this consideration will offer assistance in directing suitable antimicrobial utilization, which is profoundly fundamental. These are my references. The take-home message is that since these drugs limit the usage of antibiotics that are presently available, they should be considered very carefully, and appropriate antibiotic should be selected, and appropriately it has to be given to the necessary patients. Thank you.
Hello.
Hello.
What is the principle of ESBL?
Ma'am?
What is the principle of ESBL?
Ma'am, hello, ma'am.
Dr. Suryakala, I am asking,
Yes,
what is the principle of ESBL?
The principle of ESBL is that they hydrolyze a beta lactam ring, ma'am.
Beta lactam ring.
Yes.
Okay. And what is the principle of beta lactamase?
Sorry, ma'am, I couldn't get you.
What is the principle of beta lactamase?
The beta lactamases, they had. Sorry, ma'am.
Yes, beta lactamase. It is an enzyme that is produced by the organism. And what is the role of this enzyme? Okay, no issue. Thank you so much.
Okay, ma'am. So next, hello, ma'am. Am I audible to everyone? Yeah. So the next presentation will be done by Dr. Shubhangi Sharma.
Hello. Hello. Am I audible, ma'am?
Yes, ma'am. You can go ahead.
Okay. So, ma'am, my slide is showing on. Hello. Good morning to all. Myself, Dr. Shubhangi Sharma, and today's topic is a detection of metallo-beta-lactamase gene prevalence among the Klebsiella pneumoniae isolates by using combined risk test and the multiplex real-time RT-PCR, a study from northwest part of India. Now comes to the introduction. Carbapenems are the beta-lactams group of antimicrobial agent most commonly used as the last line agent to treat the life-threatening infection caused by the multi-drug resistant microbes. However, in the recent past, due to the increase in judicious and we can say indiscriminate use of carbapenems, resistance have been reported, which are quite alarming for all of us. Carbapenemase productions and reduced expression of porins, along with the overproduction of beta-lactamases, possessing the weak carbapenemase activity. These are the basically two mechanisms which are responsible for the carbapenem resistance. Beta-lactamases are divided into three classes: class A, B, and D, according to Umbrel classification. Class B beta-lactamase require 1 or 2 zinc ion for their full catalytic activities, and these enzymes are called as metallo-beta-lactamases. The most commonly class B metallobeta-lactamases family include the New Delhi metallobeta-lactamases, imipenem-resistant Pseudomonas, Verona integron encoded metallobeta-lactamases, German imipenes, and the Sialan imipenes. The objective of this study is to investigate the presence of metallobeta-lactamase, carbapenem, in the Klebsiella pneumoniae in our hospital setting, as well as evaluate the efficiency of phenotypic and molecular assay by detection the MBL production to determine the prevalence of class B carbapenemases by the phenotypic confirmatory combined disk diffusion test to determine the existence of the beta-lactamase, NDM, BIM, and INP gene in all combined disk positive Klebsiella pneumoniae isolate by the real-time RT-PCR molecular assay. Now, the briefing of this methodology, we used pus, urine, various clinical samples like the pus, urine, blood samples, and the respiratory secretion for isolation of Klebsiella pneumoniae by using the standard microbiological practices. We isolate the organism in a PO form, and then further we go for the screening method like the antimicrobial susceptibility testing or by the Kirby-Bohr disk diffusion method on Muller-Hinter agar by using the meropenem, imipenem, or erythropenem, 10 microgram. If the organism is resistant, then we go further for combined disk test, and if it is susceptible, then we exclude it from our study. If organism is found resistant to AST pattern, then we go further the combined disk test. If combined disk test is positive for organism, then we go for further RT-PCR test. In the positive result, we show CAPPC, and we found we also do the MBL. MBL class B, we do further the RT-PCR test. So basically, it is a descriptive cross-sectional study carried out to detect the metallobeta-lactamases by combined disk test, multiplex RT-PCR reaction among 200 Klebsiella pneumoniae isolate. Hello? Hello?
Yes, ma'am, you are audible well enough. You can go ahead.
Okay, fine, thank you. It is a descriptive cross-sectional study carried out to detect the metallobeta-lactamases by the combined disk test and the multiplex real-time PCR reactions among the 200 Klebsiella pneumoniae isolates at the MBGH hospitals during the January 23 to June 23. The standard microbiological method were used for the isolation and identification of Klebsiella pneumoniae. All the 200 isolates of Klebsiella pneumoniae were further screened by the carbapenem resistant by Kirby-Bohr disk diffusion method on Muller-Hinter agar. All the isolate which were found to be carbapenem resistant by screening test were subjected to phenotypic confirmatory test in the form of combined disk test. Further, all the combined disk test positive metallobeta-lactamases, beta-lactamases NDM, BIM, and INP isolate, we enroll for polymerase chain reaction. Now we found out of 200 Klebsiella pneumoniae isolates, we found 80 were resistant to carbapenem by the antimicrobial susceptibility testing. Among 80 carbapenem resistant Klebsiella pneumoniae isolate, we found 55 isolate were positive for MBL, and 4 isolate were found positive for CAPPC by the combined disk test. 21 isolate were not found for positive for CAPPC and MBL because they are not inhibited by the phenylboronic acid as well as ethylene diamine tetraacetic acid. Out of the 200 Klebsiella pneumoniae isolate, 55 MBL positive isolate by combined disk test could be preserved in PO form, which were further used for carbapenemase gene detection by the RT-PCR. 22% isolates were found positive by RT-PCR and rest all 11 were found negative for any other gene by the RT-PCR method. NDM was the most frequently gene, followed by, we found 27 isolate as NDM, 11, then next is followed by the Verona integron metallobeta-lactamases was 11, and consequently presence of imipenemase with OXA 48 was 3%. There was a statistically significant association between the combined test and the RT-PCR assay. Now the discussion part is the emergence of the multi-drug bacterial resistance due to carbapenem production is increasing day by day worldwide. There is more crucial to rapid identification of carbapenem resistance, especially in Indian setup, because predominance of the NDM as carbapenem encoded gene in multi-drug resistant isolate. In the present study, 80 isolate of Klebsiella pneumoniae were screened to be positive for carbapenem resistant. A similar study conducted by Satyajit K. Pawar et al. They found 21.77%, Kumar Swamy et al. Found 24%, Panchal et al. 28 reported a higher carbapenem resistant. We found MBL production in 55 isolate of Klebsiella by using the combined disk diffusion method. A similar study done by Panchal et al. They found 19.6% and Adekar they found 18% in contrast to our study. Fazlul et al. Found 52.6% and Merrill et al. Found higher prevalence MBL in their study. In this study, we found 44 isolate were positive for NDM, VIMG, and IMP gene by RT-PCR molecular assay. NDM was the most common frequently gene, followed by Verona integron metallobeta-lactamases, and further is a 6 isolate we point as a imipenem. Various authors from India have reported a high prevalence of NDM in their studies. Gupta et al. Reported 22%, Boyce reported 26%, Pawar et al. Reported 31%. Now the summary of this study revealed the increase in trend of carbapenem production and a higher prevalence of MBL gene in Klebsiella pneumoniae isolate, which were not previously established. There is an urgent need to implementation multidisciplinary approach to limit the spread of the strategy for early detection, antimicrobial stewardship program, and appropriate treatment measures are required to stop the spread of carbapenem resistant organism are the need of the hour to successfully tackle this global threat. Now, these are the references of my study.
Okay, thank you so much, ma'am, for presenting your paper over here. And so is there any questions from the session chairs that you would like to raise, ma'am? Okay, fine. So we will move on to the next presenters. So the next presenter will be Abhaya Sharma.
Hello, am I audible?
Yes, ma'am, you're audible now.
Good afternoon, everyone. I am Abhya Sharma, a PhD scholar from Department of Microbiology, SMS Medical College, Jaipur. My today's topic for the presentation is emergence of JN1 variant of SARS-CoV-2 in Rajasthan. So, introduction. The coronavirus disease 2019, that is COVID-19 pandemic, caused by severe acute respiratory syndrome coronavirus 2, continues to rage due to emergence of Omicron variant and its descent subvariants. SARS-CoV-2 has undergone continuous mutations through its evolution, giving rise to numerous variants that cause multiple global infection peaks. JN1 subvariant is rapidly becoming the dominant strain of SARS-CoV-2 and is one of the several variants circulating globally. It has many immune escape mutations, therefore it has growth advantage over other subvariants. The JN1 variant of SARS-CoV-2 was first identified in India in Kerala in December 2023. Rajasthan also detected its first case of JN1 in December 2023. In August 2023, amid the global dominance of XBB sublineages, the BA.2.86 SARS-CoV-2 variant with more than 30 protein mutations emerged. By the end of 2023, it evolved into JN.1. This variant distinguished by three different mutations, that is F19L in ORF7b, L455S in the S protein, and R3821K in ORF1A, significantly dominating the global sequences, raising concern about its distribution and clinical implications. The aim and objectives of my study was to carry out the genomic surveillance of SARS-CoV-2 in Rajasthan, to study the clinico-epidemiological profile in patients infected with JN.1 variant. Methodology. A total of 97 throat swab or nasopharyngeal swab of SARS-CoV-2 positive patients were received from all over the districts of Rajasthan at Department of Microbiology, Savai Mansi Medical College, Jaipur from November 2023 to February 2024 for next generation sequencing. Nucleic acid extraction was done on EasyMac extraction system of Biomedics. Confirmed positive samples for SARS-CoV-2 having viral load, that is CT value 25 or less than 25 for E and ORF gene, were selected for NGS analysis. Then library preparation, that is process of converting a genomic DNA sample into different fragments which can be sequenced further on an NGS instrument, was done using Iron AmpliSequence Library Kit Plus and Iron AmpliSequence SARS-CoV-2 Research Assay Panel of Life Technologies as per manufacturer's instructions. Then briefly quantification was done using Qubit HS DNA kit. Quantification is basically the process to know the amount of DNA which is present to check the DNA quality. Then Iron Chef System Life Technologies was used for further template preparation, enrichment, and chip loading. Prepared loaded chip was sequenced on the Iron Torrent System S5 System Life Technologies. Then FASTA files, that is fast alignment sequence test for application sequences, were generated from coverage analysis, variant caller, and generate consens plugins respectively as per manufacturers were downloaded. Lineage analysis was done using Nextclat. So this is the clinical guidance for management of adult COVID-19 patients given by Ministry of Health and Family Welfare, Government of India. It has divided the patient diagnosed with COVID-19 into three bases of disease. First is mild disease, that is a patient with upper respiratory tract symptoms and or fever without shortness of breath or hypoxia. The patient is home isolated and taken care. Then moderate disease, that is a patient with any one of the below, like respiratory rate more than or equal to 24 per minute, breathlessness. Second is SpO2 90 to less than 93% on room air. Patient is generally admitted in ward. Then severe disease, any one of the below, like respiratory rate more than 30 per minute, breathlessness, or SpO2 less than 90% on room air, patient is generally admitted in ICU. So we telephonically contacted each positive patient of JN1, and we have gathered the information or taken the history of patients from them, and some of the history was taken from Directorate of Medical and Health Services, Rajasthan. So we have found that total 97 samples we have received for sequencing from November 2023 to February 2024. And out of 97, 79 lineages were JN.1, 7 were XBB.1.16.17, 3 were BA.2.86, 0.6.1, 2 were GE.1, 2 were BA.2, 1 was XBB.1.16.1, and 1 was HB.1. Then out of 79 lineage identified, JN1 was the dominant, that is 53.60%. Then it was JN.1.11.1, it contributes 16.49%. And then it was JN.1.1, it contributed 4.12%. And then it was JN1.18, that is 3.09%, and continued. Then when we took the history of the patients, we find out that 21.51%, that is 17th of the JN.1 cases were asymptomatic. Among the diseased cases, we found that majority of the patients, that is 56.96%, held mild disease. 17.72, that is 14, had moderate disease, and 3.79% had severe disease with SpO2 less than 90%, requiring admission in ICU. Out of 79 cases of JN.1, 74, that is 93.67% of cases recovered, and 2 were succumbent to the diseases. In present study, 24% cases were previously infected with COVID-19, that is, they were having reinfection, while 76 were the first time infected with COVID-19. Majority of the patients were vaccinated, that is, total 79.74% of cases were vaccinated. Only one patient was found to be non-vaccinated, and 15 were the patients were children which were below the age of for eligible of vaccination. Discussion: an abrupt increase in number of SARS-CoV-2 positive cases was observed in India during late November 2023. In view of this upsurge in COVID-19 cases and detection of first case of JN1 variant in India, Government of India issued an advisory to regularly report and send positive samples for genome sequencing to INSACOG laboratories. The first emergence of JN.1 in Rajasthan was observed during Epi Week 50, whereas in India, the first case of JN.1 was reported from Kerala as a part of routine surveillance by INSACOG in Epi Week 49, that is 8th December. JN.1 was initially tracked as a part of BA.2.86, the parent lineage that was classified as a variant of interest due to its rapidly increasing spread. WHO later classified JN.1 as a separate variant of interest. The epi-week-wise data in the present study showed that JN.1 was the predominant lineage during each epi-week after its first emergence in Epi Week 50. In the present study, apart from JN.1, the other lineages identified were XBB.1.16.17, BA.2.86.1, GE.1, BA.2, XBB.1.16.1, and HV.1. 17 of the JN.1 cases were asymptomatic, and among the disease cases, majority 45 had mild disease, 14 had moderate disease, and 3 had severe disease. In the present study, out of 79 cases, 74 cases recovered with home isolation or mild treatment. Majority of the JN.1 cases were from Jaipur district of Rajasthan. In our study, death was reported in 5 cases, among which 2 were due to COVID diseases, while 3 were incidental case of SARS-CoV-2 infection. The current study shows that JN.1 variant is causing mild disease and has low mortality rate in Rajasthan, which coincide with the study of Karakarte R. et al. In Maharashtra. Conclusion. The JN.1 variant has emerged as the predominant SARS-CoV-2 variant in India, with clinical presentation similar to previous variants circulating in Rajasthan, India. Notably, while JN.1 has not been associated with most severe disease outcomes, its enhanced transmissibility underscores the importance of studying the ongoing viral evolution. It emphasizes on the need for continuous surveillance for timely detection of new emerging variants and more collaborative efforts for a global proactive approach for pandemic management. This is my acknowledgement. I acknowledge the support of my guide, Dr. Bharti Malhotra, Senior Professor, SMS Medical College. I would also like to extend my sincere thanks to Dr. Pratibha Sharma, Dr. Swati Gautam, VRDL SMS Medical College. And I would like to extend my sincere thanks to Directorate Medical and Health Services, Rajasthan. These are my references of my study. Thank you.
Yeah, so thank you so much, ma'am, for presenting your paper over here. So the presentation which will be going to done is will be Arumya Banerjee.
Yes, ma'am. Am I audible?
Yeah, you're audible, ma'am. You can go over it.
Okay, so can I share my screen?
Yeah, you can.
Okay, thank you. Ma'am, is my screen visible? Yes. Okay, thank you. Okay, so good afternoon, everyone, and welcome to my presentation. I'm identified as Arunima Banerjee, a student of MSc Microbiology of the Ananda Sagar University, Bengaluru, India. As you can see on the screen, my topic for today's seminar is impact of heavy metals and formation of biofilms. This was basically a project work that I did during my last semester of bachelor's under the supervision of Professor Shubhankar Bodua sir from Asansol Girls College, affiliated to Kazi Nazrul University, West Bengal, India. So here is the abstract, basically the summary of the entire project. It depicts the fact that how heavy metal pollution caused by various industrial and mining activities poses a serious threat to all forms of life in the environment because of the toxicity of metals. And here bioremediation acts as a sustainable tool for heavy metal removal as it is considered safe, effective, and feasible. Next, here are the topics that I'll try to cover in this presentation. Next comes the introduction part of this project work. Firstly, to study the impact of heavy metals on formation of biofilm. We need to know what are biofilms. Biofilms are complex and dynamic communities of microorganisms that are organized in a matrix of extracellular polymeric substance or EPS adhered to a surface. The microorganisms in the biofilm can be classified into different groups based on their metabolism, morphology, gene expression patterns, etc. And the EPS is composed of polysaccharides, proteins, and DNA, and it provides mechanical stability, helps to retain water and nutrients, protect microorganisms from environmental stressors such as antibiotics and disinfectants. Next, significant implications of biofilm formation in various domains can be human health, water treatment, industrial processes, etc. And next, here are the names of 5 metals that were used during this experiment, that is lead, mercury, cobalt, chromium, and nickel. Next, heavy metals and environmental pollution. These heavy metals are too toxic and can bioaccumulate in living organisms. They can contaminate soil, water, and air. They can enter environment through industrial discharge, waste disposal, etc. And once released, they interact with microbial communities, influencing their structure, function, and ability to form biofilms. Next is biofilm in various settings such as medical, as biofilms persist on various medical devices such as catheters, prosthetics, and implants. Biofilms also help in wound healing and tissue regeneration. Next, in industrial context, biofilms can cause biofouling, leading to reduced efficiency and increased maintenance costs in systems like pipelines, water treatment facilities, etc. And finally, in environmental aspect, biofilms contribute to nutrient cycling, water purification, bioremediation, etc. And lastly, we can say that heavy metals that were used during this project work like lead, mercury, cobalt, chromium, and nickel, they have a great impact on biofilm formation. Like lead is known to interfere with cellular processes such as enzyme function, membrane integrity, and nutrient uptake, whereas cobalt can induce oxidative stress and cellular damage, reducing biofilm biomass and viability. Mercury can disrupt quorum sensing, a communication process used by bacteria in biofilms to coordinate their behavior. Chromium and nickel can cause DNA damage, oxidative stress, and enzyme inhibition. Moreover, some metals are known to trigger biofilm formation, whereas some can inhibit the formation. We'll go through that part once we proceed with the upcoming slides, especially the result that we have got. Next, the actual aim of the experiment was to investigate the effects of heavy metals on adhesion, growth, and development, and survival of microorganisms within biofilms by examining changes in gene expression patterns, protein production, and cellular physiology. Next, the media that was used here was a biofilm-specific media, that is glucose media, along with BS broth, and here are their compositions. Next comes the methodology. The sample was collected from a dump yard located in Kalipahari, Asansol, a city in West Bengal, India. Then the sample was enriched with glycerol, and then the stock was kept in a deep freezer for cryogenic preservation at -4 degrees Celsius. Next, the biofilm specific media, that is glucose media, was prepared to grow the biofilm. Then the metals like lead, mercury, cobalt, chromium, and nickel were added in different concentrations like 5 microliter per ml, 4 microliter per ml, etc. to observe the colonization and development of the biofilm. Then the sample was added to the media containing the metals, and then the test tubes were kept in shaker overnight for 48 hours. And finally, after 48 hours, the tubes were observed and OD was measured at 600 nanometer. Now, next comes the result. The result of 5 different metals used at various concentrations is shown here. The first graph here shows the bacterial growth in presence of lead, and the second one shows EPS formation graphically. As in case of lead, we can see that the maximum growth observed was at 5 microliter per ml concentration. Now, this concentration will vary for other metals. Like for chromium, it is different. Here is the graph of chromium. Again, for mercury, we observed the least biofilm formation was at 1.5 microliter per ml. Next, this is the graph of cobalt. And lastly, this is of nickel. Now, the main thing that I wanted to show from this graphs is lead exhibited maximum biofilm formation at 5 microliter per ml, as it stimulated biofilm production by inducing the expression of adhesion genes and EPS production. On the other hand, mercury showed least biofilm formation at 1.5 microliter per ml concentration. The toxicity of mercury can have hindered biofilm formation by disrupting cellular processes and viability. These findings of importance important implications for understanding the role of heavy metals in biofilm development and the potential impact on environmental and industrial settings. And the main theme of this entire project work was how to save environment, which can be further understood by the next slide, that is future perspective of the experiment. That is bioremediation strategies. First is wastewater treatment. Biofilms can be used to remove organic matter and nutrients from wastewater. And next is heavy metal removal. Biofilm engineered to remove heavy metals from contaminated water and soil. Next comes the future studies of the experiment. Though due to lack of fund and time, these were not performed, but in future I want to proceed further with this, like scanning electron microscope or SEM, which is an useful technique for studying the structure of biofilm by offering resolution, imaging morphology and composition. And next is X-ray diffraction, which characterizes metal binding sites in biofilm, which are used to study how much amount of portion of heavy metals are entrapped or absorbed in biofilm. Lastly, we can conclude that the mechanisms by which these metals interact with microbial communities include oxidative stress, enzyme inhibition, and alternation in microbial community composition. And also, this whole experiment was performed in summer season. In future, if it is performed during rainy or winter season, the results that we have got here might vary, as the microbial flora community would also change accordingly with each season. Lastly, here are the references. So this was all about my presentation. It was really an honor to get an opportunity to present my topic here today. Thank you so much for your time and attention. Have a great day.
Hello.
Yes.
Arunima, can you tell me what causes biofilm to cause?
Sorry, ma'am. What causes?
What causes bacteria biofilm to cause? Can you explain the steps in the formation of the biofilm?
Okay. Ma'am, when bacteria adhere to surfaces in moist environments by excreting a slimy glue-like substance, it causes EPS, extracellular polymeric substance, and that causes to form biofilm.
Then what is the difference between the initial reversible attachment and after that irreversible attachment? Both are the step in the formation of the biofilm. Can you tell me the difference in the both?
No, ma'am, sorry.
Initial reversible attachment, after that irreversible attachment, and then maturation, and after that the dispersion of the biofilm. So these are the common steps.
Yes, ma'am.
But you have not any idea between the reversible and irreversible attachment?
No, ma'am.
Okay, fine. Thank you.
Thank you, ma'am.
Yeah, thank you so much, Arunima. So now I would like to thank both the session chair, Dr. Mukta Sharma and Dr. Ektika Hemant Kumar. So thank you so much, ma'am, for having a great part with us. So very much thank you to have a great day with you all. So there will be now the other session chair from the Dr. Uma is Assistant Professor from Bangalore University, and Dr. Shiva Veerakumar, Assistant Professor from Devangri University, who will be joining as a session chair for the other house. So now we can move forward for the other continuous presentations over it. Thank you, Dr. Mutta Sharma, ma'am. And again, I would like to thank you, the both of session chairs. Thank you, ma'am. Thank you so much.
Thank you so much.
So I think Dr. Uma ma'am and Dr. Shiva Veerakumar have joined now. So we can move for the other half of the presentation. So the other one who is going to be the presenter will be Dr. Neha. Ma'am, Dr. Neha, are you there?
Yes, ma'am.
Yeah, you can go over it, ma'am. Ma'am, if you're facing any technical issues by sharing your screen over there. Hello.
Hello, ma'am. I'm unable to share my screen, ma'am.
Okay, so if it is possible, you can share me in WhatsApp, ma'am, so that I can share you from our side.
Okay, ma'am. Yes, ma'am. I have shared.
Okay, ma'am, just give me a moment so that I can share from our side. Yeah, ma'am, you can go over it.
Yes, good afternoon, everyone. My name is Neha. I'm the PhD research scholar from Sri Siddhartha Medical College, Tumkur, India. So my topic is carriage of ESBL producing commensal Escherichia coli and Klebsiella pneumoniae colonizing the gut of healthy adult population in Tumkur. Coming to the introduction, unbridled use of antibiotics is the major factor that has transformed the healthy human intestinal gut flora into a reservoir of antibiotic resistant organisms. MDR colonizers can act as a source of infection. Resistant strains can disseminate to other bacteria and make them pathogenic, which can lead to the community outbreaks. Prevalence of these MDR strains and ESBL producing bacteria in the community is on the rise. In this report, we examined the prevalence of MDR and ESBL producing strains among healthy adult population in Tumkur. Ma'am, next slide. So coming to the aim of the study, phenotypic and genotypic detection of ESBL producers among E. coli and Klebsiella pneumoniae colonizing the gut of adult population in Tumkur. Objective of the study are isolation and identification of E. coli and Klebsiella pneumoniae from the stool samples by standard microbiological procedures, study of the AST pattern of the isolates as per the CLSI guidelines, and to screen for the MDR isolates and ESBL isolates. Next, to detect the ESBL with phenotypic conformity test and to detect the presence of genes coding for ESBL by multiplex PCR. Coming to the materials and methodology, stool samples of the healthy individuals were collected from the patients or the healthy volunteers visiting the SSMC OPD, and E. coli and Klebsiella pneumoniae were isolated according to the standard laboratory techniques. AST was done using standard methods along with the control strains were put up, and we screened for MDR isolates. All the MDR isolates were screened for the presence of ESBL detection by CLSI phenotypic conformity test, that is combined disc method we have tested. Then genotypic detection of ESBL was done to detect the presence of genes coding for ESBL like CTXM, SHB and OXA 10-bar 11 gene by multiplex real-time PCR. So in our study, we have found E. coli that is in 64.6% of the isolates and Klebsiella pneumonia from 35.3% of the individuals. Of the population studied, the total fecal carriage rate of MDR colonizers in our study was 54%. Maximum resistance was seen for fluoroquinolones group of antibiotics, that is 51%, followed by cephalosporins group, that is 48%. E. coli is the predominant organism isolated in our study, that is 67.6%, and followed by Klebsiella pneumoniae, that is 27.4%. In this study, 50.2% of the isolates were found to be ESBL producers by phenotypic conformity tests, and out of these, 424 ESBL positive isolates 82% of the isolates carried CTXM gene, then 13.5 isolates carried SHP gene, and 4.5% of the isolates carried OXA gene. The frequency of occurrence of genes coding for ESBL was higher in Escherichia coli. So coming to the conclusion, in the present study, the prevalence of ESBL producing commensal E. coli and Klebsiella pneumoniae in healthy individuals who were not hospitalized or administered antibiotics in the previous 3 months was determined. Our results emphasize the importance of the intestinal tract as a reservoir of ESBL producing E. coli and Klebsiella pneumoniae carrying antibiotic resistant genes. The increased prevalence of these organisms in human fecal flora was established in the community, represents an opportunity for these clones to become persistent. Overall, our study concluded how beta-lactam encoding genes among commensal E. coli and Klebsiella pneumoniae circulate in the community. The dissemination of these resistant genes will adversely affect the treatment strategy in community-acquired infections. This type of epidemiological surveillance is necessary in order to maintain or design efficient intervention strategies with the aim to reduce clinical infections with beta-lactamase encoding genes in humans. So these are my references. And thank you, one and all. Hello.
Yeah, ma'am, thank you so much. So I just want to know, is there any questions that you want to ask from the session chairs? Dr. Uma, ma'am, that you have any questions over it, the presenters? Okay, fine. So we will move to the next presentations, who is going to be done by Dr. Binish Gulzar. Dr. Binish Gulzar, am I audible to you?
Yeah.
Okay, can you please go over it by sharing your PPT?
Just a second.
Sure, ma'am.
Can I share it through WhatsApp?
Yeah, ma'am. Okay, no problem. We will help you out. Sorry for the inconvenience for the other participants. Kindly wait a moment. We can go over it. I gave the screening available to everyone, right?
Am I audible?
Yes, ma'am. The
Yeah,
screen
good
is visible to
afternoon.
you? Ma'am, I just want to confirm the PPT shared with everyone. Is it visible?
Yes, it is visible.
Yeah, thank you so much. Yeah, ma'am, you can go over
Yeah.
it.
Good afternoon, everyone. I'm Dr. Vinesh Gulzar, Assistant Professor, Department of Microbiology, GMC Baramulla, Jammu and Kashmir. So today I'm here to present my research about the prevalence of uropathogenic Escherichia coli and antimicrobial susceptibility pattern among patients at a tertiary care hospital, GMC Baramulla, which is in South Kashmir, Union Territory of Jammu and Kashmir. So can you please share the other? So these urinary tract infections, they are one of the most common and increasingly resistant bacterial infections encountered in healthcare settings. They have now become the most prevalent hospital-acquired infections, accounting for up to 35% of nosocomial infections and the second leading cause of bacteremia in hospitalized patients. The majority of UTIs are caused by gram-negative bacteria such as Escherichia coli, Enterobacter species, Klebsiella species, Citrobacter species, Proteus species, and gram-positive bacteria such as Enterococcus species and Staphylococcus saprophyticus. E. coli in particular is most common organism causing both community-acquired as well as hospital-acquired urinary tract infections. Can you please change the slide? Change the slide. Am I audible?
Yes, ma'am, you're audible. You can go continue with it.
You can change the slide.
Yeah, ma'am, sure.
Next slide, please.
Hello.
Hello?
Yeah, ma'am, you can see the slide.
No, no.
Ma'am, can you please tell me the topic of the slide that you want to present over it now?
It's the prevalence. Prevalence of uropathogenic E. coli.
Ma'am, I think this is the one you are expecting over it.
Yeah, yeah.
Okay, ma'am, fine, you can continue.
Please go to the introduction part.
Hello.
Yeah, please go to the introduction part.
Okay, ma'am.
Slide number 3, I guess. Yeah. Next one. Yeah, next one. Can you move to the next slide? Introduction only.
Ma'am, can you please tell me the topic that you would like to go over it? Ma'am, am I audible?
Yeah.
Yeah, ma'am, can you please suggest me the topic that you want to go over?
Yeah, introduction part, please. Yeah,
Is it
this one
fine, ma'am?
only.
Yeah, ma'am.
Yeah. Okay, shall I start?
Yeah, ma'am, please go ahead.
Yeah. So, enterobacteriaceae such as Escherichia coli have important mechanisms to avoid lethal doses of drugs such as aminoglycoside degrading enzymes, target alteration, decreasing uptake, beta lactamase enzyme degradation, and overexpression of efflux proteins. The rate of drug discovery and development in the 21st century cannot keep up with the continuous and detrimental change in antibiotic resistance trends. Moreover, treating multi-drug resistance, Escherichia coli, that causes urinary tract infections increases the cost of treatment, hospital stay, morbidity, and mortality in patients. Next. Next. The aims of this study was to determine the prevalence of Escherichia coli causing urinary tract infections and its antibiotic sensitivity pattern in patients admitted to GMC Baramulla, which is a tertiary care hospital in South Kashmir, Jammu and Kashmir. Next. So, in materials and methods, a cross-sectional study was conducted between August 2023 and May 2024, spanning over a period of 10 months, with the aim of determining the prevalence and antimicrobial susceptibility profiles of Escherichia coli in patients presenting with urinary tract infection symptoms at GMC Baramulla, both in outpatient as well as inpatient departments. So, urine samples were cultured to identify pathogens, and positive samples underwent antimicrobial susceptibility testing, followed by an evaluation of Escherichia coli's antibiogram. Next. So, to confirm the presence of pathogens, 10 microliter of well-mixed urine samples were inoculated onto high-chrome UTI agar using a sterile loop. The plates were incubated at 37 degrees centigrade for 18 to 24 hours. Morphological colony identification, gram staining, and biochemical tests were done to confirm the pathogenic organisms. Antibiotic susceptibility testing of E. coli isolates for various antibiotics were done, and diameter zones of clearance were measured in millimeters by Kirby-Bord disk diffusion method as per CLSI guidelines and interpreted according to CLSI protocol. For quality control, Escherichia coli ATCC 25922 provided in the laboratory was used as a control stain. Next. So, in our result, out of a total of 255 urine samples, 61 samples were positive for microbial isolates. Escherichia coli was the predominant pathogen, that was 26 out of 61 positive samples, and the total prevalence was 23.9%. The prevalence was higher among females, that is 11.5%, in comparison to males, that was 8.3%, and it was high among the age group of 20 to 39 years. Escherichia coli isolates were highly susceptible to fosfomycin, that is 85%, amikacin 80%, piperacillin tazobactam 75%, and nitrofurantoin 74%. But there was high resistance shown to cephalosporins like cefuroxime, that is 100%, ceftazidime 100%, naldixic acid it was 90%, in ciprofloxacin it was 90% resistance, and in gentamicin 76% of resistance was seen. Next. So here in this table, we can see the total prevalence of certain pathogenic organisms. Among them, Escherichia coli, it was highest, that is 42.6%. In Enterococcus, it was 29.5%. In Klebsiella species, it was 9.8%. And Staph aureus were seen around 6.5%. 3.2% of Pseudomonas aeruginosa were present. 1.6% Proteus, 1.6% of Citrobacter species were isolated, and a total of 4.9% of Candida species were also isolated. Next. So this is a table depicting the antimicrobial susceptibility pattern. So here in this, we can see there was high resistance to certain antimicrobial agents like ampicillin, ciprofloxacin, ofloxacin, naldixic acid, it was 73%, and but to polymyxin B, it was 0%. And similarly, we can see the susceptibility pattern where there was high susceptibility to amikacin, that was around 81%, and there was high susceptibility to fosfomycin, that is 84%, nitrofurantoin, that was 73%. Next. So from this Venn diagram, it can be depicted that there is high ASBL resistance followed by carbapenem resistance. And similarly, here we can see the susceptibility to nitrofurantoin and fosfomycin was high. Next. So this study concluded that Escherichia coli is a predominant uropathogen with significant multi-drug resistance to antibiotics due to empirical therapy with cephalosporins for the management of UTIs. The study also suggests that either nitrofurantoin, fosfomycin, and piperacillin tazobactam or their combination should be used in empirical treatment of UTIs in our hospital settings due to their high sensitivity. These results should form the basis for preliminary decision making on the appropriate line of treatment for urinary tract infections. Next. So these are certain references that I have used for my study. Thank you so much.
Yeah, thank you so much, ma'am, for giving us a wonderful explanation over this. So the next one who's going to be present will be Ravi Muria.
Ma'am,
Yeah.
am I audible?
Yeah, please.
Just a minute, ma'am. Ma'am, is my poster visible?
Sorry, Ravi, we cannot able to get that.
Just a minute.
And I also request the other participants who are facing some technical issues by sharing their PPT can do the PPT to share in the WhatsApp before 10 to 15 minutes of your slot so that we can help you some out from our side. Yeah, Ravi, it's visible to us. You can go ahead.
Thank you, ma'am.
Yeah, thank you.
Hello, everyone. My name is Ravi Maurya. I'm from Sardar Bhagwan Singh University, Balawala, Dehradun, India. And today's my topic is just a minute. Like visible, ma'am?
Yes, it is visible. You can go ahead.
Okay, ma'am.
Um,
Good afternoon, everyone. My name is Ravi Maurya, and I am from Sardar Bhagwan Singh University, Dehradun, Uttarakhand. Today's topic is metallic nanoparticle against antimicrobial resistance. Let's begin with introduction. The increase in antibiotic resistant bacteria has led to the search for new antibacterial agents capable of overcoming the resistance problem. In recent years, metal and metal oxide nanoparticles have been increasingly used to treat bacteria as an alternative to antibiotics due to their intrinsic bacterial activity. Metal-based nanoparticles are known to have a non-specific bacterial toxicity mechanism as they do not bind to a specific receptor in the bacterial cell, which not only make the development of resistance by bacteria difficult, but also broaden the spectrum of antibacterial activity. The efficacy of major metal-based nanoparticles have been studied over the years and showing promising result in both gram-positive as well as gram-negative bacteria. Different type of metal-based nanoparticle are there which were used in antibacterial therapy and biomedical applications. For instance, silver, copper, gold, zinc, iron, and titanium-based nanoparticles. Metal nanoparticles are characteristically synthesized by sol gel method. One of the attractive metal nanoparticles for medical application is the magnetic nanoparticle. These nanoparticles not only can be directed to the specific site under magnetic field guidance, but also be hidden under the magnetic forces. Now coming to methodology, a deep thorough study of microbiology and microbial sensors were done. Electronic databases including PubMed, Scopus, Science Direct, and other databases and blogs of various doctors and medical writers were searched for review articles. Research articles and books mention microbes, microbiology, and microbial sensors were collected and declaration of fact which were compiled and sequenced. Now, these are some nanoparticles: copper, silver, zinc, and gold. Now, coming to the mechanism of antibacterial activity, the first mechanism is disruption of cell wall, thus increasing their permeability as a result of electrostatic interaction between negatively charged molecule of the cell wall of microorganism and positively charged of nanoparticle. Now, second one is generation of toxic reactive oxygen species ROS. Oxidative stress led to oxidation of glutathione, disruption the antioxidant defense mechanism of bacterial agent ROS. The excessive production of ROS caused disturbance in redox homeostasis, which resulted in oxidative stress. Now, third one is binding to intracellular component, among other things, causing damaged DNA protein and inhibition of enzymatic activity. Now, coming to the result, in an era of increasing antimicrobial resistance, metallic nanoparticle appears to hold promising to improve current therapies and to develop new therapeutic agents of nanoscale nature. A possible way to increase the antimicrobial activity of conventional antibiotic is to use them in combination with metallic nanoparticles. This can be done either by synthesizing metallic nanoparticles with a modified surface functionalization with suitable antimicrobial agent, or by designing a core of synthetically acting metals. It is possible to select antimicrobial agent and use them in combination therapy like metallic nanoparticles and antimicrobial agent. These are my references where I can refer this poster. Thank you.
Yeah, thank you so much, Ravi Maria. So the next one will be the presentation will be going ahead with Aishugag. Aishugag, am I audible to you?
Yes, ma'am. Thank you
Yeah.
so much.
Yeah, kindly share your PPT over it and go ahead over of presentation.
Yeah, just 2 seconds, ma'am.
Sure.
Is it visible, ma'am?
Yeah, we can able to get that.
So, good afternoon, everyone. I introduce myself as Ishugar, M.Pharm Pharmaceutics Scholar Student from Sardar Bhagwan Singh University, Dehradun, Uttarakhand, India. And today I'm presenting a poster on microbial biosensors, a growing dimension. It's in a review. So, introducing with the concept of biosensors and microbial biosensors, here I have shared an image which is showing that how microbes are attached with the bioreceptors, and through transducers we can create or generate a signal based on the interactions between the analyte and the microbes, and that can be amplified and processed and can be displayed. Okay, so the sequence of analytes, bioreceptor, transducer, electronics, and display generates a biosensors that measures biological or chemical reactions by generating signals proportional to the analyte's concentration. And that's what's the basic concept of a biosensors. Now, integrating the science of microbiology with this biosensors is expanding in the healthcare sector in many things. I start with explaining the fact that microbial biosensors are being used in diagnostic purposes, in treatment purposes, and the most prominent problem that the globe is facing, the microbial resistance, the antimicrobial therapeutics agent that are creating resistance in the microbes can also be tackled through this microbial biosensors. The microbes provide a very good factory of enzymes, a whole cellular level knowledge to be used as a sensing agent in a biosensors. They have good adhesion properties and they could have culture growth properties, which makes them an ideal for the biosensors. Now, if I talk about the future and no sorry if I talk about the advantages, they have advantage of environmental capability. They are very much compatible with the environment, just like the other biosensors that requires a lot of chemicals to be synthesized. Then they have facile handling. They have are highly portable. They are quite sensitive and quite selective in nature. See, when we talk about the glucose monitoring type biosensors, we have so many issues in terms of sensitivity. The glucose monitoring biosensor that we are using these times can sometimes gives the measures of some natural sugars molecule that are part of the blood system itself. But the microbial biosensors are over them. They are just providing the test as like an HbA1c test, not only in the sugar leveling controls or checking. We have it for many other. Just in at a time of COVID, we have so many biosensors that are being used in RT-PCR for diagnostic purposes. Now, moving forward to the limitations, they have the problem of contaminations, because we all know that microbes get contaminated, microbial things get easily contaminated. They have viability issues. Now we have a very lesser time of viability with these microbial biosensors because of many factors that affects their growth. Next, coming to the genetic circuit, it's still a very difficult to know the exact genetic composition, genetic functionality, and genetic working of these microbes. And last, the intrinsic heterogeneity. We all know that microbes do not have a proper reproductive methods. While reproducing from one cell to another, there are chances of creating a huge level of heterogeneity, which might lead to many problems. So, coming to the fact, we have to all overcome all these limitations. The science is being working a lot in these sections where we are trying to overcome the heterogeneity. We are trying to know the exact genetic circuit of the microbes, and we are increasing the viability. Now, we all know the biosensors needs immobilization, and the microbes are highly available to get immobilized. So the success of microbial biosensors is a solitary result of encouragement in the microbial immobilization, their enhanced transducer detection system, and metabolic engineering. Now the metabolic engineering concept is developing with the fact that now we don't need any external controller. The microbial biosensors can work as an autonomous body that can themselves that can themselves detect the exact cause and can release the substance as the need, especially when we want to use them in treatment of microbial disorders. These will direct the healthcare center to advance superior, precise diagnostic, preventive, and treatment measures. Now, what I want to conclude that based on my sound science knowledge and literature survey, it can be said that combining the science of microbiology and learning and measuring biological processes is a demand of globe to tackle the lots of environment, healthcare, and AMR problems. As in environmental cases, might be aware of the Pseudomonas pitidia based biosensors that are being used to clean the water bodies. And now recently we have so many technologies and so many microbial biosensors that are being developed to treat for the environmental pollution treatments. So here comes are some references. These are the major 4 references that are referred for the introduction for this poster. And I'd like to acknowledge the faculty members of Sardar Bhagwan Singh University and ITS College of Pharmacy for their support. Thank you so much.
Yeah, thank you so much, sir, for presenting your paper. So the next person will be Arish. Dr. Arish, am I audible to you?
Yes, ma'am.
Yeah, you can go present over it.
Yes, ma'am. Ma'am, is my screen? You can see my screen?
Yes, Arish. We can get that.
Yes, ma'am. Good morning, everyone. Oh, sorry, good afternoon, everyone. My name is Harish, and I am from Sardar Bhagwan Singh University. And for today's presentation, my topic is piezoelectricity, an emerging approach against the antimicrobial resistance. So, what is piezostimulation? Piezostimulation involves the application of charge which accumulates on the surface of mechanically defined material, thus affecting the potential in cell envelope. It contribute to deassembling and destroying the bacterial cell wall envelope, thus achieving antibacterial effect. So piezostimulation is a well-known approach that used to use piezoelectric polymer as well as inorganic materials for tailoring antimicrobial production as well as to avoid the bacterial resistance. Compared to the electrostimulation, the piezostimulation has a self-powering activation by stimulating, producing charge upon the mechanical deformation and does not rely on the external electrical source and application of electrode. As an antibiotic-free approach, it used physical and stimuli to target bacterial cell non-specific, which may be greater importance, particularly when avoiding the antimicrobial drugs. This shows significant result against gram-positive as well as the gram-negative bacteria by making them a broad-spectrum therapy against the various bacteria. As we come forward to the methodology, various research articles and reviews articles were studies and polymers inorganic piezo materials was collected for the decodation and fact which are compiled and sequenced. As you can see in the figure 1, these are the two type of two types of piezo cells which are used. First one is the direct, and the second is the convective piezoelectric effect. As you can see in the second figure, the first one is the smooth polymer piezoelectric polymer, and the second one is the nanostructured polymer. As compared to the smooth one, the nanostructured polymer piezoelectric crystal is able to attach more bacterial cell and able to kill them. As we move forward to the result, the smooth and nanostructured piezoelectric polymers like polyvinyl fluoride, poly-L-lactics were studied under the piezostimulation therapy, and the result suggested that the enhanced in the structural properties and better crystalline, causing the betterment and the piezoelectric property, and consequently a stronger bactericidal effect. When we move forward to the piezo materials, the formation of ROS as well as well as cell membrane disruption both comes in the mechanism of inorganic piezo materials. As we come more forward to the conclusion, after a thorough review, series articles, and it is anticipated that the piezoelectric polymer like polyvinyl fluoride, poly-L-lactic acid, and inorganic piezo material like calcium titanium oxide and calcium titanium dioxide have a high degree of use for therapeutic usage after the mechanical stimulation, and the parameters have been optimized. For the improvement in the future, adhesive molecules increase the bacterial attachment to the surface of the film and is anticipated for the further increase in their efficacy. These are the three references which I have used for making this presentation, and I would like to acknowledge Sardar Bhagwan Singh University as well as the ITS College of Pharmacy for supporting me in the making of the presentation. Thank you.
Yeah, thank you so much, Harish. So the next presentations will be going along with Vijay Kumar. And I would like to also note that on behalf of Vijay Kumar, Dr. Ramya Bharati will be going over it. Ramya Bharati ma'am, or you on the meet?
Yes, ma'am. Yes, ma'am.
Yeah, so kindly go over it, ma'am.
Okay, ma'am. Is it my screen is visible, ma'am?
Yes, ma'am. Your screen is visible. You can go ahead.
Okay. Good afternoon, everyone. My topic is on about development of colon targeted controlled drug delivery system of capacetamine. It was presented by Mr. M. Sai Kiran and V. Ramya Bharati from second M.Pharm Pharmaceutics under the guidance of Mr. Volati Vijay Kumar Sir, M.Pharm PhD, Assistant Professor, School of Pharmacy from Satyabama Institute of Science and Technology. This was the abstract of about the purpose of current work was to develop colon targeted controlled drug delivery system of capacetamine using slight modification of post-TM technology. In this technology, it protects the drug during its passage through stomach. Tablets directly compressed with the drug and polymer in tablet press. The capacetamine tablet was prepared mixing of polyethylene glycolated drug powder, guar gum, magnesium stearate, and talc respectively. Tablets were coated by dip coating method with a polymer solution of Udragit S and L 100 in the concentration of 10% and 20%, and solvent was used as isopropyl alcohol and distilled water in the ratio of 5:1. The capacetamine is released from the coated tablets for different batches. It was studied in the 0.1 NaCl at the pH of 1.2 for the first hour, and phosphate buffer solution of the pH at 6.8 for next 4 hours. After that, same tablet was studied for release profile in poly phosphate buffer solution pH at 7.4. Release the profiles showed that the drug release was possible only in phosphate buffer solution at pH 5.0, that is colonic medium. The above test result showed that enteric and cationic coatings were able to bring the core tablet in colon. The aim of the work is capacetamine is a orally administered chemotherapeutic agents used in the treatment of advanced stage of colorectal cancer. The aim of the present study is focused on the formulation development and evaluation of pH sensitive polymer coated guar gum matrix tablet of capacetamide for treating colorectal cancer, which enhance the bioavailability, reduce the dose, minimize the side effect, and sustained drug release for 24 hours. The pharmacokinetics of this work is elimination half-life of capacetamine is 0.55 to 0.89 hours. The peak plasma concentration of 3 to 4 mg per L, and the time to reach the peak plasma concentration is 2 hours. And this was a flowchart of Coach TM technology. Methodology of this work is polyethylene glycolation of capacetamine of the drug was done by solvent evaporation method with the drug to polyethylene glycol 2000 in the ratio of 1:0.5. In this method, drug and polyethylene glycol 2000 were added to the common solvent methanol and homogenized for 5 minutes. Solvent was removed using rotatory evaporated and dried at a room temperature. The mixture was pulverized using mortar and pestle and passed through the C40. And preparation of this matrix tablet, the polyethylene glycolated drug powder equivalent to 150 mg of capacetamine and the guar gum in the ratio of 1:0.5, 1:0.75 and 1:1 sufficient for the batch of 100 tablets were passed through C40 and intimately mixed in a poly bags for 3 minutes. Talc and magnesium stearate were added to the blend and thoroughly mixed in a poly bags. The resultant lubricated blend was compressed in a CADMAX 16 station compression machine with a common compression force to obtain a hardness of 5 to 6 kilogram centimeter square using a round biconcave 9.5 mm punches. This was the ingredient used in the formulation of capacetamine matrix tablets. This was a tabulation of cumulative percent of drug released from the matrix tablets. And then this was the tablet of thickness, weight variation, drug content, hardness, friability of the 15 formulation of the tablets. And this was the tabulations of cumulative percent of drug release from the Udragid S100 and Udragid L100. The summary and conclusion was in this present study, capacetamine used as a drug for a choice of the colon targeting. In the market, only immediate release tablets were available for the treatment of colorectal cancer with the dose of 2500 mg per day, due to which dose-related side effect may occur. An attempt to decrease the side effect, capacetamine was formulated to control release-coated tablets for colon drug delivery. As the drug has a less biological off-life, it was polyethylene glycolated with the polyethylene glycolated 2000 to improve the residence time of the drug in the blood, which can avoid the optimization of the drug. Matrix tablets were prepared with a polyethylene glycolated drug and guar gum in the different ratios. However, around 30% of drug was released in the simulated gastric and intestinal fluids, which is not a desired future of colon drug delivery system. Hence, the matrix tablet was coated with the pH-sensitive coating polymer Udragid S100 and Udragid L100 at the concentration of 20% to control the release in the simulated gastric and intestinal fluid. Among all the formulation, Udragid S100 coated tablets with coating concentration of 20% fulfilled objectives in the investigations by controlling the drug release in stimulated gastric and intestinal fluid and prolonging the drug release in the colonic fluid for 24 hours. In this present work, colon-targeted capacetamine matrix tablets were designed for the first time for treating the colorectal cancer by polyethylene glycolation of the drug. Further scope of the work includes estimation of pharmacokinetic and pharmacodynamic parameters of the drug using established in vivo models. And these are the references. Thank you, ma'am.
Yeah, thank you so much, ma'am, for presenting over it.
Okay.
So I would like to call the next presenter, Mr. Shyam Sundarasan. Shyam, am I audible to you?
Yeah, ma'am.
Yeah, can you please share your PPT and start the presenter?
Yeah. Yeah, my slide is visible to you.
Yes, we can get that.
Yeah. My name is Shyam Susan from School of Pharmacy, Satyavama Institute of Science and Technology, Chennai, Tamil Nadu. And I am from Department of Pharmaceutics, second year. Today I'm going to present a topic on advances in drug delivery system and challenges and future directions of the DDS. First of all, DDS is a technology system which formulates and how to store the drugs molecules and into the suitable forms like tablets, solutions for administration. so they can reach the drug to the specific targeted sites. So the maximum, the therapeutic effects and efficacy of the targeting agent accumulates in the body. Drug have various routes. It has various routes through which they can be introduced into the body. So they can be introduced in various drug conjugate, polymeric conjugate, ionic complexation, and surface coating. So they include but are not limited to the oral route of the administration. On the review article, all the research article has been conducted on a particular combination of the drug delivery approaches, and it has been focused on this study has been focused on that the how the cells in combination with the nanoparticle reported to the DDS systems, and what are the advances has been shown in the cells from more effective than the nanoparticles of the drug delivery system. So the cell-based therapy showed that the improvement of the drug efficacy, extend off-lives, sustained release of the drug, and what are the limited immunogenicity and cytotoxicity of the drug. From this diagram, you can see hyaluronic acid. From this drug, it has been separated into drug conjugate. It has been into the small particle form. The polymeric conjugate, it has been in the spherical, and inside that, the all the particles, the red colors dotted has been there. Next is ionic complexation. The next one is surface coating. Around the spheral, the surface has been coated with a branch of it. Yeah, in that itself, EPR effect and what are the targeting agent of the cell molecules has been done, and what are the binding agent of the protein binding of the agent has been done. Through this, hydraulic groups and carbonyl groups also will take place in this process. So next the RBC blood vessels will pass to the effect of EPM cells and the plasma cells, and multiple hypotonic treatment will take place from the white blood cells to the RBC vessels. Through that, the nanoparticles will take place as sonication, polymerization, electroporation, and co-extrusion. So from this, what are the challenges we can face, and what are the future that is means the drug delivery and nanomedicine have become a very fascinating area of the research in the modern science. In the past years, it has been got a lot of attention in both research experiment and in the number of clinical trial. DDS also plays a major role in the clinical trials in the recent times in the great potential, and it could request the collaboration across the also the academic theory, laboratory experimentation, and acknowledgement of medicine, pharmaceutical, and great research to help and achieve the efficiency, and how we need to take the findings from bench to besides, and how to build the uses of cell therapies, and how long we can go to solve all the biocapabilities issues to the DDS, that is drug delivery system faces. So it also thinks it is a great signal dose that is effectiveness avoid a high accumulation of drugs in this system. And these are my references. Thank you for giving me this opportunity to present in this international conference. Thank you once again.
Yeah, thank you
Hello.
so much. Yeah, thank you so much, Shyam. We're also very much happy to have you. So thank you so much. So the next presentation will we go ahead with Jyoti.
Good evening, everyone. Is it visible?
Yeah, it's fine, Jyoti. You can go ahead.
Okay. Good evening, everyone. I am Jyoti Guleria, PhD scholar at Lovely Professional University, India. The title for my poster presentation is Isolation and Characterization of a Novel Protease from Bacillus clausii strain UBBC 07. Abstracted introduction. So, acute bowel movements or diarrhea, it's an infectious disease associated with the higher number of the outbreaks and fatalities every year. Different bacterial as well as viral pathogens are associated with the disease. And one of the major reasons for the disease persistence so far in years is the multiple drug resistance. Bacillus clausii is a probiotic strain used in the treatment of diarrhea. The consumption of the Bacillus clausii spore is reported to restore the normal functioning of the gut microbiota and cure the gut dysbiosis process. Researchers have reported the extracellular secretions from the Bacillus clausii strain have played a great role in clearing the gut dysbiosis in humans and different organisms. In our study, we have used Bacillus clausii strain UBBC07 to isolate a novel protease having a strong hydrolytic and antimicrobial activity against gram-positive and gram-negative bacteria. The strain was optimized under variable conditions, and we were able to obtain a novel protease. Further, in vivo and in vitro studies using the purified protease were performed to check its bactericidal impact against multiple pathogens. Materials and methods. So the bacterial strain, it was optimized for protease synthesis. It was grown for 96 hours at 30 degrees. Later, sulfate supernatant, it was acid precipitated and was run on STS page. We excised the target protein band, we purified it, and then analytical, physicochemical, and in vitro analysis of the protein was took place. And in vivo studies using Drosophila melanogaster as model organism were performed. Results. First image from the figure A, you can see the acid precipitated protein run on 15% ricin gel. We've used a gel overlay assay with the Bacillus cereus and Salmonella enterica, 1 gram positive and 1 gram negative bacteria to confirm the bactericidal impact of the protein. So from there, we were able to match the particular protein with the protein ladder, and we purified it later on. The figure 2, it shows a purified protein image. From here, we taken the protein for mass spectrometry analysis using Marlitov, and we were able to identify the protein from in DINB protein family of Suchella clausii. But only 36% of the protein sequence was identified because most of the enzyme they did not digest it, which ultimately concluded that this could be a novel protein with novel sequence. So we weren't able to identify it with the existing proteases as well. The purified protein, it was checked for stability at different temperature, pH, and with different detergent. The protein was found to be stable up to the temperature of 70 degrees, where it retained 60% of the activity. But at the autoclaving temperature of 121 degrees, where it was kept for 30 minutes, it completely lost its activity. Similarly, with the pH, at lower pH between 3 to 9, it had a better stability, where 7.2 pH was taken as a control pH. And later on, it slowly lost its activity, but not like completely lost the activity, but reduced its activity toward alkaline pH. We calculated the MIC, that is minimum inhibitory concentration for the protein, using 96 well microtiter plate, and it turned out to be 9.78 micromolar for 10 raised to the power 5 CFU per ml for both test pathogens. From here, we also concluded the time kill kinetics, which turned out to be 4 hours for both the bacterial strains. Scanning electron microscopy results, they depicted the degradation of the Bacillus and Salmonella enterica, and in both the cases, the protein was found to be active. Further, the in vivo assays, which were performed using Drosophila, the first image, figure 9, it shows the reduced internal bacterial load and bacterial shedding. So basically, the Drosophila melanogaster was infected with the Salmonella enterica, and then it was fed on protease treated standard fly food for 30 days, and we observed the changes related to clearance of the bacterial pathogen. So within a course of 18 days, 0% infection was observed in case of different Drosophila melanogaster test organism. Similarly, the climbing ability and reproductive ability of the infected flies, they were restored after 15 days time, and they It showed similar result to the control eventually when continuously fed on the protease supplemented food. The oxidative stress in flies, it was confirmed, and we observed that the total thiol content glutathione S transferase activity, which are basically the antioxidant molecules which are released to maintain the homeostasis state in body. They were reduced initially during the pathogen infection, but they were restored and had a similar activity to the control fly enzymes when fed on the protease treated food. Further conclusions made from this study that the novel protein had a strong proteolytic activity. It belonged to the INB protein family. It had a higher stability at almost at 70-degree temperature and with the pH 3 to 9. In vitro analysis showed the protein to be effective, to be bactericidal against a range of gram-positive as well as gram-negative bacteria. And the purified protein, it also cured the Salmonella enterica infection in Drosophila flies. Future scope of the study to study the mechanism of action of the protease on molecular level, further structural characterization by NMR and circular dichromism, in vivo studies of the protease efficacy in mice model for drug safety and data analysis, to study the pharmacokinetic and pharmacodynamic properties of the purified protein. Here are the references and my acknowledgement. I thank Lovely Professional University and the whole staff for giving me the opportunity to do this research. Thank you.
Yeah, thank you so much, ma'am, for giving a wonderful presentation. So the next presenter will be go ahead with Mr. Jafar Sadiq. Sir, am I audible to you?
Yeah.
Yeah, can you please go ahead?
It is visible.
Yes, sir, you can go ahead.
Good afternoon, everyone. Myself Jafar Sadiq from JSS AHR from Mysuru. I'm just my work on functional attributes of native isolates of lactic acid bacteria from human origin, which conducted under the guidance of Anne H. Catherine, Assistant Professor, JSS Academy of Higher Education and Research, Mysuru. So it's on the probiotic, which like in nowadays the modern lifestyle and urbanizations has been lead increase in metabolic and lifestyle diseases, which by consuming proper cooked foods or which affect our gut, that type of foods which it's harms our body. The people has addicting to that more in this generation, which imbalance our gut, and it produces lot of diseases which like diabetes, obesity, and cardiovascular diseases. So in this field, the probiotic is emerging more nowadays, which is a live organism which can restore this balance when consumed in an adequate amount. In this, there are strains include like bifidobacteria, lactobacillus, pediococcus, and lichenostox, and lactic acid bacteria. Now, lactic acid bacteria is especially using in a large scale, in industrial scale, which it has a many helps in immune support, gut health, cholesterol reduction, and potential cancer prevention and other diseases. So my study has focused on the native isolates, which has been isolated by our guide in the lab. So check the probiotic potential of our native isolates and to develop a target probiotic products for the Indian people. So probiotic need for the study, like probiotic, when we consume in adequate amount, it helps in oxidative stress, reduction of oxidative stress, and reduction of cholesterol levels. Diabetes, who are lactose intolerant, they can consume and they can be free from that intolerance and maintain gut health, and maintains gut pH, and protects the body from harmful pathogens and other diseases. So I just work on taken 19 isolates which are extracted from human origin. For that, 19 isolates conducted an antioxidant activity by DBPH method and cholesterol assimilation by TDC and bile salts. Then taking selected isolates conducted antioxidant activity by the food substrate, which is corn, skim milk, and almond milk. Then same isolates has been conducted resistance to hydrogen peroxide and beta galoxidase activity. So here are the results. In 19 isolates along with the controls, we have got 1H, 6A, and 6H, and 7C, and 15B, the highest percentage in time period of 30 minutes and a time period of 60 minutes, where same has the isolates conducted for cholesterol assimilation, where in bile and TDCA, IFS 6E, 10J, and 15B has shown the good results in the environment of bile acids and TDC acid, which organism shown a good results, where it shows that it can reduce the cholesterol levels. And in antioxidant, this 5 organism has shown the good results where they had neutralized the free radicals. So selecting this among these 5 isolates further conducted in fermented skim milk and almond milk and corn milk. In this 7C has shown in almond in skim milk, 7C has shown the good results around 32.5% at 30 minutes and 39% at 60 minutes. Where in almond milk, IF6H at 30 minutes shown 17%, and at the 60 minute 31% has shown by the 7C. So coming to the corn milk, IF6H has shown 54.9%, as same as a 7C. Comes to the resistant to hydrogen peroxide, where I had taken 0.5 molar concentration and 1 molar concentration for the same isolate in the time period of 0 hours, 8 hours, 24 hours, 48 hours, where it ranged at 0.5%, it ranged from 80% to 104.99%, where for 1 molar, it ranged from 89.30% to 100%. So it shows that this isolate has more resistant to the hydrogen peroxide, which they can be neutralized and proliferate in their environment. So has beta-gal activity, where beta-gal activity has conducted by the substrate ONPZ, where same isolate has been taken for further for beta-galase activity, where 6A, 6S, 7C, and 15B shown the results, but IFS 1H isolation doesn't show because of does not produce the galactosidase enzyme. So it's ranged from 72.14 to 156.32 millar units. So summary and the conclusion of this, that antioxidant was shown a good result from lowest range of 9.50% to 76% at the rate of at the time of 30 minutes, as same at 32.95% to 94.35% at 60 minutes. When in cholesterol assimilation rate, it's shown 12.24% to 94.59% with bile, whereas at 19.29% to 90.31% with GDCA acid. Coming to the H2O2 assay, where 0.5 molar showed 80.07% to 104.99%, and for 1 molar 89.30% to 100%. As in beta-gal activity, it's shown 72.14 to 156.32 millar units. So conclusion is that these results highlights the promising bioactive profiles of native lab isolate, setting them up for use in probiotic, functional foods, and pharmaceutical targeted at improving human health. Additional investigation into the underlying mechanism may identify new targets for therapy and tools for diagnosis, advancing scientific knowledge and creative approaches to healthcare. And I thank the authors like JSS Academy of AHR and my guide, Dr. Anachar Kadri, funded by DBT Welcome and for funding and fellowship. And these are the reference. This is, thank you.
Thank you so much, sir. So we would like to continue with the other presenter from Ms. Nihal Ezariga. Ma'am Nihal, am I audible to you? Just kindly give a couple of moments. I think there is some technical issues over there. I request all the participants to hold a moment. I think there is some technical issues. Kindly wait a moment for a minute. Okay, I think Ms. Nihal will be joining us soon. So we will go with the other one, Ms. Nick Norrell. Ma'am, am I audible? Nick Norrell is available here?
Yes, yes.
Okay, ma'am. Kindly share your PPT and go ahead.
Okay.
Yeah, thank you so much, ma'am.
Oh my God. Okay. Let me minimize here first. Can you see my slide? Okay. So hi and good evening, everyone, chairperson and all. It's almost night here in Malaysia. I'm Dr. Nik Nurul Shakira from National Defence University, Malaysia. And I'm going to present a poster on our findings on our study on antibiotic resistance and molecular characterization of Staphylococcus aureus isolates from a military hospital in Kuala Lumpur, Malaysia, comparative profiles of MSSA, methicillin susceptible sensitive Staphylococcus aureus, and methicillin-resistant Staphylococcus aureus. So a brief introduction, this S. aureus is a gram-positive bacterium, significant pathogen in both hospital and community settings that include military cohorts, where it imposes a serious healthcare and operational burden. So these bacteria have virulent properties to adapt to a host and induce diseases such as the pentavalent leukocidin, or we call PVL, and it is associated with severe form and recurrent Staph aureus infection. So this MRSA is referred to the reduced susceptibility of S. aureus to methicillin and also other beta-lactam. But by acquiring SEC MAC elements in the bacterial chromosome. So this SEC MAC includes the MAC-A gene that encodes the methicillin resistance property. So why MRSA is a major concern compared to MSSA in this study and also reported worldwide? It is because due its resistance to many antibiotics that make infections that caused by this organism is difficult to be treated, Currently, we are using vancomycin, the last choice, and increasing morbidity and mortality. So its ability to spread, in addition, its ability to spread both in healthcare settings and also the community, coupled with its virulent factors, exhibits the challenge of controlling and managing outbreaks. So therefore, monitoring antibiotic susceptibility and genotypic traits of S. aureus is very essential to select effective treatments, prevent the spread of resistant strains, and enhance infection control measures, particularly in combating MRSA. So what are the objectives of our study? The objectives are to assess the antibiotic susceptibility profile, the AST, and distribution of virulent genes that I mentioned just now, the PVL, in the MSSA as well as MRSA isolates from a military hospital named Hospital Angkatan Tentera Tuanku Muizzin, Kuala Lumpur, Malaysia, isolated in the year of 2019. So additionally, the SCC MAC type and also SPA type of the isolates were determined in the MRSA isolates. Now let's move on to the methodology. This study was approved by the National Defence University Malaysia Ethical Committee, and the patient demographic data were collected and stored confidentially. All clinical isolates of S. aureus in the year of 2019 mentioned just now, obtained from various isolation sites of patients from the hospital, included in this study. So the isolates were identified using standard bacteriological method and then subjected to AST profiles determined by using the Kirby-Bauer method based on CLSI 2019 guidelines. And then it was subjected to extracted DNA and to extraction of DNA for the molecular characterization. That include the detection of 16S RNA gene and also species-specific gene for the identification of the S. aureus, and also the detection of methicillin-resistant gene, the MAC-A gene. And lastly, the PVL gene via polymerase chain reaction, PCR, using established PCR protocols with slight modifications. For MRSA isolates, the SCC MAC and SPA type were performed using PCR and also sequencing methods. And then all the collected data was subjected to descriptive analysis to compare patient demographic data with antibiotic susceptibility profiles and also genetic characteristics of MSSA and also MRSA. So for the results, as you can see here, this figure 1, a total of 209 Staph aureus or S. aureus isolates were obtained from various clinical samples collected from patients admitted to this Hat Mizan hospital. Of these, 223 isolates were MSSA, and then the rest, 6 isolates were MRSA. And fortunately, no vancomycin intermediate or resistant Staph aureus was isolated. And as you can see here in table 1, both MSSA and MRSA were mostly isolated from patients aged above 40 years old. And for the molecular parts, all isolates were confirmed to be S. aureus by detection of 16S RNA gene and also NIP gene. And as for the virulent genes, only 42 out of 209 S. aureus isolate, interestingly, positive for PVL, and mostly are MSSA. So representative of DNA band sizes as shown in this figure 2. And for based on the site of isolation, the MSSA isolate were commonly isolated from wound and pus specimens and demonstrated low resistance to all antibiotics tested. In contrast, the MRSA isolates were predominantly from blood specimens and exhibited complete resistance to penicillin, ampicillin, and sulbactam ampicillin in table 2 here. So in terms of acquisition, as displayed in table 3 here, all MSSA isolates 123 were from community-acquired infections, while for MRSA, 60 isolates were from hospital-acquired infections and 70 isolates were from community-acquired infections. And for the analysis, descriptive analysis, we found that there was significant relationship between the methicillin susceptibility with patients' age group as well as the type of acquisition. Second, we found that there was significant relationship between antibiotic-resistant pattern with patients' age group. And lastly, there was significant relationship between the presence of PBL with the patient's age group as well as the isolation site, as shown depicted in table 4. And for the genotypic traits, a total of 9 SPA types were discovered from 85 MRSA isolates, where most MRSA isolates were SCC MET type 4, as shown in this table 5. And of SPA type T032, as shown in this figure 3. So the MRSA SCC MET type 4 and specifically H and SPA type T032 was associated with resistant traits to a few antibiotics such as cefoperazone, azithromycin, erythromycin, and also fusidic acid. Okay, so as for this discussion and conclusion, we know that these type 4 MRSA were associated with community-acquired infection. So for discussion of the conclusion and also conclusion, the study highlights the antibiotic resistance differences between the MSSA and MRSA isolates. And vancomycin resistant Staph aureus has not yet been established in hospital Tuanku Mizan or this military hospital. And so far to date, we are confident there is no vancomycin resistant Staph aureus yet in Malaysia, thus reflecting the adequacy of infection control measures in the hospital specifically. So the judicious use of antibiotics is very important to prevent the emergence of multi-drug resistant Staph aureus and MRSA as well. So MRSA prevalence, and based on the site of isolation, MRSA is prevalent in blood samples, and its resistance underscore the need for ongoing surveillance and infection control. And based on the prevalence of the virulent genes, the majority of Staph aureus or S. aureus isolate that tested positive for the PBL gene were MSSA. This PBL is associated with severe and recurrent infections. Those we suggest that both MRSA and MSSA are of concern. So in terms of predominance of MRSA type, SPA T003 in our isolates, our findings supports previous studies finding where T0 was shown to replace the previous predominance SPA type T037 MRSA clones in Malaysian hospitals, as well as in Portugal, Australia, Ireland, and also Germany. And this also suggests potential clonal spread, highlighting the need for effective infection management in both communities and hospitals. As the conclusion, our study underscored the need for ongoing surveillance, effective infection control, and robust antibiotic stewardship to manage MSSA as well as MRSA, particularly in military settings and also hospital settings. Future efforts should focus on continuous monitoring, molecular studies, community education, and also exploring alternative treatments to combat antibiotic-resistant strains. So these are my key references. So I thank all my team members and the Ministry of Higher Education of Malaysia, as well as the Clinical and Applied Microbiology Secretariat for this opportunity to present. With that, I thank you.
Yeah, thank you so much for giving us a present overview, ma'am. And we are very much happy to have you here. So the next presentation will be going ahead with Sid Habib in the presentation topic of microbiome and human health, correlations and implications.
Hello.
Yes, sir, you're audible.
Sorry. Just give me 1 minute. I'm in a troublesome condition. Just give me 2 minutes. Uh, Hi. Hi everyone. Can you hear me?
Yes, sir,
Hello.
you're pretty good. You're pretty audible, sir. You can go ahead.
Okay. Hi, I'm Said Abid, one of the junior doctor of Lista Hospital, UK, and I'm honored to present one of my part of the research here. So I just start right now, and I'm sorry for and apologize for my inconvenience because I'm in a troublesome condition with my family. If someone is sick, and sorry for that. My presentation e-poster name is Microbiome and Human Health Correlates and Implications. Mm, uh, so uh, Now I start. The human microbiome, a vast collection of microorganisms residing primarily in the gut, has been increasingly recognized for its profound impact on human health. Comprising trillions of bacteria, viruses, fungi, and other microbiomes, the microbiome outnumber human cells by approximately 10 to 1 and contributes an estimated 2 million genes compared to the 20,000 to 25,000 human genes. The symbiotic relationship between human and their microbiota plays a crucial role in various psychological processes, including digestion, immune function, and even mental health. So what's the objectives? There are basically main three objectives behind my research work. The first one is to identify and analyze the key mechanisms by which the human microbiome affects metabolic health. The second is to investigate the relationship between the gut microbiome, diversity, and the prevalence of mental health disorder. And third and the last one is to examine the role of gut-brain axis in the etiology of neurological condition. So let's go to our methodology. Data for this research was collected from a variety of reputable sources, including peer-reviewed journal articles, meta-analysis, systemic review, and major health databases such as PubMed, Science Direct, and Google Scholar. The data we collected was analyzed using thematic analysis to systemic organize, code, and analyze the qualitative data from the literature. Now, move on to the analysis part. The thematic analysis revealed mainly the four keys. The first one is metabolic health, linking gut microbiota to obesity and diabetes. The second is mental health, associating gut microbiota with depression and anxiety. Third one is gut-brain axis, exploring its impact on neurological health. And the fourth and last one is microbiome modulation strategies, evaluating interventions like diet and probiotics for health environment. So let's move on to our results of findings. The analysis revealed significant associations between the human microbiome and various health aspects. So in regarding metabolic health, lower gut microbiota diversity was linked to obesity and type 2 diabetes, suggesting that the microbiome modulation could be a therapeutic strategy. In mental health, differences in gut microbiota profile are profound between patients with depression or anxiety and health controls, highlighting the potential for microbiome-based intervention. Now, the gut-brain axis. So regarding the gut-brain axis was crucial in neurological condition with specific microbial pattern linked to the autism spectrum disorder and microbial metabolites influencing brain function. And the last is modulating the microbiome through diet, probiotics, prebiotics, and fecal microbiota. Transplantation showed promise in managing chronic disease, although personalized approaches and further research are needed to develop the targeted treatment. So finally, the research underscore the critical role of the human microbiome in influencing metabolic health, mental health, and neurological condition. The analysis revealed consistent associations between gut microbiota diversity and various health outcomes, suggesting that alterations in microbiome composition can significantly impact the disease risk and the management. And thank you. Hello.
Yeah, thank you so much, sir.
And I'm again apologize because I'm in a crowded condition because I'm in a really troublesome condition. I'm really apologize for that.
Yes, sir, we can understand. No problem in that. Anyway, we thank you a lot to be a part of this conference and presenting your paper here. So thank you so much, sir.
Thank you so much.
Yeah, thank you.
Thank you so much. Okay, bye.
So I think our Ms. Neha has been joined the session. So we'd like to call the Ms. Neha to present over it. Sorry for the inconvenience. I think still there is some technical issues facing from our side. So I think we can go ahead with the other presenter. Yeah, so the next presenter will be going ahead with Dr. Sri Dharini Mungala. On behalf of Dharani Muhargala, we would like to have Akhil to present a presentation here as a co-author.
Good afternoon. Is my screen visible?
Yes, Akhil, you can go ahead.
Good afternoon, everyone. I'm Dr. Akhil Balarao. Today's topic of presentation is identification and differentiation of Candida species from various clinical isolates through conventional methods in the era of molecular diagnosis. Candida species is a commensal of the skin, mucous membrane, and gastrointestinal tract. They are endogenous opportunists which cause secondary infection. The incidence of Candida infections has increased substantially in last few decades due to the increased use of broad-spectrum antibiotics, immunosuppressants, and indwelling devices. The study aims to speciate Candida using conventional methods. A total of 54 Candida isolates from various clinical samples were included in the study. These isolates were subjected to gram stain, germ tube test, urease test, and inoculation on commercially available chrome agar media. Gram stain was performed from direct samples and inoculated on Sabara dextrose agar, incubated at 37 degrees C for 24 hours. Chlamydospores formation was detected with Dalmor technique. All the isolates were subjected to a sugar assimilation test for final confirmation of species. Germ tube test. The germ tube test is considered as a simple, economical, and efficient procedure for differentiation of Candida albicans from other species of Candida. It is based on the observation that Candida albicans produces tube-like structures called germ tubes. 0.5 ml of sheep or human serum into a small tube is placed. Using a pasture pipette, touch of a colony of Candida and gently emulsify it in the serum. Incubated at 2 to 3 hours at 35 to 37 degrees C. Then it is transferred into a serum, into a slide, and the cover slip is placed, and this is examined microscopically under low and high power objectives. A short hyphal or filamentous extension arising laterally from a yeast cell with no constriction at the point of origin. Germ tube is half the width and 3 to 4 times the length of the yeast cell, and there is no presence of nucleus. Germ tube test provides a rapid identification test for Candida albicans. Positive control used was Candida albicans. The ATCC strain was 10231, and negative control used was Candida tropicalis. ATCC strain was 13803, and Candida glabrata, which was 2001 ATCC strain. And this is a picture where we can find the germ tubes. Next technique was Dalmor technique. Certain Candida species like Candida albicans, Candida dublinensis, and a few strains of Candida tropicalis produces chlamydospores on nutritionally deficient media like corn meal agar. Few saprophytic, non-pathogenic Candida species like Candida australis and Candida clauseni also produce chlamydospores. Chlamydospore formation test is more time consuming than germ tube test. With the Dalmo technique, large and highly refractile walled chlamydospores may be seen at the terminal branches or on the short lateral branches of Candida albicans. Although cornmeal and rice extract agar are best known chlamydospores, including media, a number of other growth media have been suggested and indicated for chlamydospore formation, such as casein agar. It is useful test to differentiate between Candida dublinensis and Candida albicans. Next used technique was chrome agar. High media India was the chrome agar which we have used in a laboratory. The Candida species were inoculated on chrome agar and incubated 37 degrees overnight, and the species were identified by the color of the colonies on chrome agar. So if it was Candida albicans, the color seen was light green. For Candida tropicalis, it was metallic blue. Candida glabrata, white. Candida cruzi, rose pink. Candida parapsilosis, pale green. And Candida dublinensis, it was dark green. The results which we found were a total of 54 Candida isolates from various clinical samples were included in the study. In the current study, majority of the Candida isolates were from urine, that is 50%, followed by sputum, 26%, high vaginal swab was 14%, pus from surgical sites and other constituted 10%. Candida albicans were 28 isolates which we have found and was the most common Candida species, followed by Candida tropicalis, which was 8, Candida cruzi was 7, and Candida dublinensis was 6, and glabrata was 5. Sensitivity and specificity of chrome agar was 100% for both the isolates of Candida, that is Candida tropicalis and cruzi, but the sensitivity and specificity for Candida albicans was 100% and 85.7% respectively. Sensitivity and specificity for Candida glabrata was 66.6% and 100%. For dublinensis, it was 20% and 100%. Discussions. In the present study, Candida albicans is predominant. Predominance of Candida albicans was also seen in a study by Manjunath et al. However, higher incidence of non-albicans Candida, ranging from 54 to 74%, have been seen in numerous studies. Among the non-albicans Candida, Candida tropicalis is reported to be the most predominant species. In our study, also, Candida tropicalis was the most common non-albican species. Conventional speciation of Candida isolates were performed by germ tube test, urease test, Dalmor's technique, sugar fermentation, and assimilation test. They are labor-oriented and time-consuming. Chrome agar is a rapid method to speciate the various species of Candida within 24 to 48 hours. In our studies, sensitivity and specificity of chrome agar for Candida albicans was 100% for both the both the isolates of Candida tropicalis and cruzi. Sensitivity and specificity of Candida albicans was 100% and 85.7% respectively, which was in agreement with Jain N and Mathur P et al. Conclusion. A rapid and accurate identification of the disease-causing species of Candida is crucial for treatment. That is to identify species which might be intrinsically resistant to commonly used antifungal agents. Along with Candida albicans, non-albicans Candida species like Candida tropicalis, Candida cruzi, and Candida dublinensis, and Candida glabrata are increasingly being isolated from various clinical samples. Chrome agar is a rapid and inexpensive method for identification of Candida species. These are my references. Thank you.
Is there any questions for the participants? Okay, thank you. I guess there are no questions from the session chair. Hope every participant has presented their oral presentation and poster presentation. If anyone are yet left, kindly let us know. Okay, I hope there are no other participants left to give their presentation. Now I would like to conclude the session by giving the vote of thanks. Ladies and gentlemen, as we conclude the third International Conference on Clinical and Applied Microbiology, I would like to extend a heartfelt thanks to all who contributed to its success. Firstly, our deepest gratitude goes to our esteemed speakers for sharing their invaluable insights and groundbreaking research. Your expertise and dedications has been the cornerstone for this conference. I also thanks all the session chairs for giving their valuable time and giving the scores for the participants. Lastly, I thank you to all the participants for your active engagement and contribution. Your enthusiasm and commitment to advance in the field of microbiology have truly made this conference a remarkable event. We look forward to seeing you at our next conferences. Thank you once again. Have a great day ahead. Thank you. The certificates for the participants will be shared to your register mail ID in a period of 1 week. Thank you. If you have any further queries, you may contact Ms. Priyanka, the conference coordinator. I repeat, if any of the participant is having any further queries regarding certificates, you may contact Ms. Priyanka. Thank you. Have a good day ahead.
Thank you, ma'am. Thank you, Master.
Thank you, ma'am.
Thank you. Bye.
అంటే వద్దు. చెప్పండి ఉద్దిగా చెప్పండి ఉద్దిగా ఓకే బాయ్ బాయ్ సార్